Hi students, hello. Hello. Hi students, am I audible? Hello, am I audible? Tell me, am I audible? Hi. Am I audible? Am I visible? Am I visible? Am I visible? No. So hi everyone, good evening. Welcome to Unacademy NEET English. Hi. Good evening, good evening, good evening. Welcome to the session, guys. Welcome to the session. Quickly, let me know in the chat section which day it is. Which day it is. Hi. Quickly, let me know in the chat section which day it is. Yes. Tell me, tell me, tell me. Day. Arey yaar, not the 5th November, not Monday, Tuesday, Wednesday, not like this. Game of NEET, dash day. Game of NEET. 39th. Okay. So, welcome to the session, guys. Welcome to the session. Hope you guys are doing very well. Hope you guys are preparing properly for your NEET examination because you are left with 6 months. So, please do not waste your time. And if you are confused that, ma'am, 6 months are sufficient for the NEET preparation or not, so let me tell you, yes, they are. Yes, they are. But the thing is, you have to follow a proper strategy and you have to be consistent for the remaining 6 months. And I am literally not saying that you can get AIIMS, Delhi, you can get this college, that college. But yeah, one thing that I can assure you is that if you start studying properly, consistently, trust me, you can get a good, not good seat. Obviously, you can get your MBBS seat in a good government medical college. Right? Definitely in a good government medical college. So if you really want that, please be consistent. And when it comes to the Game of NEET 2.0 series, obviously it is one of the best series, most popular series, right, bache? You can follow all the lectures and definitely the content is more than sufficient for all of you. Okay? The content is more than sufficient for all of you. Okay, so let's start the session, and today we are going to start with a very basic chapter, and that is cell, the basic unit of life. And let me tell you, here from this particular chapter, you guys can expect 4 questions, means 16 marks. And trust me, this chapter is very simple. It's a very basic chapter. Right, it is a very basic chapter. So we are obviously going to follow the NCERT here, right? No need to do anything extra. NCERT will be sufficient here. So from this particular chapter, right, even last time also one question was there from the cytoskeletal elements. So yes, do target this topic, right? Main functions of all the organelles. Main functions of all the organelles. This is important from this particular chapter, right? Cell membrane is a very important topic. Okay, and what? And what? Endomembrane system, right? Which organelles are the part of endomembrane system? So that type of questions are very important. And when it comes to cell theory, it's a very basic topic that we will complete. So there is nothing to worry about. Okay. Okay. So this is what we have to target from this particular chapter. So let's start the class, guys. And please do not forget to join the batch. The link is given in the description box. Click on that link and you know that you'll be redirected to a page here. See, there is a all-star fast track batch also, but that is in the Hindi. And see, basically this is something very important. You guys will get 55% off on NEET UG subscription. So if you guys are preparing for NEET 2026, right, it is something very amazing for all of you. Even if there is somebody who's preparing for NEET 2027, this offer is going to be amazing. You will have the access of all the batches, all the classes till then. Okay, so you are getting 55% off. Right, 55% off, just imagine. So you can join this batch, right, bache, and there is Nimbus Early Achiever batch as well, right, as I said, for the NEET UG 2027. So without delay, click on that link. It will take you to another page, right? You have to follow the steps and you will be able to join the batch. Okay, so without delay, go for it. And you know that our Phoenix batches are going on, and trust me, the content there is superb. It is amazing. You're going to love that batches. So without delay, be the part of the batch, and please be consistent when it comes to your NEET preparation, because it is the consistency that is going to change your game, right? Keep that thing in your mind. Okay, so now let's start the chapter, and obviously the first thing is cell. We need to know that what exactly is the meaning of cell. So yes, define cell, everybody. Everybody define cell. What do you understand by the word cell? Yes, what do you understand by the word cell? What do you understand by the word cell? Tell me quickly. Yes? Cell, define it. Define it. What is the meaning of cell? Structural and functional unit of life, basic unit of life, okay? Independent existence of performing all the functions, okay? And some students are sharing their Insta account. Keep it up, bache, keep it up. It's good for you, right? There are so many social media accounts and the social media apps, and you are using the YouTube class for exchanging these Insta account and everything. Very good, very good. Go for it, go for it. After a few years, you will realize the importance of this time. Yes, so what is that? So cell, yes, you are right. Cell is the fundamental. Yes, bache, what is cell? Cell is the fundamental structural and functional unit, right? It is the structural and functional unit. Indeed, it is. It's something very simple, right? It is structural and functional unit of life. This is what we know. What is the meaning of that? See, bache, cell is something which is living, And why is it living? Obviously due to the metabolism. I'll take one example. From the chapter Living World. In the chapter Living World, when we talk about the characteristic properties, there are some characteristics properties of the living organisms that living organisms are going to show, and there are some defining properties as well, isn't it? There are some defining properties as well. And when we talk about the defining properties, you know that all the living organisms, without any exception, they are going to show this. And this is something unique. You are not going to see such properties in the non-living organisms. Right, bache? In the chapter Living World, that's what we discuss. We know that there are certain characteristic feature of a living organisms. Just all the living organisms, they will show growth, they will show movement, right, bache? They will exchange the gases, this and that, right? There are some characteristic features, right? But when we are saying, my dear students, when we are saying defining features, when we are saying what? When we are saying defining features, these features are unique to the living beings, right? Non-living organisms are not going to show these features. And without any exception, all the living organisms, they are going to show such features, right? But for an example, when we talk about the growth and the reproduction, these are the characteristic feature. Living organisms are going to show growth and the reproduction. So it is the characteristic feature that living organisms, they grow, they reproduce, but not all the living organisms, they reproduce, right? The growth, it is also there in the non-living organisms also. Isn't it? But when we are saying defining properties, they are unique to the living organism. Without any exception, they are going to show this. Okay, ji? I know some students will ask about the respond to the external stimuli in the coma patient and all. Let's right now, rather than that, let's focus on the main thing here, right? What is the context of telling you this? The context here is that when we talk about the defining properties, we talk about the cellular organization. right? We talk about the cellular organization. We say that all the living organisms, they are made up of cell, right? If something is living, if there is a life, you are talking about the cell there, right? It is the basic unit of life, basic structural and functional unit of life. You people will see that single cells, right? We have the examples, right? We talk about the unicellular organisms. We have the organisms which are single celled and efficiently they are living, right? We have the examples of the organisms which are multicellular, which are made up of many cells as well. So basically, whenever you are talking about a living organism, right, bache, whenever we are talking about any of the living organisms, we will say that they are made up of cell. It is the cell which is the basic unit of life. It is the cell which is making a living body. Are you getting my point? It is the cell which is making a living body. It is a cell which is going to make a living structure, right? Right. So yes, there is one line in NCERT that below the level of cell, there is no independent existence of life, right? So it means that this cell is having everything which is required for being living. Let's take the example of a eukaryotic cell. You talk about a eukaryotic cell, you know that it is having the organelles, right? It is having the lysosome, mitochondria, that Golgi apparatus, like this. Different organelles are there. But let's take the example of this Golgi apparatus. I'm taking it outside, right? I'm taking this organelle outside. Is it able to live? Of course not. Of course not. But when you are talking about this cell, about this living organism, it is having everything to survive. It is, this unicellular organism is having everything to survive. It is having the genetic material, right? It is having the genetic material. It is having what? Cytoplasm. So in simple words, it is having the protoplasm. It is having the protoplasm, and that protoplasm is also, this protoplasm is actually the physical basis of life as per Huxley. Okay, as per Huxley, it is the physical basis of life, protoplasm. So when I'm talking about a single celled organism, it is having this protoplasm, it is able to live. It is able to live. So when you talk about even a unicellular organism, even a single cell organism, it can live, it can survive, it will show the chemical reaction, the metabolism which is required for it. Okay, which is required for it. Is that clear? Is that clear? So that's why it is the basic structural unit. And now one more thing. Let's discuss that. So you know that we have cell, but many cells together, right? Many cells together having similar structure, similar function, and the most importantly, they are having the common origin. That cells together, they form the tissue. So tissue, if I have to define tissue, what is tissue? What is tissue? It's a group of cells. What is tissue? It's a group of cells. Now, group of tissues will form organ. Then we have organ system, right? Then we have an organism. We know this hierarchy, isn't it? Isn't it? We know this hierarchy, that cells together will form tissue, tissues together will form organ, organ together will form organ system, and then we have what? We have an organism. We know it very well. Now, if I'm talking about this tissue, right? If I am talking about a tissue, guys, let's say it is an epidermal tissue or the muscular tissue or the neural tissue, whatever it is. Like, let's say if I'm talking about the neural tissue, we know it is having a specific function. We know it is going to do that job, right? So even in that complete tissue, right, what are we going to discuss? Overall in nervous system, what do we consider? We talk about the neurons, we talk about other neuroglial cells, right? We talk about the epidermal tissue. You know it very well. We talk about the simple epithelium, we talk about the compound epithelium. These are the different types of tissues, right? That tissues are specific. They are performing certain functions. And as I said, tissue is a group of cells. So when you see the cells here, if you will take out the cells, if you will focus on or if you will notice their functions individually, that is different. And all these cells together, right, collectively, right, because all the cells, they are working and collectively, let's say if I'm taking out a cell from this tissue, I'm checking its function, what this cell is doing. Okay. Now, when I compare the function of this single cell and a tissue, it is different also, right? Why? Because this tissue is, whatever this tissue is doing, it is collective effort of all the cells here. It is collective efforts of all the cells here. So even in a tissue, these single cells, they are going to do their job, right? They are going to perform some reactions, right? Which and tissue will collectively show a particular function that will be done by that tissue. We know it very well, isn't it? We know it very well, isn't it? So obviously at the level of cell also, we'll see, right? We'll see, we know about the functions, right? We know that this cell will also show the metabolism, the chemical reactions will be there. I'll give you one example. Just say we talk about the cellular respiration, if you remember, right? We have completed this part now, so I believe you people know this. We talk about the cellular respiration. Even if you want to relate it with breathing and exchange of gases, you know that firstly the external respiration, internal respiration, finally cellular respiration will be there. Right, finally the cellular respiration will be there. I hope you guys are aware. So here we see at the level of cell that how it is going to break the glucose and will take out the energy from it in many steps, right? So we'll see that metabolism there. So for this particular cell, we know, as I said, what is the important thing, my dear students, here? What is the important thing, my dear students, here? We know that protoplasm is there, right? What is there? Protoplasm is there, right? The physical basis of life. Any doubt here? Any doubt here? Gitika, I just started the class and you're talking about the hours, bache. Let's focus on the topic. Let's focus on the topic. Right, so single cell is also capable of independent existence, right? It is performing all the essential jobs. You can take the best example of Mr. Amoeba, the most important and a very simple organism, amoeba. This amoeba can eat, it can move, it can reproduce. We know it very well, right? It can digest, it can excrete, all the functions the single cell can perform, isn't it? Isn't it? Yes or no? Isn't it? Yes? Yes? Doubt? Any doubt here, guys? Yup, tell me, do you have any doubt here? Do you have any doubt here? Yes? So this is about the living organism. So basically when you talk about the living organisms, my dear students, right, obviously they need to work in a delicate environment and they are having the boundaries as well. If you will see the conditions within a cell and on its outer side, you know that they are different, right? A cell is going to, right? I don't know whether you know it or not, right? I don't know whether you know it or not, right? A living cell keep its chemical composition steady, right? A living cell keep its chemical composition steady within its boundary, right? A living cell keep its chemical composition steady, right, within its boundaries, right? Because obviously, and this is homeostasis. What is it? What is it? Yes, bache. What is it? Unmol, unmoli, I think. Right. So ZR, right. So what's the point here? This condition is what? This is the homeostasis. Right, bache? That same conditions will be maintained within a cell. Okay. Okay. Yes, so this is about the cell, right? This is about the cell and this is something that you should know. So cell is the basic unit of life. If you're talking about any of the living organism, definitely they are made up of cell. So we have the single celled organisms also, we have the multicellular organisms also, and when we talk about the multicellular organisms, we will even see that some are having tissue level of organization, organ level of organization, organ systems are also there, right? Let me give you the example here. So I'm going to relate this part a bit with the diversity portion. So you know that as per R.H. Whittaker, I hope you all are aware of it, right? You know that R.H. Whittaker has given us what? 5 kingdom classification system, right? He has given us what? He has given us 5 kingdom classification system, yes or no? 5 kingdom classification system, yes or no? And when we talk about these 5 kingdoms, we talk about kingdom Monera, we discuss about kingdom Protista, right? Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia is there. And Kingdom Animalia is there, isn't it? Isn't it? Kingdom Monera, Protista, Fungi, Plantae, Animalia. We have this. We have this. So when you talk about these 5 kingdoms, my dear students, here in Kingdom Monera and Protista, what are we going to see? We will see unicellular organisms, single cell organisms, right? They are present on this planet Earth. They are surviving. They are efficiently doing their job, right? Even in fungi and Plantae, almost, right? Or you can say that mostly, yeah, most of the organisms, they are what? They are multicellular. They are what? They are multicellular. They are made up of many cells. Obviously, exceptions are there. Exceptions are there, right? Exceptions are there. They are multicellular, but they do have the exceptions. Just say in the case of fungi, I'll give you one example of the unicellular organism, your yeast, right? You have the yeast, Saccharomyces. Yeast is there, that is unicellular. In Plantae also we have the example of unicellular algae. But when you talk about the Animalia, Kingdom Animalia, here you will see that all the organisms, they are what? They are multicellular. What are they, my dear students? They are multicellular. What are they? They are multicellular. So Kingdom Monera is there, Protista is there, having unicellular organisms, right bache? But the main thing that you need to focus is that in Monera, you'll talk about bacteria. Okay, means it is the kingdom having prokaryotic cell. Primitive cells are there. Primitive cells are there. And when you talk about the kingdom Protista, what will you see? You'll see eukaryotic cells are there, and they are unicellular eukaryotic cell. What are they? They are unicellular eukaryotic cell, right? Unicellular eukaryotic cell. But if somebody asks you that in which kingdoms you have the unicellular organism, these 2 kingdoms, and here we have exception, overall we'll see the multicellular organisms there. Is that clear? Is that clear? Is that clear? Done? Done? Sure? So that's what you need to keep in your mind. Is that clear? That is what you need to keep in your mind. And now, when we talk about the kingdom Animalia, when you talk about the animals, right? Here you will see cellular level of organization. You will see tissue level of organization. You will see organ level of organization. And you people are going to see organ system level of organization. So basically, even in the kingdom Animalia, there are certain animals which are just multicellular. In their case, even the tissue is not there. They are just multicellular. They do not even have the tissue. Cellular level of organization. In phylum Porifera. In some phylum, just say I'll give you the example so that you can relate it better. Sponges. Sponges are there. Now when we talk about the tissues, we talk about the phylum Cnidaria, also known as Coelenterata, or the Ctenophora. They are just having tissue, means these animals are just having the tissue in their body. They are multicellular, they are just having the tissue. Right, bache? They are just having the tissue. So I'll give you a very famous example of Hydra, which is a Cnidarian, right? Which is a Cnidarian, just tissue. Then your flatworms, platyhelminths, they are organ level of organization. Remaining phylums are having organ system. So basically what I'm trying to say, you can pick up any of the living organism, you will see that basic unit is cell always. Basic unit is cell always. Are you getting my point? Are you getting my point? So start relating each one thing with another, right? Start relating this, that it should not be like, yeah, I'm reading this cell chapter only, right? I'll just read NCERT only. You have to understand the chapter. You have to understand the topic. You should know how to correlate two chapters in a better way. Okay? Okay? Yes, you should know these things. Are you getting my point? Yes, bache, are you getting my point? You should know these things. So now obviously we will also consider the NCERT, right? Right, we will also consider the NCERT. Chaliye, so let's revise this. So cell. So before starting this part, so you should know that who discovered the cell. So you know that we talk about the Robert Hooke. Robert Hooke in 1665. Robert Hooke in 1665 discovered the cell. But that cell is a dead cell, bache. Dead cell. That cell was not alive. It was dead. It was dead. So Robert Hooke. So when you talk about the discovery of cell, we talk about the English scientist Robert Hooke, and he gave that in his book Micrographia. Yes, you should remember this, right? So Robert Hooke in 1665, he introduced the term cell. And in which book? Micrographia. Is that clear? Micrographia. Yes, right? Yes or no? Yes or no? Right? So when we talk about the Robert Hooke, what he observed, see bache, by that time na microscopy was not that advanced, right? He had that very primitive microscope, and what he did, he observed, yes, what he did, he observed, 1 minute. Right? What he did bache, he observed the cork of an oak tree under the microscope. Right, under a microscope he observed, right? He observed the cork of the oak tree under the microscope. And what he called them, just let's say he was like, these are kind of compartments, right? These are some tiny compartments. Okay, some tiny compartments. And you know that in some books it is also referred that they look like the honeycomb, right? They look like the honeycomb. So he observed some tiny compartments, and for that he used the word cellule, right? He used the word cellule. So now that cellule is known as cell, right bache? Right bache? So he examined that slices of cork under the primitive microscope, right? He saw these tiny compartments, right bache? He called them cellulae, and now we call them as cell, right? Now we call them as cell. Are you getting my point? Right? So he gave these these compartment as an appropriate name, and that is the cell, mane chamber, right? Just these, he used the word cellule, cellule, and now this word is cell. He named it like cell. So it means compartments, right? It means compartments, that tiny compartments. What is the meaning of this here, Anamika? It means compartments. Are you getting my point? Are you getting my point? Right? Even he observed that these compartments are acting like a passage to conduct the fluid, because we know that this cork, what is this? It is a kind of dead tissue. It's dead. Right? It is dead. So obviously he didn't observe the living cell, he observed the dead cell. Right? Right? So no doubt we talk about the Robert Hooke, that in 1665 he was the first one to observe the cell, and he coined the term cell. Right, bache? But he saw the dead cell, not the living one. Is that clear? Is that clear? Yes? Yes? Done? Yes, bache? Sure? Sure? Okay. So now we have to talk about the another scientist. His name is Anton von Leeuwenhoek. Right. So obviously after this cells discovery, you know, scientists, they were even studying the microscopy, right, bache? So by the time of Robert Hooke, the microscopes were not that advanced, right? They were simple kind of. And later on what happened ki some scientists, they were also studying the microscopy. They started developing the microscope, right bache? So here we need to talk about Mr. Anton von Leeuwenhoek, who was a Dutch merchant and he pursued microscopy as a hobby, right bache? So after Hooke, you will say that early microscopists, they start studying the fine structure. Matlab there was some microscopists who start studying the fine structure of these cells, of these living organisms basically, right? So Anton von Leeuwenhoek, जैसे we say ना, he's a Dutch scientist, or right? So he was a Dutch merchant and he pursued the microscopy as a hobby, right? So he was the one who saw the first living cell. When it comes to first living cell, he was the one who saw that first living cell under the microscope. He made a very simple kind of microscope, but obviously better than Robert Hooke, right? That was better than Robert Hooke. So he saw the moving creatures right under that microscope, right? But so they were named as animalcules, right? Just he observed human sperm under the microscope, right? Even the bacteria, right, the protozoans under the microscopes, okay? So he called these living creatures, they were named as animalcules. What is the term here? The term is animalcules. Is that clear? So he was the one who described the free cell, right bache, the moving cell, just human sperm, bacteria, protozoan, RBCs, etc., right, okay? So he called these swimming creatures as animalcules. Done bache? Animalcule. So he was the first one who observed the first living cell. Is that clear? Alright, so different scientists, they noted the living material in that, right? And obviously accordingly they explained. Is that clear? Is that clear? Sure? Are you sure? So there are few scientists' names that you need to remember. I'm not saying all of them, but yeah, few. So there is one more Swiss botanist, right? And he was Robert Brown. So he was the one who discovered the nucleus in the roots of orchid cell, in the roots of orchid plant, right? But in the orchids, he observed that. Is that clear? Is that clear? Sure. So here you can see Anton von Leeuwenhoek, first living cell, Robert Brown, nucleus. Now we'll also talk about the Schleiden and Schwann for the cell theory, Rudolf Virchow. So these are the name of the scientists, right? And yes, in this chapter it is given and we have to remember it, right? We have to remember it, okay? Okay, so we'll come out to it one by one. So this part clear? This part clear? So here you guys can see that unicellular organisms are also capable of, right? Unicellular organisms, they are also capable of independent existence. They are also capable of independent existence and performing the essential functions of life. So this line is very important. Even it is a previous year question, that anything less than a complete structure of cell does not ensure independent living. So anything which is less than this complete structure of cell, having the genetic material, having the cytoplasm, right, it cannot ensure independent living. It cannot ensure independent living. And when we even talk about the unicellular organism, right, be it the prokaryotic one or the eukaryotic one, it is having the genetic material, it is having that cytoplasm. Okay, okay. So cell is the fundamental structural and functional unit of all the living organisms, right? It is structural and functional unit of all the living organisms. So do not forget that Anton von Leeuwenhoek, he first saw and described a live cell, right? He called that moving creatures, the swimming creatures, as animalcules, right? What was that? Animalcules, animalcules, animalcules. Write down this word in the chat section. Quick, and quickly hit the like button, guys. Hit the like button as well. And do subscribe to this channel if you are new to this channel. Done? Done. And I think I used the incorrect thing here. Yes. So Robert Brown was a Scotch botanist, right? He was a Scotch botanist. Chaliye. So Robert Brown, yes, Scotch botanist. So he later discovered the nucleus, the invention of microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. So with advancement in microscopy, we got to know more about the structures of the cell. Is that clear? Is that clear? Chaliye. Next thing that we need to discuss is, bache, it's the cell theory, right? What is it? Cell theory. Obviously, it is important. So when we talk about the cell theory, we need to remember the name of two scientists, right? It's not like that they worked together. No, they worked independently. Okay, it's not like that they worked together. No, they worked independently. But yeah, we need to remember their name, and that is your Matthias Schleiden and Theodor Schwann. So in 1838, yes, even years are important, right? Even years are important here, right? Even years are important here. Okay, so we are talking about the cell theory. Okay, cell theory. What we need to discuss? We need to talk about the cell theory. So basically for cell theory, these are the two names that you need to remember: Schleiden and Schwann. Schleiden and Schwann. Okay, MJ Schleiden या Matthias Schleiden or Theodor Schwann. Matthias Schleiden or Theodor Schwann. So when you talk about the cell theory, these are the two names that you need to keep in your mind: Schleiden and Schwann. And here, and this is again a PYQ, right? Schwann the sheep, or Schwann on zoologist. He was a zoologist, and Schleiden is a botanist, right? So obviously, ideally, if Schleiden is a botanist, he should observe the plant cell and Schwann animal cell, but it is not like that. Schwann even observed the plant cell. That is what we have to discuss. Okay, Schwann was like that, neighbors, that CCTV aunties. Yaar, okay, I'm a zoologist. I need to study the animal cells, but I will also study the plant cell. I'll tell the other botanist, see, I'm being a zoologist. I know this, I know that. Okay, chalo. So let's talk about it. Again, let's consider the NCERT, and as I said, the scientist's name is important. Right, the scientist's name is important. So in 1838, right, Matthias Schleiden, a German botanist, obviously he's a botanist. So he did what? He observed, or he examined large number of plants and observed that all plants, right? What he observed, bache? He observed that all plants, yes, 1 minute, that all plants are composed of different kind of cells, which form the tissue. So ultimately, when he observed the plants, he was like, cells are there, right? What is there? Cells are there without any doubt, right? All the plants are composed of cells. And then, bache, Mr. Schwann, right, after a year, in 1839, right, a British zoologist studied different types of animal cell and reported that cells had a thin outer layer. But first thing, yes, even here you need to know that which line was said by which scientist. Okay, so that is why it is important for you to know that Mr. Schleiden, he was a botanist, and Mr. Schwann the sheep, zoologist, right? Zoologist. So he was the one who said the cells are plants are made up of cells, and that cells are making the tissue, right? And when you are talking about the Schwann is like, he studied different types of animal cells, different types of animal cells he studied, and he was like that in the animals there is a very thin layer. There is a very thin layer that is covering it, and today we know that layer as plasma membrane. We know that layer as the plasma lemma. Right, bache? So he was that neighbor auntie, that CCTV auntie. He also observed the plant cells. Do you know that? He also observed the plant cell. Right? Being a zoologist, he was not like, yeah, I will just observe animals. He was like, no, I'll see what Sharma ji's beti is doing. Right? What Sharma ji's daughter is doing. So basically, he also studied the plant cell and he observed, right? He observed that the presence of cell wall is a unique feature of the plant cell. So in the NEET examination, they can give you this line and they can ask you this thing. Right? They can ask you this thing that who gave this? Yeah, right? It is as per Schleiden or as per Sean that there is a presence of cell wall is a unique feature of the plant cell, obviously by Sean. Right, so Sean proposed the hypothesis. So again, if you will practice the question in the Ames, right? Even if you will solve that Ames PYQ, you will get this question, right? That Sean proposed the hypothesis that the bodies of animals and plants, they are composed of cells and product of cells. What is the meaning of product of cells? Just say tissue, they are made up of cell. Organ, ultimately, so they are made up of cells, no? Ultimately, all the structures, they are made up of cell. If we are living, it is obvious, right, that cellular organization has to be there. Without any doubt, it has to be there. Okay, so after a year, after this Schleiden thing, right, the Sean, he said that. So, bache, Schleiden and Sean together, right, together, then they formulated the cell theory. So that is why the name of the scientist is important. So this theory is like, right, what this theory is saying, this theory is saying nothing. It is saying, right, it is saying that all living organisms are composed of cells, right, bache, and cell is what? It's a mass of protoplasm having a nucleus and bounded by a cell membrane and in some by cell wall also. Right, so no doubt in the NCERT that points are not given, but if I have to tell you, so obviously, right, what that cell theory is saying? Cell theory is saying that all living organisms are made up of cell. So cells are having an outer covering. And today we know that as plasma membrane. By that time, they were not clear. So cells are having what? They are having a thin outer covering, right? They are having a thin outer covering, right, bache? Yes or no? They are having a thin outer covering, right? And and that is the cell membrane. Just a minute, let me charge my laptop. Done. So cells are having a thin outer covering that is enclosing protoplasm, right, and that covering is cell membrane. And in some cells, cell wall is also there. In some cells, cell wall is also there. Are you getting my point? Are you getting my point? Right? Yes? Yes? Tell me. Done? So all living organisms are basically alike in structure and function. And when you talk about, I'm not writing it here, but the function of that organism, right, bache? The function of an organism as a whole is the result of what? Of the activities or the metabolism and interaction of all the living organism. Like if you're talking about of all the cells there, if you're talking about a living organism, right, its function as a whole is the result of what? It is the result of the cells, the function done by that cells, the interaction, and collectively, right, collectively that it is showing the function of that organism. Is that clear? Is that clear? So this is about the cell theory. But here in the cell theory, there is a loophole. There is an alert, and alert. right? There is a loophole, there is an alert here, right? So they gave the cell theory, but they were not able to explain, right? They were not able to explain one thing. And what is that thing, right? That how do cells arise, right? They were not able to explain it, right? So here comes one more scientist, Mr. Rudolf Virchow, right? Mr. Rudolf Virchow. So he gave us one mantra to chant, and you all need to do that. That is Hominis Cellula E Cellulae. What is the meaning of this line? You should know this. They can give you the statement and they can ask you, right? They can give you the statement and they can ask you who gave this. Hominis Cellula E Cellulae, right? Yeah, Rudolf Virchow said this or that. You should know that, right? Just sometimes students, they get confused there. Who gave Contagium Vivum Fluidum? Who gave Hominis Cellula E Cellulae? So please do not make mistake. Okay, do not make mistake there. So Rudolf from hominis. It is starting from hominis, right? Rudolf Virchow. That's how you can remember this. Okay, so they didn't explain that how the new cells they form, right? So Rudolf Virchow in 1855, imagine, Rudolf Virchow in 1855, he explained that he gave us this Hominis Cellula E Cellulae. So what is the meaning of that? It means that cell exists from pre-existing cell, right? So cells, they'll form from the cells which are already existing. That's why we are saying cells arise from pre-existing cells. Cells arise from pre-existing cells. So cell divide and new cells are formed from pre-existing cells. Is that clear? Is that clear? So scientists name these discoveries, they are important. Okay, they are important. Yes, Rudolf Virchow. So write down the name of the scientist. So who discovered the cell? First cell, your first living cell, tell me. Who discovered or described the first living cell? First living cell is the word, Namita. First living cell is the word. Anton von Leeuwenhoek, very good. And when it is who discovered the first cell, that's it. If only this line is given, matlab you are talking about Robert Hooke, who gave the first but dead cell. So if they have specified the living cell, then you have to select Anton von Leeuwenhoek. Okay, chali. So after this Rudolf Virchow discovery, right bache, right, the cell theory, it was modified, right, the cell theory, it was modified, and the modified cell theory is known as cell theory, it was modified. So modified cell theory is known as cell principle or cell doctrine. Cell doctrine. Okay? Cell principle, cell theory, cell principle, cell doctrine, modified cell theory. And it includes what? It includes this point that all living organisms are composed of cells and product of cells, and all cells, they arise from pre-existing cells, right? All cells, they arise from pre-existing cells. Is that clear? Is that clear? Now, bache, they have given the overview of the cell, and after that, individually we have to discuss that which the prokaryotic and the eukaryotic cell. So, bache, overall, if we need to discuss the overview of the cell, I believe you all know about it, right? So, I'll explain it here. Yes, students, I'll explain it here, and then we will just read, or we will just highlight the things. We are going to highlight the things from the NCERT, right? I'm making a rough diagram here only, right? Let's say this is one cell, this is another. Done? Okay, so when we talk about the cell, see, obviously we will discuss about prokaryotic cell. We are going to discuss about a eukaryotic cell. Prokaryotic cell will be there and eukaryotic cell will be there, right? So how can you remember this? This is my way to remember that. From this P word, I remember that P means primitive, right? This is a primitive kind of cell. It's a primitive kind of cell, prokaryotic. So when the word is this, na, karyon, it is associated with nucleus. So actually in this primitive cell, you are not going to see a true nucleus or a well-defined nucleus. You will see that genetic material is naked here. What we are going to see, we will see that there is the naked DNA. There is the naked DNA. It is not, this particular cell is not having a well-defined nucleus. The genetic material is not covered with the membrane. There is no nucleus here, rather than there is a structure that is nucleoid. Here we say the DNA is naked. The DNA is naked, right? But your nucleoid, you can remember it like this. You can remember it like this, that it is devoid. Devoid means without. Devoid histone proteins. Right, so histone are the positively charged proteins on which the DNA coils. But here in this primitive cell, you will see nucleoid is there. It is not the nucleus, it is the nucleoid. It is devoid of histone proteins, right? It is devoid of histone proteins. It is the naked DNA without any nuclear membrane and without that histone proteins. That's what we will see. And here when you talk about a eukaryotic cell, we will see a nucleus, a well-defined nucleus having the DNA. Right, having the DNA, and obviously here we will see the linear DNA. Here we are going to see the circular DNA. So ultimately genetic material has to be there, right? Because it is going to take care of each and every activity of that cell. It is having the all information, right? This genetic material is having the entire information, isn't it? Isn't it? Isn't it? Yes or no? Isn't it? Yes or no? Tell me. Tell me, tell me, tell me, tell me, tell me. Yes, bache. Right, so this is what we have. Okay, this is what, Are, I'm here only, yaar. I'm here only, focus here. So basically what am I trying to say, that whether it is a eukaryotic cell or a prokaryotic cell, right? I'm just giving you the overview. In detail we have to study. So you will see that genetic material will be there. The DNA will be there because it has to take care of everything, right? This genetic DNA, this genetic material is having the information. It will make the different types of from this DNA we will form the different types of RNA, the proteins, right? You know that mostly enzymes, what are they? Most of the enzymes, what are they? They are the proteins. We need them, right? We need them, right? It has to form the structure and everything. And when you talk about this thing, this kind of matrix or what, this is the semi-fluid. What is this? Right, we will see that there will be the semi-fluid matrix, and that semi-fluid matrix is what? Cytoplasm. In the eukaryotic cell, in the prokaryotic cell, as well as in the eukaryotic cell, there will be semi-fluid matrix. There will be semi-fluid matrix that is known as cytoplasm. That is known as what? That is known as cytoplasm. Are you getting my point? Are you getting my point? Right? So semi-fluid matrix, that is cytoplasm. So here also you will see the cytoplasm. Here also you will see the cytoplasm. So it is the main arena of cellular activity. Right? It is what? It is the main arena of cellular activities. Okay, means all that cellular activities will be here in this cytoplasm. So overall, when you talk about a structure, you will see that there will be a limiting membrane. Right, the outer covering will be there. They say here it is cell membrane. Okay, we can also call it plasma membrane. Right, bache? We can also call it plasma lemma. So it is cell membrane, plasma lemma is there. Right, and even in some eukaryotic cells, we will also see that there will be a cell wall also. And now when you talk about the prokaryotic cell, here also you will see the cell membrane, cell wall, and even we will talk about the cell coat. But overall, this genetic material and this cytoplasm has to be there, and a membrane should be there. And as I said, this membrane is important. It is just like a boundary. It is separating this part from the outer non-living, let's say outer non-living environment and living, whatever it is. So it is important for maintaining that homeostasis. Initially I have used this line, I hope you know that, right? Ki living cell keep its chemical composition steady within its boundary, right? So within this boundary, this living organism has to keep its chemical composition steady, right, bache? Right. And that is important, And this is the homeostasis. So this is the basic structure. The genetic material, the cytoplasm, the outer covering has to be there. We'll discuss in prokaryotes the cell wall, how the prokaryotic cell wall is different from the eukaryotic cell wall. Here we will also talk about the cell coat, right? And now when it comes to the eukaryotic cell, so bache, the word is U means true, karyotic, nucleus. A proper well-defined nucleus is here. And here we will see that cell organelles are also present. Right, cell organelles are also present, and that too proper, covered by membrane, right? That too which are properly covered by membrane. That is what we are going to see. Done? Yes? Yes? Okay. Chaliye. So this is the overview, and individually we have to discuss it. So let's read it and let's mark the important points from the NCERT, and then we'll discuss these structures individually. So you know that now they are talking about the cells in an onion peel. They say, I don't know whether you have performed it in your school or not. They say in the case of onion, there is a very thin, film-like structure, right? That is a kind of, I won't say properly transparent, but kind of translucent, kind of very thin film is there, right? So when we stain it or observe it under the microscope, basically what are they? These are the cells, right? So an onion peel we can select, and from the human cheek, like this, we can take the cells, right? But and we can see them, we can observe them under the microscope. So onion peel means you are talking about the plant cell, and definitely it is going to show what? Cell wall, right? And humans, it's an animal cell. It is an animal cell. So, bache, when we talk about the eukaryotic cells, we'll talk about the cells of fungi, we will talk about the cells of plants, cells of animals. So, in the case of animal cell, there is no cell wall. Only plasma membrane is there, but it is a plant cell, and plant cell is having the cell membrane. Okay, so in an onion peel and human cheek, we have to observe it under the microscope. So onion peel is what? A typical plant cell. And what is the beauty here? Right, there are other things also which are going to differentiate a plant cell from the animal cell, but we have to, right, bache, we have to remember this, that there is a distinct cell wall as its outer boundary, and just within it the cell membrane is there, right? There is a cell wall, right? There is a cell wall as its outer boundary, and just within it is the cell membrane. But when you are talking about the human cheek, right, there is an outer membrane only. There is an outer membrane only, the delimiting structure of the cell. The delimiting structure of the cell. The delimiting structure of the cell. Are you getting my point? So inside each cell, you know that a dense membrane bound structure, nucleus is there. What is there? A nucleus is there, and that will contain the chromosome, and that chromosome is nothing but the DNA, right? The proteins, isn't it? Isn't it? The basic structure I told you. Okay, so if there is a membrane bound nuclei, eukaryotic cell, but if it lacks a membrane bound nucleus, then it is prokaryotic cell. And in the case of prokaryotes, as I said, what is the word that we are going to use, bache? We'll use the word nucleoid there. Nucleoid there. We also call it as genophore, right? We also call it as genophore. Okay? Okay, so in both prokaryotes and eukaryotes, there is what? There is a semi-fluid matrix. What is there? There is a semi-fluid matrix. It is not even the fluid, it is semi-fluid. Semi-fluid, that is the cytoplasm, and it occupies the volume of cell. And this line is also a previous year question, my dear students, that the cytoplasm is the main arena of cellular activities in both plant and animal cells. Right? It is the main arena of cellular activities in both plant and animal cells. Is that clear? Is that clear? So various chemical reaction will be there in the cytoplasm, will keep it in the living state. So that is how the metabolism is important, right? So in addition to nucleus, so besides the nucleus means in addition to nucleus, you will see there are distinct structure, the cell organelles, membrane bound cell organelles are there. But bache, there are non-membrane bound organelles also, which are there in a eukaryotic cell. Right, non-membrane bound organelles are also there, which are there in a eukaryotic cell. So when you talk about the non-membrane bound organelle, we talk about the ribosome, right? And we talk about the centriole. So this centriole is present in animals. So yes, you can say that it is absent in plant cell. Few plant cell can have, few exceptions are there in plant cell as well, but overall it is absent in the plant cell, right? So these two organelles are non-membrane bound. They are not covered with any membrane. But let me tell you students, in prokaryotes, this ribosome, it is considered as a universal organelle, right? It is considered as what? It is considered as a universal organelle. Why? Why? Because it is present in both prokaryotic as well as eukaryotic organism. Right? Prokaryotic as well as eukaryotic organism. Is that clear? Prokaryotic as well as eukaryotic organism. Is that clear? Sure? Are you sure? Are you sure? Right? So, endoplasmic reticulum, Golgi complex, lysosome, mitochondria, microbody, vacuole, these are the organelles. So, prokaryotic cell, they lack such membrane bound organelle. So, in prokaryotic cell, the only organelle that is present is ribosome, and that ribosome is also non-membrane bound. That ribosome is also non-membrane bound organelle. Is that clear? Is that clear? So ribosomes are found not only in the cytoplasm but also within the two organelle. Again, in eukaryotic cell we will discuss it in detail. We will see that in the prokaryotic cytoplasm it is 80S type, in chloroplast and in mitochondria it is of 70S type. We'll discuss that. Okay, bache? Okay, so centriosome is nothing. It is basically two centrioles perpendicular to each other plus covered with the pericentriolar material. They are forming the centrosome. So one by one, as I said, again we will discuss it. Don't get confused. So centriole is there in the animal cell, right? It is also non-membrane bound organelle. So two centrioles when they are perpendicular to each other, right? When they are perpendicular to each other plus when they are covered with pericentriolar material. Right, plus when they are covered with, some students, they don't even know the difference in the centriole and in the centrosome. So two centrioles perpendicular to each other, when they are covered with pericentriolar material, they form centrosome, which is also referred as cell center or cell sphere. Is that clear? Is that clear? So that is obviously without any membrane. Okay, that is obviously without any membrane. Understood? Understood? Sure? Yes? Okay, so cells, they are having different shapes, size and their activities. So the information written here is also very important. So they are talking about the mycoplasma, but here mycoplasma is what? It is the smallest cell. It is the smallest cell. Again, a previous year question. And it is only 0.3 micrometer in length. Okay, so mycoplasma is what? It is a only bacteria without cell wall, right? It is the it is the only bacteria, mycoplasma I'm talking about, which is without cell wall. It doesn't have any cell wall. Even in biological classification we discuss about it. Mycoplasma, only bacteria which is without cell wall. So it is a facultative aerobe. Is it facultative aerobe or facultative anaerobe? What is it? Is it a facultative aerobe or facultative anaerobe? Anybody in the class? Yes? Anybody in the class? Anybody in the class? Facultative aerobe or facultative anaerobe? What should be the word? Shraddha, are you sure? Are you sure? Yes? Are you sure? I'm more concerned about this word. Right? Mycoplasma. What is it? Facultative anaerobe or aerobe? Bache, this is the facultative anaerobe. Right? What is the meaning of this particular line? See, mostly mycoplasma, they are facultative anaerobe, means they can survive in both aerobic and anaerobic condition. But the point here is, what is the point here? It is normally aerobic. It is normally aerobic. Normally it needs oxygen for its activity. But if oxygen is not available, still it can survive in anaerobic condition. So facultative, जैसे two words are there, obligatory parasites या facultative या obligatory anaerobes या facultative anaerobes. So obligatory. So how do I remember it? Obligate, obsession, right? Obligate aerobe matlab it is obsessed for oxygen. If it will not get oxygen, it is not able to survive. Obligate anaerobe matlab it is obsessed for anaerobic conditions. If anaerobic conditions are not there, it cannot survive. So if it is facultative anaerobe, means if oxygen is not available, matlab even if oxygen is there, it can survive. But if oxygen is not available, it can survive. That is why facultative anaerobe is the word. So Bacuoplasma is the smallest cell, 0.3 micrometer in length. You have to note it down. Plus, it is the only bacteria without cell wall. So if they do not have the cell wall, obviously their structure is not going to be the same, right? The structure rigidity will not be there. So they will have different cell shapes. So sometimes they are also referred to as jokers of microbiological park, right? They are referred to as jokers of microbiological park, sometimes jokers of plant kingdom, because when they were discovered, they were kept in plant kingdom. That's why. Okay, right? So bacteria overall is 3 to 5 micrometer, but here when you are talking about the mycoplasma, they are the smallest cells and they are 0.3 micrometer. And here in the mycoplasma itself, bache, you talk about PPLO. What is that PPLO? That is pleuropneumonia like organism. Pleuropneumonia like organism, right bache? And this pleuropneumonia like organism is for 0.1 micrometer. It is for 0.1 micrometer. So largest isolated single cell is the egg of an ostrich, right? You have to remember this data. Okay, so among multicellular organism, it is the human RBC, which is around 7.0 micrometer in diameter. Again, these things are important. Nerve cell, you know that as per their function, they look like a tree. They are the longest cell. They are the longest cell. So this information is important, and yes, they do ask the question from this part. So cells, they have different types of shape. They can be polygonal, columnar, cuboidal, regular, irregular, right? So it depends upon the function they perform. So here you people can see RBC, the diameter is 7.0 micrometer. Okay, so that is your homework, right? So in the NCERT, they have given us these pictures, right? So you are going to tell me their dimension. This is your homework. You have to tell me this. Okay, so look at this. It's a tracheid, right? It's a dead cell. Tracheid, it's a dead cell. Sclerenchymatous it is, right? Tracheid. Remember xylem tracheid, right? Xylem tracheid. Xylem vessels, right? So it's a dead cell. It's part of this complex tissue xylem. Okay, mesophyll cells, round and oval they are, right? They are parenchymatous one. Columnar, right? Look at the shape, tall and cylinder, or you can say that long and narrow, right? You can say that long and narrow. And here, look at this. This is the nerve cell, branched and long one. Is that clear? Branched and longer one. Is that clear? Is that clear? Sure? Sure? Okay, so here you guys can see, right, though the prokaryotic cell and the eukaryotic cell, look at this nucleoid, nucleus, the other organelle. Here you will only see the ribosome. Just observe this picture once. Yes, observe it once. Done, bache? Great. Chali, let's go. So, see, in the plant cells also, this is what? This is an animal cell. Why? Centriole is there. The nucleus is there in the center. It is not eccentric, it is there in the center. Right, bache? No cell wall is there. It's a animal cell. It is an animal cell. This is a plant cell. Its nucleus is on one side. It is not there in the center. Why? Because of this vacuole. Right? So in the plant cell, you will see very large vacuole, and that vacuoles are going to move the animal cell to the nucleus to one side. So it will not be there in the center here. It will not be there in the center here. Plus, you will see the chloroplast or the plastids there in the plant cell. You're going to see the cell wall there in the plant cell. Are you getting my point? Are you getting my point? So now we have to cover this. But firstly, let's talk about the bacterial cell, the prokaryotic cell. Okay, let's talk about the bacterial cell, the prokaryotic cell. So again, I am telling you, bache, in kingdom Monera, this kingdom Monera, right, it is having all the organisms having prokaryotic cell. So basically, when you are talking about this, what are you going to read? Bacterias, obviously. Bacteria, or you can say that photosynthetic bacteria, cyanobacteria, also known as blue-green algae. Mycoplasma, the bacteria without a cell wall. Even actinomycetes, that's what we discuss here. Just say eubacteria, true bacteria, right, under kingdom Monera, eubacteria, true bacteria, archaebacteria, right, mycoplasma we discuss, right, blue-green algae, photosynthetic bacteria we discuss. Other than that, even actinomycetes, right, kind of, only this, that is all you have to focus. Okay? That is all you have to focus. Let's not confuse you people. So in the prokaryotic cell, right, so prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO. So this PPLO is 0.1 micrometer. Okay, so they are smaller, multiply more rapidly, and their structure is going to be quite simple if you compare it with the eukaryotic cell. Right, bache? So they multiply very quickly. I'll give you one example. Remember E. coli, Escherichia coli bacteria having generation time of 20 minutes. Right, having generation time of 20 minutes, E. coli. Right, having what? Generation time of 20 minutes means in 20 minutes it will divide into 2 daughter cells. Okay, okay. So they are generally smaller, multiply more rapidly than the eukaryotic cell. They have different shape and size. Okay, so when we talk about bacteria, there are different shapes. Jaise bache, if it is spherical, you will call it coccus, or plural is cocci. So question can come from this part. Just you have the example of streptococcus. So this bacteria is having a spherical shape, right? You can have the Vibrio. It's a comma-shaped bacteria, right? Just Vibrio cholera causes cholera, obviously. Right, you have the example. Then bache, rod-shaped bacteria means cylindrical they are. Bacillus is the word. Rod-shaped bacteria. Right, cylindrical they are. Bacillus thuringiensis is there. Right, bache, and spiralum. Spiral-shaped bacteria. So in the bacteria also you will have the different type of shapes. Is that clear? Right, but the organization is fundamentally similar, even though they exhibit a wide variety of shapes and functions. So these bacteria, their shapes are different, right? They're different. Their even structure is similar. I'm not using the word same, right? Fundamentally they are similar, right? Sorry, structure, the shapes and functions, they are having the wide variety, right? And on the basis of metabolism, on the basis of composition, so we can see there are different types of bacteria. Right, we do not even have that, you can say that we are not even well equipped to check that. That's what I can say. Okay, right? So okay, so we have to discuss it, but firstly, please make sure that you do not miss any diagram here. So ye dekho, typical eukaryotic cell 10 to 20 micrometer, okay, having proper organelles and all. Typical bacteria 1 to 2 micrometer, PPLO about 0.1 micrometer, viruses 0.0 to 0.2 micrometer. So viruses, you know that they are confused kind of creatures, They are even referred to as a connecting link in between living and non-living organism. Outside the body, they are non-living. Inside the body, they are living. So that's what you need to keep in your mind. Is that clear? Is that clear? So let's talk about the bacterial cell. Okay, so you know that here you are going to see nucleoid, you're going to see plasmid, you will see some finger-like extension, mesosomes. These are the words that we are going to read. So you better remember them. You will see ribosomes there, right? We will also see some polyribosomes there. Okay, we will also talk about the inclusion body, the storage granules here. Okay, right, so these are there in the cytoplasm. So when you talk about a bacterial cell, so we are going to start the discussion with the cell coat, right, the outer covering, right? So this outermost layer is cell coat. After that there will be cell wall and then the plasma membrane. So these three, right, these three coverings are basically the outer covering of the prokaryotes, right, Pache? So what is the sequence? The sequence is from outer side to inside. What's the sequence from outer side to inner side? Yes, what is the sequence from outer side to inner side? So outermost is the cell coat, also known as Glycocalyx. Then cell wall, then cell membrane or plasma membrane. That's what you have to keep in your mind, right? That is what you have to keep in your mind. Is that clear? Is that clear? Is that clear? Sure? Done? So let's start the discussion. Okay, so we will focus on the cell wall first. So basically in the prokaryotes, there are three, right? Three outer protective coverings, right? There are three outer protective coverings, right? So again I'm writing this. Cell coat, also known as Glycocalyx, and here we will talk about the capsule and the slime layer. Don't worry, we will discuss this. Then cell wall is there, which is different from the eukaryotic cell wall, and there is the cell membrane, also known as plasma membrane. That is what we have. See, in the case of prokaryotes, we use one thing, that is they are having one envelope system. Do you know that? In the case of prokaryotes, that is the thing that we use, that they are having the one envelope system. Can you define it? What is the meaning here? What is the meaning here? Right? Can you just define it? What is the meaning here? Yes. What is the meaning of this line, that they are having the one envelope system? Yes. Then one envelope system, yeah, single membrane system. But you see, in the case of prokaryotes, you will only see the outer coverings, right? जैसे ये देखो, look at this diagram. You people are only going to see the outer covering, right? Outer covering, जैसे cell wall, cell membrane या फिर glycocalyx. But when you look inside, there is no organelle which is covered with the membrane. Even the nucleus is not covered with the membrane. So only outer coverings are there, right? So sometimes we also call it a single membrane system or one envelope system, that inside, right, only the outer covering is there, but inside no organelle is covered, right? No organelle is covered with any of the membrane. That's why we call it like this, right? Right? Done? Sure, one envelope system it is, right bache? One envelope system is that clear? Right? So bache, we will start the discussion from the inner side first. When you talk about the cell membrane, even its cell membrane is also like the eukaryotic cell membrane, right? We will talk about the phospholipid bilayer, but obviously there are certain exceptions. Just say when you talk about the eubacteria, so when we will start the eukaryotic cell, then we will discuss the cell membrane. So when you talk about the eubacteria and the archaebacteria, eubacteria and the archaebacteria, guys, here also archaebacteria is the primitive bacteria, very primitive bacteria, and this particular bacteria can survive in very harsh conditions, right? It can survive in very harsh conditions as in where the pH, right, where the pH is very low, matlab acidic conditions are there, temperature is high, जैसे thermoacidophiles, methanogens, right bache? You talk about the halophiles where salinity is more. So these archaebacteria, their structural composition is in this way that they can survive under extreme conditions, right? They can survive under harsh conditions. Archaebacteria it is. What is it, my dear students? Archaebacteria it is. What is it, my dear students? Archaebacteria. So here, right, even in the prokaryotic cell, you will see eubacteria and archaebacteria. Definitely in the chapter biological classification, we are going to discuss that. But I want you people, right, bache? I want you people to focus here. Yes, I want you people to focus here. Done? Okay, right? So there also there will be a difference in the cell membrane. So cell membrane and its structure in detail we are going to discuss in the eukaryotic cell. So remember one thing, right, bache? Yes. So in the case of cell membrane, you know that it will control the movement of molecules inside and outside. Overall in the cell membrane, right, you know that it will control the movement of molecules inside and outside, right, when you are talking about the cell membrane, right? This is what we know. And you will see that cell membranes will show some infoldings, right? Some infoldings. We will call it as mesosome. That is what we have to discuss in a prokaryotic cell, that even the cell membrane will show some enfolding, mesosome. Even we will see that cell membrane will also bear some respiratory enzymes. Just this mesosome is having the respiratory enzyme, and this is also the enfolding of cell membrane. Plus, bache, the cell membrane will also have certain receptors there, right? The one thing that you need to keep in your mind is that in the case of the cell membrane, my dear students, in the case of the cell membrane, here, right, no cholesterol is present. I will discuss it in today's lecture. When I will start the eukaryotic cell membrane, I am going to discuss it, right? Right? Done? So, no, in the case of cell membrane of a prokaryotic cell, there is no cholesterol, right? No cholesterol is there. Instead, you will see hapenoids are present here, right? Hapenoids are present here. Hapenoids are present here. Done? Is that clear? Yes. So bacterial cell membrane lacks sterols like cholesterol. So they are having pentacyclic sterol like molecule, that is hapenoid. So it is not a sterol, but it's a sterol like molecule. Right? So as I said, in detail we will discuss the plasma membrane under eukaryotic cell structure, right? But that's what you have to keep in your mind. Okay? Okay, so this hapenoid will also stabilize the structure of cell membrane. That's what you have to keep in your mind. But here mainly we are going to focus on the cell wall. So matlab, we are moving from inside to outside. So there is nothing much that we have to discuss here. We'll cover it under the eukaryotic cell membrane. Is that clear? Then comes the cell wall. Now when you talk about the cell wall of the bacteria of a prokaryotic cell, bache, here you are going to talk about the murines, right? Or you can say that peptidoglycans. What are they? Peptidoglycans. Jaise plant cell wall is having cellulose, hemicellulose, pectin. Here you will talk about the peptidoglycans, and that is NAM, NAG. What's that NAM and NAG? This NAM means N-acetyl muramic acid. It means N-acetyl muramic acid. Right, bache? And this NAG is N-acetyl glucosamine. N-acetyl muramic acid, N-acetyl glucosamine. So what do we have here? In the cell wall, what's the composition of the cell wall? Right, it is having the murines or the peptidoglycans. Murines or the peptidoglycans. NAM or NAG. N-acetyl muramic acid, N-acetyl glucosamine. N-acetyl muramic acid, N-acetyl glucosamine. Right, bache? Peptidoglycans. Done? Right, so peptidoglycans are making the cell wall here, right, bache? So they are N-acetyl glucosamine, N-acetyl muramic acid, and they are cross-linked by, right, short peptides. So these murines or peptidoglycans, they are cross-linked by short peptides. Done? Cross-linked by short peptide. So if you don't know what are these peptidoglycans, you can simply say they are the modified polysaccharides or modified sugars. In the chapter biomolecules, you will understand that thing in a better way, right? You guys will understand that thing in a better way. Guys, I'll give you the break, right? I'll give you the break, but not now. Let's finish the prokaryotic cell. After that, there will be a break. Okay, after that there will be a break. Okay, so this part clear about the cell wall. This part clear about the cell wall. So cell wall is very important because it will protect the cell, right? Right. So cell wall is very important. Why? Because this cell wall is going to protect the cell. Provide cell shape. It protects the cell. It provides the cell shape. Right, so cell wall, it protects the cell, it will provide a shape, and plus it prevent the cell from bursting in hypotonic environment. So what is this hypotonic environment? Yeah, hypotonic medium. hypotonic environment and hypotonic medium. Just say, let's say you are keeping this cell in a solution and that solution is hypotonic. Basically, you are comparing the chemical composition of this solution with the cytoplasm. If it is hypotonic, means it is having more solvent in comparison to the cytoplasm, less solute. So obviously endosmosis will occur, right? This water will move in, and that will result in the swelling of the cell. That will result in the cell swelling, right? Or cell can even burst. So this cell wall will protect this bacteria from bursting. Are you getting my point? Are you getting my point? Yes? Yes, yes, or yes? Tell me, yes. Done? Okay. So guys, you know that when we are sick and we are having the bacterial infection, doctors, they give us antibiotics. They give us what? They give us antibiotics. So that antibiotics, anti means against, bio means life, against life, and life of what? Life of these disease causing bacteria. Antibiotics. So just say there are different types of antibiotics and they behave in a different way. I'm giving you one example of penicillin. Penicillin. You know, this is the first antibiotic to be discovered, and this discovery was also serendipity, by chance discovery, penicillin, right? So Alexander Fleming, he discovered it, but he was not even aware about its full potential. Penicillin I'm talking about. So when you talk about this antibiotic, right, bache, it doesn't allow that peptidoglycan to form. Matlab, these antibiotics, they kill the bacteria in different way. This antibiotic penicillin, it prevents the formation of peptidoglycan. It prevents the formation of peptidoglycan. Right, it is going to prevent the formation of peptidoglycan. That is how it acts. Right. And when you talk about the another antibiotic, just I'll give you the example of lysozyme, and it is going to hydrolyze. It hydrolyzes the peptidoglycan cell wall. One will not allow it to form, one will just hydrolyze it. So that's how these antibiotics, they kill the bacteria. And it is not just the only way. Sometimes, you know, some antibiotics, they can just prevent the transcription, yeah, translation, it is like that. So they behave in a different way. So this penicillin is going to do that. Not just penicillin, even cephalosporin is also behave in the same way, right? And this lysozyme, I don't know whether you know it or not, it is there in saliva, it is there in our tears, right? It is there in our nasal secretions. You know, lysozyme, it is there in saliva having antimicrobial property. It is there in the tears, right? It is there in our nasal secretions also. Is that clear? Is that clear? Is that clear? So this is about the cell wall, right? This is about the cell wall. So sometimes the peptidoglycans will not form, or they will not even be allowed to cross-link with the short peptide. So that is how it works. So this is about the cell wall. So bacteria, they do have the cell wall, but you should remember this, that Mycoplasma is the bacteria, only bacteria which is without cell wall. Which is without cell wall. Is that clear? This bacteria is what? This bacteria is without cell wall. Understood? Understood? Sure? Are you sure? Done? Right, so this is about the cell wall. Okay, so then bache, there is the Glycocalyx, and which is also known as cell coat. It is of two types. That is capsule or slime layer. That is what we have, capsule or slime layer. What are we talking? we are talking about this glycocalyx. But before that, let me tell you, even when it comes to the cell wall now, this thickness and all will also form two different types of bacteria, like gram negative and gram positive. It is also given in your book. And after this topic now, that is what we have to discuss. Let me finish the prokaryotic cells first, and then we will talk about the gram negative and the gram positive bacteria also, because that bacteria, their gram negative and gram positive, two different types of bacteria are there, and it is on the basis of their cell wall, and we have to discuss that. Is that clear? We have to discuss that. Is that clear? Is that clear? So glycocalyx, cell coat, you have the capsule and you have the slime layer. We have the capsule here, we have the slime layer. So glycocalyx or cell coat is outside the cell wall. Yes, bache. Where is it present? It is outside the cell wall, yes or no? It is outside the cell wall, yes or no? Yes or no? Yes. So this capsule and slime layer, they are made up of polysaccharides. Right? Polysaccharides are going to make it, but can contain polypeptides also. So when you talk about their chemical composition, polysaccharides and polypeptides can be there. Polysaccharides and polypeptides can be there. What can be there, bache? Polysaccharides and polypeptides can be there. Now when you are talking about the slime layer, it's a loose, it's a covering, it's a loose. In the gram negative bacteria, you will see this loose gelatinous covering, jelly like it is, right? And it is going to be thick and tough structure. So the loose and jelly like covering is your slime layer, right bache? Loose gelatinous covering is your slime layer, and thick and tough covering is known as your capsule, right? It is known as your capsule, right bache? So they are going to protect. So this cell coat, it protects the bacteria from desiccation, from host enzymes, yeah, etc. Right? So obviously these coverings are meant for what? These coverings are meant for what? They are meant for the protection. Right, bache? So it can protect the bacteria from the desiccation, from the host enzyme, and yes, even from the antibiotics and the virus attack. Is that clear? Sure. Is that clear? That is the function of glycocalyx. Okay, bache? That is the function of glycocalyx. Okay, bache? Yes. Yes, and one more thing you can say that, because see, it is something jelly like, slime layer, right? Gelatinous. So obviously it can also help that bacteria to attach to the surface, right? To attach to the surface. So that is what you can keep in your mind. So this is all about its covering, about its outer coverings, right, bache? It is all about its outer covering. Is that clear? Chalo, let's discuss it quickly. So prokaryotes, they are having a cell wall surrounding the cell membrane except in mycoplasma. So mycoplasma, smallest cell and jokers of microbiological power, right? And they are facultative anaerobes. They are facultative anaerobes. Anaerobes. Define nucleus, that is nucleoid. So next is about the nucleus we are going to discuss. So bache, you know that I'm not going to make the covering again and again. Chalo, let's. Let's say this is the covering, okay? So when we talk about this genetic material na bache, it is nucleoid, right? I told you already about it. the DNA here is going to be circular one, bache, and it is devoid of histone proteins. Means histone proteins are not there in the case of prokaryotic cell. Okay, bache, so DNA is circular as well. Matlab here also the DNA will be coiled. It will also form some loops. Right, it is also going to form some loops, but here there are some other positively charged proteins, but not, right, bache, other positively charged proteins are there, but not histones, right, but not histones like eukaryotic cells. Is that clear? So that positively charged proteins, they can be polyamines. Okay, so it is the nucleoid. Right, so it is circular. So we also use the word genophore for it. Right, genophore for it. What is the word? Genophore for it. Genophore for it. And obviously, it's the circular DNA. What kind of DNA it is? It is the circular DNA, right? It is the circular DNA. So it will make only one chromosome. So we also use the word prochromosome here. We also use the word prochromosome here. Right, prochromosome. It is going to make one chromosome only, circular DNA. It's just like a circle. So we also use the word prochromosome. So you can call it nucleoid, genophore, or prochromosome. Is that clear? Is that clear? And other than this now, bache, you will see that there is extra chromosomal DNA also. What are you going to see? That there is extra chromosomal DNA. And that extra chromosomal DNA is plasmid. Right, this is the extra chromosomal DNA. Dekho, ultimately it is the DNA. It is the nucleic acid. Obviously it is going to have the genes. Right, the genes are present here. Yes or no? Yes or no? I am talking about the plasmid, bache. I am saying this is the extra chromosomal DNA. Done? Yes? So extra chromosomal DNA. It is a DNA, obviously it is having the genes also, and that genes will also express itself. Students, this is something that we read in the chapter molecular basis of inheritance. Basically in the class 12th we are going to discuss that, right? This is your DNA. DNA, we know that gene, it is having the genes, the basic unit of heredity. That DNA, right, it has to, that genes, they need to express itself. And how are they going to do it? They have to make the proteins. They'll make the proteins. So from DNA to mRNA, messenger RNA will form, we call it as transcription, and mRNA will form proteins, that is known as translation. That is known as translation, right, bache? So DNA will form mRNA, that is transcription, right? mRNA will form protein, that is known as translation. That is what we have, transcription and translation, right? So ultimately, right, this is how a gene present on the DNA is going to express itself. It will make the proteins, and that protein, you know that it can have that thing like it is basically, it can be the enzyme, it can form the enzyme, right? It can contribute there in the formation of that enzyme. Anything can be there. It is going to have that different functions. We know it, right, bache? Right, bache? So basically, I am talking about the plasmid. If I am talking about a plasmid, that plasmid is also having the genes. That plasmid is also having the genes, and that genes, obviously they will also express itself. They are also going to make some protein, and this protein is going to give a kind of unique phenotypic appearance, right? Unique phenotypic appearance to that particular cell. Are you getting my point? Unique phenotypic appearance. What is the meaning of this phenotypic appearance? Phenotypic, PH, physical appearance we are talking here. Here, what are we discussing here? Physical, just from phenotype, pH, physical appearance. Just let's say something which you can see. Just for an example, you are looking at a plant and you can say plant is tall. You are talking about its phenotype. You are talking about its morphological form. You are talking about this appearance there, right? So basically, I am saying that when this plasmid, it is also having the genes. That gene will express itself. It will make protein by this flow of information, obviously. So that protein can give the unique phenotypic appearance to that particular bacteria. Just for an example, let's say this plasmid is having the genes which is providing antibiotic resistance to this bacteria. Let's say there is an antibiotic A. That antibiotic A cannot destroy this bacteria because it is having that gene which is providing it the resistance against that antibiotic. It can be drought resistant gene. It can be any other character as well. Are you getting my point? Are you getting my point? So above, and this plasmid, it is the extra chromosomal DNA, or you can say that it can replicate by its own. It is not dependent on this chromosomal DNA. It is having its own origin of replication. So about it and about its role, you will discuss in the chapter Biotechnology, class 12th, right, where you will study that plasmids, they act like a gene taxi. They act like the vectors. That's what we will see there. Okay, so I hope this part is clear. Now, when you talk about the cytoplasm here, in the prokaryotic cell, there is no cyclosis. That is also a difference in between the prokaryotic cell and the eukaryotic cell. Prokaryotic cell, they do not show the cyclosis. They do not show the cyclosis. What is this cyclosis? बच्चे cyclosis माने cytoplasmic या protoplasmic streaming, that movement here, right बच्चे? Cytoplasmic streaming, that movement here, the movement of cytoplasm, which is important for moving that content from one part to another as well. So cytoplasmic streaming is not there. Are you getting my point? In the prokaryotes, the cytoplasmic streaming you people are not going to see, right? You people are not going to see. And when it comes to the organelle, we are going to talk about the ribosome. And what kind of ribosome it is? 70S. What kind of ribosome it is? 70S. I'm writing it here. So ribosome is always like this. Ribosome is basically the protein factory. What is the role of ribosome? What is the role of this particular organelle? It is a protein factory. It helps in the synthesis of protein. And it is a non-membrane bound organelle. It is the non-membrane bound organelle. Okay, bache. So in the prokaryotes, it is 70S. Actually, bache, ribosome, it is made up of two subunits, larger and smaller. It is made up of two subunits, larger subunit or smaller subunit. What is that, bache? It is the larger subunit and there is the smaller subunit. So when you talk about this larger and smaller subunit, right, they will only join with the help of magnesium ion at the time of protein synthesis. If there is no protein synthesis, they'll stay apart, right? So when you talk about its larger and smaller subunit, it is 50S and it is 30S. So this S here is Swedberg unit. It is the sedimentation coefficient. It is what? It is the sedimentation coefficient, right? So I'll explain that to you. So it is having two largest subunit, smaller one. So here there is no need to add it. Madam, 50 plus 30 is 80. Why are you saying 70? No, it's not like that, right? Sedimentation coefficient. Actually, bache, when we study the cell, na, we go for self-fractionation. Matlab, in a centrifuge, right, you are going to break the cell components, right? You are going to rotate it at high speed. Obviously, the different components, they'll also break. As per their weight, they will settle down. I'm not explaining it in detail. I'm just giving you an idea. As per their weight, they will settle down. So this, as per their weight, their density actually, they'll settle down, right? They will settle there. So when it comes to these ribosomes, right, just say in the eukaryotic cell also we have the ribosome. There we will talk about 80S and 70S ribosome. So this S is that sedimentation coefficient. For that we use the unit Swedberg unit, this S. So larger subunit is 50S. It is telling us about their sedimentation, right, bache? This one is 30S. So these ribosomes, they are made up of different type of ribosomal RNAs and proteins. So when we will start the eukaryotic cell, once again we will discuss it. You will have the better idea of this. But the thing that you have to remember is that ribosomes, they are having the 70S. the prokaryotes are having 70s ribosome. Now which is cell membrane also forms some finger-like enfoldings. Just here, in the case of this prokaryotic cell, right Karan? In the case of this prokaryotic cell is not having the mitochondria. It is not having the mitochondria. Then how is it going to fulfill its duty? Yeah, who's going to make the, who's going to provide it the energy? Right, just say mitochondria is the powerhouse of the cell. It forms the ATP. It doesn't have the mitochondria. So mesosome is going to play that role. Mesosome is going to help there. That finger-like enfoldings, they are having the respiratory enzymes. They are playing role in the cell respiration here. Very important thing that you are going to see in the prokaryotic cell. Right, we will also talk about the inclusion body. So we will discuss it from the NCERT here. So what NCERT is saying? NCERT is saying ki genetic material, that genetic material is basically naked. What is the meaning of that naked thing? Not enveloped by a nuclear membrane, plus I told you, devoid of histone proteins, right? It is not having the histone proteins. It is not having the histone proteins, right bache? Right bache? Yes? Yes, so genetic material is basically naked, not enveloped by the nuclear membrane. So, in addition to the genomic DNA, that is the circular DNA or single chromosome, bacteria is having the circular DNA outside genomic DNA. Matlab it is the plasmid that is present there. It is the plasmid that is present there. Are you getting my point? Are you getting my point? So, as I said that this plasmid, it will confers certain unique phenotypic character to such bacteria. Matlab obviously it is also having the genes that are going to express itself and that will provide a unique character to that bacteria. Jaise ki it can be antibiotic resistant, it can be drought resistant, it can be like this. Okay. So obviously we'll discuss about the role of plasmid as a gene taxi in biotechnology. Now what are they saying? No organelles like the ones in eukaryotes are found in prokaryotes except the ribosome. So ribosome is the non-membrane bound organelle. Non-membrane bound organelle, keep it in your mind. Okay bache? Okay bache? Okay. So prokaryotes have something unique in the form of inclusions. Right? So you are going to talk about the inclusion bodies in the case of prokaryotes. Wait, I'll discuss it. So, they are saying they have something unique in the form of inclusions and a specialized differentiated form of cell membrane, mesosome is there, right? So, these are the enfoldings of cell membrane. So, from this part, we are going to discuss it and let's discuss it very quickly. So, most prokaryotic cells, bache, right, particularly the bacterial cells, they have chemically complex cell envelope, right? So, here they have mentioned that they have 3-layered structure. Do I need to explain it again? Do I need to explain it again? But all the 3 layers, they behave together as a single protective unit. They behave together as a single protective unit. Isn't it? Isn't it? Now they are saying ki on the basis of that, we have 2 different types of bacteria on the basis of the cell wall there. That is gram positive and gram negative. So, bache, we will discuss about it. Let me finish the other things in the prokaryotic cell. Then we will talk about the gram positive and negative bacteria. So, as I explained, right, the slime layer thing, the capsule thing is given in your this part, okay, in your NCERT as well. So plasma membrane is selectively permeable. It will take care, it will monitor the entry and exit of the things. It interact with the outside world as well. Okay, done. So a special membranous structure, it is the mesosome, which is formed by the extension of plasma membrane. So these are the kind of modifications of that cell membrane, right? Yeah, formed from that cell membrane. Is that clear? Is that clear? So these extensions in the form of vesicles, in the form of tubules, right, in the form of that lamellae, right, they are present. They will help in cell wall formation, even in DNA replication, even distribution of that DNA to the daughter cells. So again, it is also a previous year question. Are you getting my point? It is also a PYQ. You need to note that. Okay, so they are also helping in respiration, secretion to increase the surface area of the plasma membrane and enzymatic content. So in some prokaryotes, जैसे कि your cyanobacteria, that what is cyanobacteria? It is your blue green algae, right? I hope you remember the example also, जैसे कि nostoc. Okay, I don't know whether you know it or not. Nostoc, right? So these are the photosynthetic, oxygenic photosynthetic bacteria. They produce that oxygen there. Okay, right? So other than this, so in cyanobacteria, because they are photosynthetic, they need to perform the photosynthesis. So in their case, you will see that membranous extensions are also there in that cytoplasm. That membrane will form some structure, chromatophore. So chroma word is associated with color, right? So it contains the pigment. So it is not properly the thylakoid or something, but some structures are like that, right? Some structures are like that. Are you getting my point? Are you getting my point? Okay. So now in the case of bacteria can be motile, that is movable, or immotile. Let's say if it is motile, you will see flagella there. Now this flagella is very simple kind of flagella. Very simple kind of flagella, and it is made up of flagellin protein. It is made up of what, bache? It is made up of flagellin protein, right? Now, even in the eukaryotic cell, I will explain it. When you talk about the eukaryotic cell, there you will see the flagella, and that flagella will show 9+2 arrangement, and that flagella is made up of microtubules, or you can say that tubulin protein. So here it is made up of flagellin protein, but in eukaryotes it is made up of tubulin protein. You should know that. You should know that. And it's a very basic structure. It will be having a filament, this long thread-like part, a hook, and a basal body from where it forms. Just a basal body here, a kind of hook is there, and then there is a filament. Are you getting my point? So it is the longest portion and extend from cell surface to the outside. Okay, extend from cell surface to the outside. Understood? Sure? Are you sure? So in addition to flagella, bache, you will see pili made up of pyelin protein. Jese, let's say this is one cell. You can take the example of E. coli. This is another cell. They are having this tube-like structure made up of pyelin. They are also having in the conjugation. They are also helping in the conjugation. In biological classification, we will discuss it. Right? Matlab, jaise, let's say this tube-like structure, it will join with another bacteria. Right? Here, bache, we talk about some fertility plasmids. We discuss about some fertility plasmids. So basically, it's a method by which, you know, from this tube, it is connecting these two bacteria. This plasmid will pass to this other bacteria also. So it is known as sex pilus. Okay, so it is helping in the conjugation. So it is playing no role in the movement or motility, but in that kind of sexual reproduction, or you can say that genetic recombination, it is helping. Right? So another thing is fimbriae. So they will help this bacteria to get attached to the host surface. So pylae and fimbriae, they are also the surface structure of bacteria. They are playing no role in motility. Matlab, they are playing no role in the movement of that bacteria. Very important thing. So pylae are elongated tube-like structure. They are made up of special protein. I said pyelin it is. Fimbriae are bristle-like fibers. They'll sprout out of the cell. So in some bacteria, they are known to attach the bacteria to the hooks in stream and also to the host tissue. I'll give you one example. Let's say we talk about Neisseria gonorrhoeae. Right, it causes gonorrhea. What's that gonorrhea? It is there in your syllabus. It is an STI, sexually transmitted infection. Sexually transmitted infection it is. It is STI. So this particular bacteria, with the help of fimbriae, right, with the help of these fimbriae, it will attach to this. It will attach to the host tissue, right? I'm giving you one example here. It will attach to the host tissue. Is that clear? Baki bache, ribosome, as I said, they are about 15 nanometer by 20 nanometer in size. 50 and 30 subunit is there, right? And they are the site of protein synthesis. Now, see, in the case of bacteria, here you have the genetic material DNA. no nucleus or nothing. So the phenomenon, this transcription and translation. Transcription or translation. What is transcription? Transcription means your DNA is forming the mRNA. And when we translate that mRNA so that we can get the protein from it, it is the translation. It is the translation. So both the things in the case of a bacteria, in detail you will read about it in class 12. Both the things are taking place in cytoplasm. Both these things are taking place in cytoplasm. So in the case of bacteria, we see that, let's say this is the mRNA, matlab messenger ribonucleic acid. So to this messenger ribonucleic acid, ribosomes are attached. Many ribosomes, they are attached to that single mRNA, right? And what is happening? Simultaneously, the protein synthesis is also going on. Just in the case of prokaryotes, we say that transcription and translation, they take place simultaneously. They are not separated by any location or something. They take place simultaneously. Okay, ji? So here, to one mRNA, many ribosomes are attached and simultaneously protein synthesis is going on. So, bache, this structure is polysome or polyribosome. What do we call it? We call it as polysome or polyribosome. So many ribosomes, they are attached to single mRNA. They are forming a chain, polysome or polyribosome, right bache? And inclusion bodies, in simple words if I have to tell you, they are the storage granules. Again, they are non-membrane bound. Again, they are present in the prokaryotic cell. Something unique here in the prokaryotic cell, right bache? So the reserve material, right, reserve material in prokaryotic cell is stored in cytoplasm in the form of these inclusion bodies, not bounded by any membrane system, and they are freely lying in the cytoplasm. So phosphate granules, cyanophasian granules, glycogen granules, they all are present. So gas vacuoles are also there in blue, green and purple and green photosynthetic bacteria. So gas vacuoles will definitely provide the buoyancy there. Okay, they're definitely going to provide the buoyancy there. Right? Okay? Done? This is all about the prokaryotic cell. Now we will talk about the gram positive and the gram negative bacteria. Okay, bache? Gram positive and the gram negative bacteria. Now this is what we need to discuss. So up to this part, if there is any doubt, you are free to ask. Students, let's finish it, then let's have a break. And so in one flow we will just finish the prokaryotic cell and then let's start the eukaryotic cell. What say? Right, let's finish this first and then let's talk about the let's have a break. Okay, then let's have a break. Okay? Okay. Yes, ready? Are you ready? Okay, chaliye. Bache, the word is grams. First of all, let's talk about the gram stain. But it is a very important procedure in microbiology. It is a very, very, very important procedure in the microbiology. And it is, right, bache, used to differentiate some bacteria. Right, it's a very important staining technique in the microbiology, and it is used to differentiate two sets of bacteria, and that is on the basis of their cell wall. That is on the basis of their cell wall. We are differentiating, we are we'll be able to check two different types of bacteria. That's what I'm trying to say. So now when you talk about this Gram stain, who gave this? Kristen Gram. Right, a scientist, Kristen Gram. Right, he gave this Gram staining technique, and on his name, that's why we are calling it as Gram stain. So it's a procedure, it's a staining procedure. Right, basically Gram staining is a staining procedure. We are going to color some bacteria, we are going to differentiate them, and what is the basis of that? Their cell wall. What is the basis of that? Their cell wall. So we are basically differentiating two large group of bacteria there. Is that clear? Is that clear? Yes, bache. Is that clear? Yes or no? Yes or no? Right. So we have two types of bacteria. One is Gram positive bacteria and one is Gram negative bacteria. Gram positive bacteria is also there. Gram negative bacteria is also there. But check Gram positive. What is the meaning of a Gram positive bacteria? The one that gives color with the Gram stain. Yeah, that will get colored with the Gram stain. And like it will, after this staining procedure, it will be somewhere violet, right? And this Gram negative bacteria, it will give red color after the staining technique, right? It is the one which will not get colored with the Gram stain. Done, bache? Done. Gram positive or negative bacteria. So one will give the color with the Gram stain, that will be Gram positive, and the one which is not going to give the color, right, that will be the Gram negative. Right. Yes or no? Yes or no? Chaliye. So, right? Okay, so here we are talking about a stain, but I'll tell you the procedure. I know this procedure is not for you, right? Matlab you don't need it, but you should know it to understand actually. Come in and stain. Here we have a trick, that is come in and stain. This come from the C, you need to know crystal violet. I means iodine here. A means alcohol here. S means saffron. Okay, come in and stain. Come in and stain. Come in and stain. Right, bache? C for crystal violet, I for iodine, A for alcohol, S for saffron. Actually, when you talk about the Gram stain, we are talking about this crystal violet stain. It's a water soluble stain. What's that? It's a water soluble stain. It is the one that will give the violet color. Then iodine. This is the Grams iodine. Here you can say we take potassium iodide, and iodine will be taken. And that is basically water insoluble. So firstly, right, A for alcohol or even acetone, that is also for the washing. And this is the next stain here, that is saffron. Saffron, that will give the red color. Actually, what's the procedure? Let's say here you have the bacterial culture. Actually, why do we need this stain? Matlab, it is very commonly used, but it's a very common technique. It is used to check the infection also. Let's say if we are suspected with some infection and we are like Gram negative bacteria is causing it, we can check it with the help of this Gram stain. Right. So let's say here you have a culture. You have a culture of bacteria. Obviously, bacteria are not visible. So imagine that these are the colonies of bacteria. Right. So now here you want to check which bacteria are Gram positive, which bacteria are Gram negative. So as I said, firstly you have to, I'm not explaining this procedure in detail. I'm just telling you, firstly what will you do? Just come in and stain. The first step includes the addition of the crystal violet. We have to stain the cells with crystal violet dye. Right. We have to stain the cells with the crystal violet dye, right? This is the very first step. This is the very first step. Then bache, after that you will add I wala step, iodine, potassium iodide. It is acting like a mordant. It will fix that stain here. It is going to fix that stain here. Are you getting my point? Are you getting my point? So that potassium iodide, iodine and potassium iodide is added, right? It will form a kind of complex with the crystal violet, right bache? Right. Just I told you crystal violet is water soluble, right? Then you are adding a mordant because we want to fix that color there. We want to fix that color here. So obviously this crystal violet and iodine, they will form a kind of complex and ultimately it will become water insoluble, right? So now the next step is the come in and stain. A means alcohol. This alcohol is, acetone is used for the washing. But these are the steps of staining, right? It is just like a decolorizer. Right, it is just like a decolorizer, right? Just let me write it. It can be alcohol or acetone. Okay, so it is a decolorizer, right, bache? Right, matlab we do the step. It is mandatory here, right, bache? So basically, when it will be added to that sample, it is going to dehydrate, right? Right. It is, what is the role of this? It is going to dehydrate peptidoglycans. You know, the cell wall is having peptidoglycans. So if peptidoglycan is getting dehydrated, obviously, don't you think its shrinking will occur? Don't you think its shrinking can occur? The cell wall can shrink. Please tell me. Don't you think the cell wall can shrink here? Yes. Yes. Cell wall can shrink here. Yes. Yes. Right. So basically, when we add this decolorizer, it can be alcohol or acetane. It will dehydrate the peptidoglycan. It will result in its shrinking. It will result in its kind of tightening. And because of that, what's going to happen? The gram negative, this crystal violet and iodine complex, right? This crystal violet and iodine complex will not be able to cross, will not be, right? It cannot cross the cell wall in gram positive bacteria. Just I am saying that some bacteria are gram positive, matlab they will give color. They will give color with this gram stain, and some bacteria they are not going to give it, right? How can we identify it? We can identify it with the gram staining procedure. So basically we have to add this alcohol, a mandatory step, which is a decolorizer. Actually 2 types of bacteria are there, right? In the case of gram positive bacteria, alcohol will not be able to decolorize it. Matlab even after adding the alcohol, gram positive bacteria will not lose that stain. It will keep it. It will show that violet color. But if in the case of gram negative bacteria, right, they will be destained. Why? Because obviously the answer is in their structure. The answer is hidden in their structure, in their cell wall thing. I will discuss that. So basically in this sample, the bacteria which are gram positive, they are not going to get decolorized, right? They'll just keep that stain, and the bacteria which are gram negative, they will just, after adding the alcohol, their stain will come out. That's why we further add safranin. Why? So that this negative bacteria can pick up this color. Okay, bache? Right? So this is a kind of counter stain. Right, this is a kind of counter stain. We have to add it to the sample and it will give that gram negative bacteria red color. Okay, right, bache? So this safranin is what? It is a counter stain. It's a counter stain. It is little lighter than the crystal violet. So it will not disrupt the color of this gram positive bacteria, but it will provide coloration to the gram negative bacteria. So now why is it so? Why in the case of gram negative bacteria, the stain is coming out, the crystal violet iodine is coming out, but why it is not happening in the case of gram positive bacteria? The answer is there in their structure. When you look at the gram positive bacteria, look at its cell wall. Just look at its cell wall. Its cell wall is so thick. It is having peptidoglycans, obviously. The cell wall is very thick, very thick. So even after the addition of alcohol, even after that dehydration, even after its tightening and shrinking, that stain, crystal violet iodine complex, is not able to come out. It is not able to come out. It will be there in this part. Right, the cell wall is quite thick. Now in the case of gram-negative bacteria, look at this gram-negative bacteria. Gram-negative bacteria is having one inner membrane made up of phospholipid. Look at its peptidoglycan cell wall. It is very, very, very thin. Plus in gram-negative bacteria, there is one more outer envelope. There is one more outer membrane, and that is also made up of phospholipids. So what's going to happen when we add the decolorizer? Obviously this membrane will shrink and you have added the alcohol, right, and this is again the phospholipid bilayer. So basically what's going to happen, crystal violet and iodine complex, that color will be able to come out because the cell wall is not thick here. So it is, that is why it is not able to retain the color. It is not able to retain the color, right bache, right? Here some pores will be created, the color, the complex will come out and it will not be able to retain the color. That's why in this staining procedure we add the counter stain that will provide it the red color. So if we will see the red color, we'll say these are the gram-negative bacteria. If we will see the violet color, we will see that they are the gram-positive bacteria, right bache? So that is how this gram stain is helping us, right? That is how this gram stain is helping us. Clear bache? So crystal violet is the primary stain, saffron is the secondary or the counter stain, right bache? Right, so mordant is your iodine solution. Is that clear? Is that clear? Are you sure? Yes, my dear students, are you sure? Are you sure? So you have different examples of disease causing gram-positive, gram-negative bacteria. So right, bache, I hope you guys know about it, right? I hope you people know about it. जैसे gram-positive bacteria में, if I talk about the examples, Streptococcus species, Staphylococcus species. There are many other examples also. Gram-negative में Escherichia coli, right, and your Neisseria gonorrhoeae. Just now I used that example, no? That is also the example of gram-negative bacteria. So yes, you should know the examples. So your Neisseria gonorrhoeae, your E. coli, right here, Streptococcus, Staphylococcus species are there. So this is about the gram-positive and the negative bacteria. This is all about the prokaryotic cell. Next is the eukaryotic cell that we will start after the break, right? So let's meet at 9:15 p.m. Okay, done? Chal. Hi students, welcome back. Hello, welcome back. Hi. So let's continue guys, let's continue, and now we are going to start eukaryotic cell. What are we going to start, people? What are we going to start now? We are going to talk about the eukaryotic cell. Okay, that's what we have to discuss. So remember, kingdom, protista, fungi, plantae and animalia. These 4 kingdoms are having prokaryotic cell. Is that clear? These 4 kingdoms are having, right? Yes. Hello, hello, hello. Please focus here. So these 4 kingdoms, right? These 4 kingdoms are having prokaryotic cell. I hope now I'm perfectly audible. I hope now I'm perfectly audible. Thank you, Bache Abdul. Thank you so much. So they are having the eukaryotic cell. So this U word means true, right? So karyon is associated with nucleus. So true nucleus. So here we are going to see a well-defined nucleus. We are going to see a well-defined nucleus. Here in the fungi, in the plantae, you will see the cell wall. And when we talk about the protista, right? Just say I'll give you the example of euglena, right? I'll give you the example of euglena here. So here, right, when we talk about the outer covering, this euglena, the spindle organism. So when we talk about the outer covering there, that is pellicle, right? That is kind of providing it the flexibility. Okay, so that is what we have to discuss. So now one by one, we will talk about it. We'll talk about the outer structure. We'll talk about the organelles. That is what we have to discuss. Okay. Okay. Show some fire in the chat section. I know now you people are drained, you are not interested in marathon, and you are like, yeah, nahi madam, now it's the day 39 and we don't want the marathons. We don't feel like studying or something, but trust me, you have to break this thing. Right, bache? If you really want to crack your NEET examination, you have to be consistent. If you are thinking that for 3 days, for 5 days, for 10 days, you're not going to attend the marathon, or you're not going to do the self-study, and then you will start, it is not going to help you. Trust me on that, it is not going to help you. So it's better, it's good, right? It's better, it's good to stay consistent. I know it's tough for attending the lectures for 5 hours, 6 hours, 7 hours, but do you have any option? Do you have any option at this point of time? Right, if you have already covered this chapter, you can use this lecture for the revision. And I'm not saying watch the lecture live. I'll be very honest about it. Right, it feels good when students, they watch us, when we have more number of students in our live session, but it's okay, it's fine. If it is not convenient for you and you want to attend the first, you can say that you want to attend the lecture for 2 hours, then you can give a break to yourself. You can watch the recorded one. Okay, you can do this, but please make sure that you attend the class daily, right? Please make sure that you people revise, right? That you people revise, please. That's a request. And do not underestimate biology. That's what I can say. Okay, do not underestimate biology. Chaliye. So now let's start the eukaryotic cell. So if you people are having any doubt, right, do let me know, because I'm going to start it then. Yes, I'm going to start it then. And that is your homework. You guys are going to write down the differences in prokaryotic and the eukaryotic cell. Though I'm going to discuss that, but it is your homework. You have to write it on a paper, okay? You have to write it on a paper, okay? Right? Yes? Okay? Chaliye, let's start. So that's okay. That is what we have in the NCERT. So what NCERT is saying, it is saying that eukaryotic cells will include protista, plants, animal and fungi. I told you. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm. But it's very simple. You have the cell. You have a nucleus. Nucleus is also covered with the membrane. If you have any organelle, let's take the example of mitochondria. Let's take the example of plastid. Let's take the example of lysosome, endoplasmic reticulum. They are also having the membranes. They are also having the membranes. Here you are not going to see the outer membrane only. Right? We have the outer membrane, but here also these organelles are also covered with the membranes. These organelles are also covered with the membranes, right? So they are basically making some compartments in the cytoplasm. That is why we are using the word compartmentalization here, right? That it is dividing the cytoplasm also, right? Because that membrane bound structure is present, right, in the cytoplasm itself. So let's say this area of the cytoplasm, here you have this organelle, this organelle, this organelle. So basically it is responsible for the extensive compartmentalization of cytoplasm through the presence of membrane bound organelle. Is that clear? So eukaryotic cell में proper nucleus with nuclear envelope, definitely. Variety of locomotory and cytoskeletal elements are there, definitely. And genetic material is organized into chromosomes. Genetic material is organized into chromosomes. So here one line is written, that is that all eukaryotic cells are not identical. They are not same. You cannot say that all the eukaryotic cells are same. No, they have the differences. They have the differences. Just plant and animal cells. Plant cell is having cell wall. It is having plastid and it is having a very large central vacuole. And as I said, bache, this central vacuole is going to push the nucleus to one side. So in the plant cell, the nucleus is not there in the center. And these things, the plastid, the cell wall, the large central vacuole, it is absent in an animal cell. And animal cell is having the centriole, which are absent in almost all plant cell. Again, the word is almost all plant cell. Are you getting my point? Are you getting my point? So this is the diagram which is given in NCERT. You guys can see here. So here you have the vacuole, right? Here you have the vacuole, right? Can you see that? So because of that, your nucleus is pushed to one side. Okay? So it is eccentric here. So look at the rough endoplasmic reticulum, right? The mitochondria here, right? Microtubules, the cytoskeletal element, peroxisome. That's basically small organelle. Microbodies they are. Chloroplast is present there. Ribosome, proper cell wall, middle lamella. So we have to discuss this. Right, and this is the animal cell. The nucleus is there in the center, and look at the centriole. So again, I'm repeating, two centrioles perpendicular to each other, covered with the pericentriolar material, right? Covered with the pericentriolar material, they are forming centrosome. They are forming centrosome. Is that clear? Is that clear? Chali. So let's start with the cell membrane. That is the plasma membrane, something which is very, very, very important. Why? You know that this is a cell. This is its cell membrane. So don't you think that this cell membrane is acting like a, it is just like a boundary in between the inner composition of the cell and between the outer side? Yes or no? Yes or no? When I am talking about the cell membrane here, tell me, when I am talking about the cell membrane here, so can I say that it is acting like a boundary? Yes, it is acting like a boundary, right? It's like a barrier between the cell and its environment, right? Yes or no? Yes or no? So can I say that through the cell membrane, the cell is going to communicate with the exterior, with the surrounding. With the help of the cell membrane, cells, they communicate with its surrounding. And you know that the cell membranes, it is going to decide that what is going to enter here in the cell and what is going to leave the cell, isn't it? what is going to enter there in the cell, what is going to leave the cell. And this cell membrane is also helpful bache to respond to the stimulus as well, right? We will see that there are some receptors are also there on the cell membrane to which different molecules can bind, and further, you know, we have studied that in the mechanism of hormone action, right, that some receptors are there on the cell surface, and how, you know, that will help, that complex will help in generating the second messenger within the cell. So there are different roles, right, bache, there are different roles. So basically with the help of the cell membrane, cell is going to communicate with the exterior, right bache. Cell membrane is going to control what is going to enter there in the cell and what is going to leave the cell. So these are the points that you need to take care. Okay, so when we talk about the cell membrane, yes, we do call it as plasma membrane, right? We do call it as plasma membrane, and we also call it as plasma lemma. So bache, when we use the word lemma means covering. What is the meaning of lemma here? Lemma means covering. And let me tell you, this plasma membrane and the membrane of all the organelles, because here the organelles are also covered with the membranes. So we call them as biomembranes because of their extensive, or because of their vital role in that, right? Because of their vital role. Is that clear? Is that clear? Right, because of their vital role for that particular cell, we call them as biomembranes, or we can also call it as biological membrane. Is that clear? Is that clear? Yes. Yes. Tell me. Yes. So when you talk about the structure of cell membrane, there are different models. Jaise we talk about the Overton's suggestion. Obviously, I'm not going to discuss that in detail, but still I'm giving you an idea here that one is grain. This Overton suggestion is there. Now when we talk about this Overton suggestion, his suggestion, he said that plasma membrane or the cell membrane is what? I will use the short form PM here. It is made up of lipids. Why? Why? On what basis he said so? As per him, right, bache? As per him, right, the plasma membrane is made up of what? It is made up of lipids. It is made up of what? It is made up of lipids. Right, bache? Right. Yes? Yes, right. So on what basis he said so? On what basis he said so? Basically, he's observed that lipid soluble things can cross the cell membrane. So he was like, yaar, plasma membrane is made up of lipids, right? The plasma membrane is made up of lipids, right? Then bache, there is some, I don't remember their name properly. Gotter and Grendel, some hypothesis is there. We mainly need to focus on the fluid mosaic hypothesis, but I am telling you what other things were there, right? One Gotter and Grendel model was there. So when they studied the plasma membrane, they were like, yeah, it is made up of lipids, but it is a bilayer of lipids, right? It is a bilayer of lipids. It is not a single layer, it is a bilayer of lipids. So they studied the RBCs, right? They studied the RBCs, and on the basis of that, they suggested, they studied the, what do we say, this, the surface area of that RBC and all. Okay, bache? So Namita, on the basis of that, they also provided some suggestion. Then comes the Daniel and Davidson model, right, bache? Then comes what? Then comes the Daniel and Davidson model, and this model is also known as sandwich model, right, bache? This is also known as sandwich model, or you can say that it is basically trilaminar model. It is also known as trilaminar model. So what am I trying to say? I'm saying that before this fluid mosaic hypothesis, right, bache? Before this fluid mosaic hypothesis, some other popular models were also there. But now you know that widely accepted one is what? Widely accepted one is your fluid mosaic hypothesis, right, bache? Right, bache? It is right. It is what? It is the fluid mosaic hypothesis. But before that, some other models were also there. Right, bache? Some other models were also there. So it is sandwich or trilaminar model by Davidson and Daniel. Is that clear? By Davidson and Daniel. Is that clear? Sure? Sure. So we will discuss it now. Yes? Yes, bache? Is that clear? Done. So now why is it trilaminar model or why is it a sandwich model? Just here, right? Before them, the Gotter and Grendon model was also saying कि plasma membrane is a bilayer, right? Two layers are there. So as per them, when they studied it, मतलब they were clear कि there is a phospholipid bilayer, right? See guys, I am not going to explain it in detail, right? I will just provide you an idea. Then we will discuss fluid mosaic hypothesis in detail. Just for an example, you know that this is how we can draw, right? That is how we can draw this, right? The phospholipid bilayer and all. This is how we can draw this phospholipid bilayer and all, right? Let's say this is it. Phospholipid bilayer is there, right? How do we draw it? As I said in fluid mosaic hypothesis, we will discuss that. But when you talk about this part now, it is your polar head. It is your polar head. It is your polar head. And this part is non-polar tail. Polar head is there or non-polar tail is there. And this tail is actually that carbon hydrogen chains are there basically. This polar head and non-polar tail. We again, as I said, we will discuss that in detail. Polar head or non-polar tail is there. Now when you talk about this head, just imagine, right, here in the case of trilaminar model also, this is something which is common even in fluid mosaic hypothesis or even here in this model, because these are the facts only. Let's say this is the cytoplasm, right? This is the cytoplasm. This is, let's say, extracellular matrix. Now, my dear students, let's magnify this part. This is, let's say, this is a cell membrane. Right? What is it, guys? Quickly in the chat section, do let me know, everybody. Do let me know. I want to see some energy in the chat section. Right? Don't be like, yeah, she's saying something and we have to note down. Guys, I want some energy in the chat section, so do it quickly. Right? Do it quickly. Yes, so that is what we have. Yes, so this is your phospholipid bilayer. So basically this side of the membrane is also facing, if it is this matrix, so you can say that somewhere it is also facing the water and this side is also facing the water, right? Cytoplasm is also having the water, no? So both the sides are facing the water, right? Let's say that both the sides are doing what? Both the sides are facing the water, right? Both the sides are facing the water. Are you getting my point? Yes? Yes? Tell me, is there any doubt here? One second. So both the sides are facing the water, right, bache? Both the sides are facing the water here. Right, so here what you need to remember that when you are talking about this head part here, no, when you are talking about this polar head part, it is facing the water. That is how you can remember. So it has to be hydrophilic, right? So this polar head is hydrophilic. This polar head is hydrophilic. Hydro means water, philic means loving. This polar head is hydrophilic. Hydro means water. This philic is loving. So even this side also, right? This side also, जैसे ये देखो, this side hydrophilic, right? This side is also hydrophilic, water loving, right? Polar head. And when you talk about this tail part, it is what? It is hydrophobic. right? So phobia means, right, you are scared of something. Hydro means water, so phobia from the water, right? So this tail here, this non-polar tail here is water repelling, right? It is water repelling, and the head here is water loving, right? So even in this particular model, when you, which is your Daniel and Dixon, right, this is what they explain. They explain that there is a phospholipid bilayer with a single continuous layer, right? Phospholipid bilayer with a single continuous layer of proteins on either side. Right. So when you are talking about the phospholipid bilayer here, so they were like, let's say this is the phospholipid bilayer, so both the sides are having proteins, and as per that, these proteins are basically what? They are the globular proteins. They are the globular proteins, and they are held here with the help of electrostatic forces, right, bache? With the help of electrostatic forces. Are you getting my point? So phospholipids and proteins, they are held together with the help of electrostatic forces. Is that clear? Is that clear? Right? Yes or no? Yes or no? Right? So now when we talk about this particular model, this model was not able to explain the movement of the substance across it. It was not able to explain the active transport. It was not able to explain the movement of the water soluble substances. Right, bache? It was not able to explain the movement of the water soluble substances across it. It was like, yeah, how is it possible? Right. So these were the drawbacks of this particular model. So they were like, there's some globular proteins are present on the either side. This protein, and these lipids, they are held together by electrostatic forces. Plus this layer is also, this phospholipid bilayer is also having the van der Waals forces which are holding it together. But they were not able to explain how things are moving, especially the water soluble things. Right, bache? Right, bache? So these were the drawbacks. So after it, we have another model, that is Robertson. Right, that is Robertson model. Do you know anything about that? Yeah, Robertson's hypothesis. Yes, bache. Robertson model, yeah, unit membrane hypothesis. Right, you can call it as unit membrane hypothesis, or basically we can say that it is the Robertson model. Unit membrane hypothesis. So basically, if you say that there is no major change in this Daniel-Davidson model and this Robertson model, right? Some major changes are not there. It is almost same, and yes, by that time they were able to explain the dimensions as well. As per them, this phospholipid bilayer is 35 angstrom, and this each protein layer, this 20 angstrom, and this one is also 20 angstrom. So overall 20 plus 20, 40, 50, 60, 75 angstrom. So this plasma membrane, its thickness is 75 angstrom. This thickness is 75 angstrom. Right, bache? Right. Yes, bache. Right? Yes or no? Yes or no? Okay. So why do we call it as a sandwich model? You just see this, right? Trilaminar mane 3 layers are there. 1 phospholipid bilayer, 2 protein layers. So isn't it like this phospholipid bilayer is sandwiched in between 2 protein layers? Right? Imagine the sandwich, bread in between stuffing. Same is the case here. Protein in between phospholipid bilayer is there. Okay, now this Robertson model, as I said, it is no major changes are there in this unit membrane model and in this what do we say, trilaminar or sandwich model. Actually, the Robertson, he studied RBC. He studied the RBC membrane. He stained it, right? He stained it, and then he got that there is one light staining, like 3 layers were there, one light stained, and 2 dark stained layers, right? 2 dark stained layers. So obviously he supported this trilaminar model, and here this is the phospholipid bilayer again. So but it's just that proteins, they are not globular, they are maybe the extended one, right? The extended one, or you can say that the kind of fibrous one. That is the change here only. That is the change here only. Again, the drawbacks are also same here, right? This model is also not explaining the active transport. This model was unable to explain the movement of the water soluble substances across it. Is that clear? Is that clear? Yes or no? Right? Yes or no? Right? Tell me, any doubt? Any doubt? So all membrane he studied, they are the unit membranes, basically 3 layered they are, okay, right? So 2 protein monolayers and 1 phospholipid bilayer is there. So they are basically the unit membranes, okay bache, right bache? Done. So drawbacks are same. So now the next model which explained everything, the most widely acceptable model is what? It is the fluid mosaic hypothesis. What's that bache? It is the fluid mosaic hypothesis. What's that? It is the fluid mosaic hypothesis. And here you need to remember the scientist name as well. That is a PYQ, your Singer and Nichols. They gave this, right? They gave this hypothesis, and this explains the plasma membrane structure, even the transport thing as well, right? Even the transport thing as well. So guys, here matlab I'm giving you the highlight here, fluid mosaic hypothesis. Fluid mosaic hypothesis. So as per them, plasma membrane is quasi-fluid in nature. As per them, plasma membrane is quasi-fluid in nature. It is semi-fluid, right? It is not, it is some semi-fluid structure. It is not even solid, not even liquid, some semi-fluid structure is there. That's the point, right bache? Plus proteins are in iceberg. Proteins are the iceberg in the sea. Proteins are just like icebergs in the sea, right? Matlab I am explaining the basic structure here, then one by one we will discuss that in detail. Fluid mosaic hypothesis given by Singer and Nicholson bache, right? So biomembranes, these membranes, they are not solid but viscous fluid they are, right bache? Right bache? Yes or no? Protein iceberg in the sea of lipids. Protein icebergs in the sea of lipids. It's like that. Protein icebergs in the sea of lipids. So when we talk about this particular structure, so as I said, it is quasi-fluid. Yeah, you can say that it is a viscous fluid in nature. It is not even solid, right? So protein iceberg in the sea of lipids. What is the meaning of that? Ki proteins and the lipids, as per this particular hypothesis, right? As per this particular hypothesis, proteins and lipids, they are not arranged like this ki one monolayer of proteins or the one monolayer of proteins and the bilayer of this lipid. No, it is not like that. It is like ki proteins and lipids, matlab their arrangement is just like a mosaic. Matlab it's not uniform, right? Their arrangement is not uniform. Arrangement is not uniform, but mosaic arrangement is there. They say, let me show you the diagram. Let's see this phospholipid bilayer. See, these are the proteins in between. These are the proteins in between. It's not like that this is the lipid bilayer and it is covered with the proteins. No, they are present like this, right? They are present like this. Have a look, right? This is directly from the NCERT. So arrangement, this arrangement, matlab here, what's the point? What is the point here, bache? The cell membrane, the lipid and protein, yes, they are present as a mosaic. Is that clear? Is that clear? Yes. So phospholipid and proteins, they are not arranged uniformly but as a mosaic, right bache? Okay. Done. Is that clear? So this structure is of phospholipids. These are the proteins in between. So let's talk about the phospholipids first and then we will talk about the proteins. So just have a look. Just have a look. This is the membrane. Phospholipids are there, right? Phospholipids are there. So that part is same, bache, right? This part is same. Jaise this head, globular head, it is the polar head. I explained you already. It is hydrophilic. It is what? It is hydrophilic. Remember it like this now. From both the sides it is facing the water, right? It is facing the water. It has to be water loving. It has to be water loving. And just imagine the setup is in such a way, right bache? Right bache? Just imagine the setup is in such a way, this is forming a kind of water resistance barrier. We will discuss about it, right? It is forming a kind of water resistant barrier. We will discuss about it. So basically this is the polar head, and here the structure that we are making like this, it is nothing but it is the carbon hydrogen chain, right? It is the carbon hydrogen chain. So when you talk about this, we are using the word phospholipid here, right? So students, I don't know whether you know it or not. And let me tell you, in biomolecules, we are going to discuss it. Like when we are talking about the lipid, the simple fats, what do we consider? What do we say? That there will be the alcohol and the fatty acids will be there. Alcohol will be there and the fatty acids, and they join together with the help of ester bonds, right? They join together with the help of what? With the help of ester bonds. So there will be the alcohol, there will be the fatty acids, and they will join with the help of what? They will join with the ester bond. And when it is the simple fat, so this alcohol is going to be glycerol there. What is it? Glycerol. Glycerol, trihydroxypropane. Right, we will discuss the same in the biomolecule chapter as well. So it is trihydroxypropane. What's the word? Trihydroxypropane. So propane it is, means 3 carbons, trihydroxy means 3 OH group, right bache, like this, and then you just need to satisfy this valency here, and it is going to be like this. This is your glycerol, 3 ols are there. Okay, so now fatty acid, we have to add the fatty acid here, just giving you the overview here. Bache, fatty acid, if it is an acidic compound, so this is going to be the functional group. Carboxylic acid, this is going to be the functional group, yes or no? This is going to be the functional group. So obviously one thing is for sure, that here you will see this, and there will be the carbon hydrogen chains. There will be the carbon hydrogen chains, right? There will be the carbon hydrogen chains. So this carboxylic acid, this OH here, the bond formation will be there, the ester bond will form, esterification will occur. So if one fatty acid is attached, monoglyceride, 2 fatty acids are attached, diglyceride, 3-4 fatty acids are attached, triglyceride, the most common type of lipids. So glycerol, it will be 1 plus 3 fatty acids, they are forming the triglyceride. General, I'm talking about the lipids. So now if I'm specifying it is a phospholipid, matlab that phosphate group has to be there. So just say this is the phosphatidylcholine, right? They are showing the structural formula here, right? So if you will see its globular head, its head part, right? What are you people going to see here in this head part? This, right? This, this, right? You will see the glycerol here. You are going to see the phosphate here, right? So because it is a phospholipid, that's why the phosphate group is also there now to this glycerol. Just one side is having the phosphate group, other 2 sides are having this fatty acid chain, right? Other 2 sides are having this fatty acid chain. Just you will see the saturated fatty acids also, unsaturated one. Unsaturated one mane, here you will see the double bond. And wherever you will see the unsaturated fatty acid, there will be a kind of kink or bend will be there. I'll explain you the reason. Firstly, understand this. So this is the fatty acid chain, right? The hydrophilic tails are there. Just observe it carefully, right? So this head part will be having glycerol and phosphate here, right? Because it is phosphatidylcholine. It is given in your NCERT also, the structure, right bache? So that's why these groups are there, and this is the carbon hydrogen chain. Carbon hydrogen chain. Carbon hydrogen chains are here. Is that clear? Is that clear, guys? Is that clear? This is about its structure. Again, just look at this. Just look at this saturated, or wherever you have the double bond, unsaturated fatty acid, and you are going to see a kind of kink or a bend there. So glycerol and phosphate are there in this hydrophilic head. So this structure part clear? Tell me. The structure part clear? So from this, can I say that phospholipids are amphipathic? Can I say that phospholipids are amphipathic? Yes. Why amphipathic? Amphipathic means dual. They have both hydrophilic and hydrophobic part. They have both hydrophilic and hydrophobic region. That's why the word is amphipathic. That is why the word is amphipathic, right bache? That is why the word is amphipathic, because it is having both the hydrophilic part and the hydrophobic part. Any doubt? Yes, any doubt here? Right, it is having both hydrophilic and the hydrophobic part. Is that clear? Is that clear? The arrangement here clear? Right, so hydrophobic part is inward, so it is forming a water resistant barrier. Right, जैसे ये देखो, this hydrophobic part is inward, it is forming what? It's forming a water resistant barrier. Right, it is forming what? Water resistant barrier, and this barrier will not allow it will only allow the lipid soluble things to cross. Right, there is a water resistant barrier, and because of that, what will happen bache? Only lipid material will be able to pass through it. Is that clear? Is that clear? Is that clear? Done? Done. ठीक है. Now, these are two leaflets here. Okay, I'm not going to draw the structure again. I hope it is clear, बच्चे. जैसे let's say if this is your, guys, is it clear? Is it visible? This is the phospholipid bilayer. Now, bache, here one more thing is there. These phospholipid, this is one leaflet, outer leaflet you can say, and this is inner leaflet, right? This is outer leaflet, right, students? This part is the inner leaflet. Matlab phospholipid bilayer is there. I am saying outer portion is the outer leaflet, inner portion is the inner leaflet. Now, bache, both these sides are having, both these leaflets are having. Right, let me tell you, both these leaflets are having different lipids. Means you cannot say that lipid composition is also there. Matlab both the leaflets are also not same, they are asymmetrical. This is the outer leaflet. It is having some oligosaccharides attached to it, but you are not going to see it on the inner side. Plus, here the lipids which are present in the outer leaflet, they are different from the one that are present in the inner phospholipid layer. Is that clear? Is that clear? Hanji, is that clear? Tell me. Tell me quickly, guys. Tell me quickly. Yes, tell me quickly. Any doubt? Any doubt so far? I'm not going to mention the points here. I just want you people to understand this. Yes? Yes? Is that clear? Right, so both the leaflets, they are also asymmetrical. First of all, they do not have the same phospholipids. Plus, here to these phospholipids, you will see some oligosaccharides are attached. They are not on the inner side. So now, when I'm talking about these leaflets, outer and inner, right, I'm not going to write it. I'm not going to write what I'm explaining. You have to note down the points. You have to note down the points. Be quick. So when I'm talking about the outer leaflet, guys, in the outer leaflet, you will see, just I'll give you the example, phosphatidyl. Dialcholine, it's the name of a phospholipid, right? Just we discussed na, lecithin, the most common type of phospholipid in our body, biomolecule. Do check it, this lecithin and phosphatidylcholine, what is this, do check it. So phosphatidylcholine, your sphingomyelin, right? I'm not saying that you have to remember it. I'm just telling you, I'm just giving you an idea that outer leaflet is having the different types of phospholipid, inner leaflet is having the different type of phospholipid. Jaise maan lo, phosphatidyl ethanolamine is there, Yes, ethanolamine is there. I still remember. I studied that thing in the masters. Even it was not there in our graduation, it was there in our masters. Right, it was there in our masters. So phosphatidyl ethanolamine was there, and one more was there, that is phosphatidyl serine. Right, that is phosphatidyl serine. Matlab, these are this is the composition here. Right, this is the composition here, and I'm not saying one phosphatidyl inositol was also there. Right, but again I'm saying no need to note that, but try to understand. If I'm making the phospholipid bilayer, right, if I'm making the phospholipid bilayer, let's say this is the outer leaflet, this is the inner leaflet, right, this is the outer leaflet, this is the inner leaflet. So I am trying to say that the lipids here and here, they are of different, they are not same. It's not like that if it is having phosphatidyl choline and this one is also having that. No, right, it varies, that is what I'm trying to say. So let me tell you, if a person is having cancer, this is a very rare type of movement. Sometimes phospholipids, right, sometimes phospholipids from this outer leaflet, they came to the inner one, it's a very rare type of movement. It's a very rare type of movement. What is it? It is a very rare type of movement in which phospholipid from this inner leaflet, they are moving to the outer leaflet. This is a very rare thing. In the case of cancer, right, this has been reported in the case of cancer, and because of that, what is the thing, bache? It is also a kind of, if let's say if a phospholipid is present in the inner leaflet, if it came to the outer leaflet, it gives a sign that the cell is not functioning fine, right? The cell is not working fine. It's required to kill this cell. It's required to do its cell death. It's just like a kind of marker ki yaar destroy this cell. So it's a very rare type of movement, and it is known as flip-flop. Do write it down. Flip-flop. So flip-flop is when the phospholipids, they are moving from one leaflet to another, right? They are moving from one leaflet to another. Now, जैसे ये देखो, this is your phospholipid bilayer, right? This is your phospholipid bilayer. You know that we have the phospholipids, and we know that there are proteins also in between, right? There are proteins also in between. Obviously, we will discuss about it. Without any doubt, we are going to discuss about it, right? So one more movement is there. That is the part of your syllabus. Flip-flop is not given in NCERT, but you should know this, right? Flip-flop is not given in NCERT, but you people should know this. Okay, you people should know this. So basically what is happening, just let's say here you are having the protein as well, right? You are having the protein as well, you are having the phospholipids as well. So basically, you know, these phospholipids, I'm talking about the individual leaflet, right? Just let's say this phospholipid is exchanging the position with its neighboring, right? Just let's say this phospholipid can exchange the position with its neighboring phospholipid. So what's that? Obviously in the same leaflet I'm talking about. If it is in the inner or outer, if it is in the same leaflet. So phospholipids can exchange the position with their neighboring, right, phospholipid. So this property is fluidity. This property is fluidity. It is important. This property is fluidity. It is important, right? What is it? It is important. Are you getting my point? What is it? It is important, right? It is important. Are you getting my point? Fluidity, right? And not just about the phospholipids, even with the proteins also, right? Proteins can also exchange the position with their neighboring phospholipids. This is the fluidity, a very important property of the plasma membrane. Why? Because of this property, plasma membrane is not solid, it's fluid like na. So plasma membrane under stress, right? Let's say if there is any kind of stress, right? If there is any kind of stress, because of the property of this fluidity, plasma membrane can stretch. It can stretch, my dear students, right, bache? So under plasma membrane can stretch under stress if there is any such kind of, because of this fluidity. Plus plasma membrane can reseal if disrupted because of this fluidity thing, right? So it is a very important property. It is a very important property. So what is flip-flop? It's a rare movement when the phospholipid from the outer leaflet is moving to the inner side. It's a marker. It's marking the cancer as well. Are you getting my point? Are you getting my point? Yes, right. So basically in the phospholipid, obviously phospholipids are there. Proteins are embedded here. The proteins are present here. This distribution is not uniform, it is just like a mosaic. We'll see different types of proteins. Some are intrinsic, integral, some are extrinsic, exterior, yeah, peripheral proteins are there. We'll discuss about it. Right now I'm talking about the phospholipid thing. Is that clear? Is that clear? Right? Right. Yes. Okay, chali. Now this fluidity is very important, right? It is very important here, because if membrane, if it has to function properly, it must remain in, yes, it must remain in what? It must remain fluid, right? It must remain in fluid state. Yeah, you can state it must remain fluid for its particular functioning. Right, bache? For its particular functioning, it has to be like this. It has to be like a fluid. If it will be solid, don't you think permeability will get affected? Matlab, we are saying plasma membrane. This is what we generally say, na? Plasma membrane is permeable, या selectively permeable. देखो बच्चे, I don't know whether you know it or not. जैसे we use 3 words. We say a membrane is impermeable, semi-permeable, selectively permeable, या differentially permeable. Right? These are the 3 words. When we are saying that the membrane is what? When we are saying the membrane is impermeable, not permeable. Things cannot move across it. It is impermeable. It is not allowing the movement. Then the word is semi-permeable. Semi mein what is going to happen, bache? Only solvent is allowed to move across it. Only solvent is allowed to move across it. Solvent. But when it is selectively permeable, जैसे your plasma membrane, we say ना it is selectively permeable, differentially permeable. मतलब as per the need of the cell, as per the need of the cells, things will come in, things will go out. It can be solute, it can be solvent. It's not mandatory that it has to be. It's not mandatory that it has to be. It has to be what? That it has to be solvent only. It is like that. So if the plasma membrane, if it becomes solid, obviously this permeability thing will get affected. Obviously this permeability thing will get affected. It has to be in a fluid form. It has to be in a fluid form. Are you getting my point? It has to be there in the form of a fluid. Are you getting my point? Are you getting my point? So some changes can be made to this thing, right? And just for an example, in some plants before winters, right? Before winters, in the plasma membrane, the composition of unsaturated fatty acid, it increases, right? So to avoid its solidification. Are you getting my point? Are you getting my point? So whether the composition of saturated, unsaturated fatty acids and all, it is important here. These things are going to provide these things are important for this structure. I'll explain you this thing. This is the structure. We are talking about the phospholipids. I'm saying proteins are also present in between. Moreover, you will see one more thing. Let me show you the diagram. This is the diagram. Some proteins embedded there, integral protein. Even cholesterol is also there. Right? This diagram is from NCERT. Yes, here in the membrane, you guys are also going to see cholesterol. It is providing the structural stability. It is providing the structural stability to this membrane. The cholesterol is present in animal cells. It is providing the structural stability. I will tell you how. But note down, in plant cell, it is not the cholesterol. It is not the cholesterol, but some other sterol is present. Matlab it's not cholesterol, but it is some other sterol is present here. Are you getting my point? It is present here in the plasma membrane of plant cell. Okay? And you know that in prokaryotes, in bacteria, they do not have the sterols, right? Sterols are absent. Sterols are absent there. Sterols are absent there. But you remember hopenoids that will provide it the structural stability. It's a pentacyclic sterol-like compound. Remember this? Sterol-like compound. Remember this? Yes or no? Yes or no? Tell me. Yes? Done, बच्चे? Sure? Done? Are you sure? So cholesterol is there in the animal cells. In the plant cells, they do not have the cholesterol, but some other sterol can be there. In bacteria, in prokaryotes, sterols are absent, but hopenoid, it's a pentacyclic, right? It's a pentacyclic sterol-like compound. It's a pentacyclic sterol-like compound that is present. Is that clear? Is that clear? So now here, I gave you the example of winter. I told you that, sorry, I gave you the example of some plants. Just say, let's say in the case of, what do we say? Let's say in the case of wheat. In the case of wheat, in the plasma membrane, before winter's approach, it get more unsaturated fatty acid. So जैसे I'm telling you that when you talk about this fatty acid chain, you will see the saturated fatty acid, but unsaturated fatty acid is also there. That bonds are there. That unsaturated fatty acid, it increases. And to be more specific, it is cis unsaturated fatty acid. It increases membranes fluidity. Right? It increases membranes fluidity. Matlab these fatty acid chain, you will see at times unsaturation is also there, because it will provide it the, it is going to provide it the, what kind of thing? It will maintain its fluidity. It is going to maintain its fluidity. So unsaturated fatty acids, they are very important here because they are going to provide the structural stability plus the fluidity to this plasma membrane. So let's say in winters, obviously temperature is less. There are chances the solidification of the fluid can occur. So if unsaturation is more, unsaturated fatty acids are more. So obviously they will increase the membrane fluidity. So as per the need, these things can be maintained. Jैसे it is advised to take, जैसे we use the oils ना, that they should be unsaturated. जैसे mufa, I don't know whether you know it or not. Monounsaturated fatty acid, polyunsaturated fatty acid, right? So that kind of oils, they are even liquid at room in winters, right? Even they will stay liquid in winters also, right? I don't know whether you have noticed it or not. Okay? And cholesterol is also there to provide the structural stability. So is that clear? Yes. Is that clear? Tell me, is that clear? So cholesterol is also, right? It is more stiff than phospholipid. It will also help in stabilizing the plasma membrane. Okay? Done? Right? Now, when we talk about the proteins here, but it is on the on the ease of extraction, we are giving it different names. Just we have extrinsic protein, or we have intrinsic protein. Extrinsic or intrinsic. Just integral. They are extrinsic. You can also call them as peripheral proteins. Right, peripheral proteins. Right, bache? There are two types of proteins also. What do we have? Two types of proteins also. Just integral proteins. Right, integral protein. They are deeply embedded in the phospholipid matrix. Yes. Yes, bache? Yes. Is it clear? Integral protein, matlab they are embedded in phospholipid bilayer, of course. They are embedded in phospholipid bilayer, of course. It's not like that these proteins are very deeply embedded, right? They are, matlab it depends. Just integral protein. If you will see the diagram, let's say this is the phospholipid bilayer. We see there are some there are some proteins, right? And yes, about its distribution, I already told you it's not uniform. Proteins are just like, in between, they are just like scattered in between that lipids. That's why we are saying protein icebergs in the sea of lipids. They are scattered in the lipids. It is giving it a mosaic pattern. Okay, and 70% of the proteins are kind of integral protein. Okay, so when you talk about the integral protein, you will see that there are some proteins which are deeply embedded. They are traversing this phospholipid bilayer. We call them as tunnel proteins, or we call them as transmembrane protein. What are they? Tunnel protein or transmembrane. They are crossing this membrane. And because these proteins, they are making some kind of channels also. Jaise bachche, see, other models, they were not able to explain that how things are moving across the across the membrane. So let's say if we have the tunnel proteins, they are acting, some are acting like carriers, some are acting like channels. Jaise the things which are lipid soluble, they can easily cross the membrane. No doubt it depends upon the size and everything, but we know that lipid soluble things can cross the membrane. But if let's say something is water soluble, something is ionic, so with the help of these tunnel proteins, because they are like the channels, they'll help that things to move across, right? To cross this plasma membrane, to move in or out as per the cell need. Through these tunnel proteins, it is possible, right? Through this tunnel protein, it is possible. Are you getting my point? Are you getting my point? Just say, let's say this is the cell exterior and this is the interior of the cell. That is the exterior side of the cell. That is the interior side of the cell. Right? Right. So things can go in, they can go out. It depends there. One more thing. Some proteins, just say I told you about the fluidity, that proteins can also exchange the position with their neighboring lipids, fluidity it is. But some proteins can be immobile as well. They can be fixed also. They can be fixed also. Just say, let's say if this is the interior side of the cell, the cytoplasm is there. Maybe let's say it is attached to cytoskeletal element. It is not able to move. Exteriorly also in the matrix, it is attached to some such kind of thing. So it is not able to move. So that are the points that you people should know. Right, bache? Right, bache? So integral proteins are majorly making the total membrane protein around 70%. Right, they are tightly held in place, bache. Okay, and they are very large. Sometimes they cross the membrane. We are calling them as tunnel protein then. Right, so water soluble things can move across it. Is that clear? Is that clear? Yes? Done? Done. Yes? Okay, so proteins are present. So these proteins, bache, they provide structural and functional specificity to the cells. to the cells. Is that clear? What are they going to do? They are going to provide the structural and functional specificity to the cell or to the cell membrane. So extrinsic, which are on the outer side, of course, right bache, which are on the outer side, of course, right, they are located superficially. Okay bache, okay, done. So they are present superficially, the extrinsic protein. So bache, as I said, it is on the basis of their ease of extraction. Just let's say these peripheral proteins, yeah, extrinsic protein, they are superficial. So without even destroying the cell membrane, we can isolate it. It's very easy to extract them, right bache, right? So they are present. Okay, they are present. So integral protein are the major protein, and if you will see, these proteins are also attached to the oligosaccharides, right here. They are also attached to the oligosaccharides here, your carbohydrates here. Obviously, towards the outer leaflet, that oligosaccharides are also there, right bache, right bache, yes, right, right. So towards the exposed side of cell, just say, let's say this is your, let's say this is your cell, right? So this side is your outer leaflet. So this exposed side, this outer leaflet will be having these oligosaccharides attached to it. Usually they are branched, but we cannot say that always they are, but usually they are branched. So they are having different roles. Just say, let's say if this oligosaccharide, if it is attached to the protein, so they'll form what? Glycoproteins. If it is attached to the lipids, it will form glycolipids or glycoproteins are there. So that things can help in cell to cell recognition also, right? Cell to cell recognition also can be done with the help of these oligosaccharides. Are you getting my point? Are you getting my point? Sorry, cell to cell recognize self cell from the non-self cell. Just we say that in our immune system, we know that in our immune system, if there is no abnormality, our immune cells, they can kill, they can destroy the foreign molecules, they can recognize them, they can kill them, right? In autoimmune disorders, no doubt, sometimes our immune cells, they are failed to recognize their own cells, they start destroying it, but usually that is how the things are. So how that immune cells, they get to know that this is our own body cell and this is an intruder. These oligosaccharides, they help in that recognition of self cell from the non-self cell, right? So we are not going to enter in the detail of that, but that is how it is. That is how it is. And moreover, in the plasma membrane, bache, sometimes we have to move the things against concentration gradient. Against concentration gradient means we have to move the things from low to high, right? Just let's say you are not rich, you don't have money, but still you are giving money to the one who's rich, right? And that too as what? Yeah, theek hai yaar, I'm giving it to you as a debt. Okay, so low to high against the concentration gradient. So when we have to move something against the concentration gradient, ATP is used, right? ATP is used. ATP, so this is the active transport. So sometime across this, right? Sometimes across this plasma membrane, we have to move the things as against their concentration gradient. Just let's say outside the cell it is high, it is low. Inside the cell it is high, but still we have some ion channels, or rather than we call them pumps, because they will use the energy from the ATP hydrolysis and they will move that thing from outside to inside against the concentration gradient. And if something is moving across the plasma membrane along the concentration gradient, means down the concentration gradient, if something is moving down the concentration gradient, then it is the passive transport. Like diffusion, like osmosis, right? Along the concentration gradient, down the concentration gradient. So bache, when we talk about the passive transport, no use of ATP. Right? So you have diffusion, and we have one more word, facilitated diffusion. I hope now you understand these things. Facilitated diffusion. So when it is facilitated, there is a facility available. Matlab there is the requirement of a channel protein or a carrier protein. Just let's say this is the plasma membrane, right? Things cannot cross. It is something is moving down the concentration gradient, but it cannot cross the plasma membrane. So in that case, it will move across this channel protein, but it is down the concentration gradient. So if that is the case, it is going to be your facilitated transport. Okay. And if something is moving against the concentration gradient, so A for ATP, right? A for ATP, A for active transport. Then it is the active transport. And for active transport also, pumps are required. So you can say that carrier proteins are also required here. Is that clear? Right? Is that clear? Yes, please tell me, is that clear? Sure? Are you sure? Chaliye, let's discuss that from the NCERT. Right, from the NCERT. Okay. So, advent of electron microscope in 1950. So they are saying detailed structure of membrane was studied only after the advent of the electron microscopy in 1950. Again, important it is. Right, so obviously with the help of electron microscopes only, we are able to study the structure of plasma membrane in detail. So meanwhile, chemical studies on the cell membrane, especially in human RBC, this is important. They enable the scientists to deduce the possible structure of plasma membrane. So yes, again, this is also important. Right, bache? Now, cell membrane, as I said, lipids and proteins, and obviously lipids are there, proteins are scattered in between. That's why we say proteins iceberg in the sea of lipids. So major lipids are the phospholipids, bache, that are arranged in a bilayer. You know that phospholipid bilayer is 35 angstrom thick. Right. Also the lipids are arranged within the membrane with the polar head outwards and hydrophobic tail inwards. So this ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. Right, bache? So phospholipid membrane also contain the cholesterol that will provide it the additional stability. Cholesterol is more stable than the phospholipids. So it is here for the stability. Is that clear? Is that clear? Right. So biochemical investigation revealed cell membrane also possesses protein and carbohydrate, oligosaccharide that I'm talking. So ratio of protein and carbohydrate, lipid, it vary, right? But here in human beings, please note down in your notes, you have to add these, in your short notes, you have to add these points. So in human beings, the membrane of erythrocyte has approximately, we are talking about the RBC here, they are having more protein, 52% protein, 40% lipids are there. Okay, ji? 52% protein, 40% lipids are there. Is that clear? Is that clear? Sure. Sure. So depending on the ease of extraction, membrane proteins can be classified. Depending upon the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral will be there on the surface. Integral are partially or totally buried in the membrane. Any doubt? Any doubt? Right. So Singer and Nicholson, they gave in 1972, right? They gave this model fluid mosaic hypothesis and it is widely accepted. Clear, bache? So whatever I have explained, it is given in NCERT, and if there is anything extra, I have highlighted this. Okay, bache, please note down, please note down, please note down. So that is how you are going to make the notes. Okay, bache, that is how you are going to make the notes, and please be consistent, right? Please be consistent. Dekho, after this we'll start the cell wall, and these two things are very lengthy. Remaining structures are very easy, right? So yeah, after completing the cell wall now, let's finish the cell wall, and then let's move to the cytoskeletal element, because some students, they are unable to understand it. So we are going to emphasize there, right? We will discuss the cytoskeletal element after the cell wall. Then we will move to endomembrane system and to other organelles. Is that clear? Is that clear? So I will try my best to finish it before 1, right? So today, yes, the session can go till 1 AM and maybe 1:30. So if you are really interested in knowing all the things, or you want the quality content, then obviously you have to be patient enough. Okay, you have to focus here, you have to be patient enough, right? So this chapter is lengthy. Cell cycle and cell division is not that lengthy. There you have to understand the concept. Once that concept is clear, everything is easy then. Okay, so please pay attention, and if you want to leave the class, just leave it right now. Okay, chaliye. So according to this quasi-fluid, semi-fluid nature enables the lateral movement of protein within the overall bilayer. It is not just the protein. Phospholipids can also interchange their position, proteins and phospholipids also. So this is the fluidity. So fluidity is also important. I have explained it. So basically we need to learn how to decode the NCERT. So when it comes to the notes, if you are thinking madam will write each and everything by herself, no, I am not going to do this for today's lecture, right? Whatever point I am saying, na, this is important, and I am writing on the board. Please take a note of that. Okay. Okay, so fluid nature of the membrane is very important for cell growth, for formation of even the intercellular junctions, for the secretion, for endocytosis and cell division. So I'll pick up this endocytosis thing. What is this? Endo means inside cell, cytosis. It is the invagination of the plasma membrane. Just take the example of amoeba. Right, we have studied that in class 9th also. Take the example of amoeba. Let's say if there is a food particle here, the amoeba will move, right, and it will form a cup-shaped structure around this. And with the time, what is going to happen? These plasma membrane, right, it will cover this food completely. Right, here it will join. The invagination of the plasma membrane will occur. The food molecule will enter, and this plasma membrane will reseal. Endocytosis. Endocytosis it is, right? Endocytosis it is, right? So this fluid nature of this plasma membrane is helpful here as well, right? It is helpful here as well. Are you getting my point? Are you getting my point? So it is invagination of the plasma membrane so that food molecule can enter. You can take the example of amoeba, that its plasma membrane is forming a cup-like structure around the food vacuole. Then that membrane is joining, it is invaginating. Firstly, food will enter and then membrane will reseal. It's like that. Right, it is like that. Okay, okay. So one of the most important function of plasma membrane is the transport of molecule across it. So that's why it is selectively permeable, because it will allow some molecule present on either side of it to. So many molecules can move across plasma membrane. Some they do not need energy, passive transport. Just a neutral solute which are not charged, they may move across the membrane by simple diffusion. It depends upon their charge also, or their size also, right? They can directly cross. So this is the passive transport, no usage of energy. Passively things are moving along the concentration gradient, things are moving, right? Just say water. When water is a solvent, you know na, how do we define osmosis? How do we define osmosis? I think while in the game of NEET 2.0 itself, I have explained it. So when you talk about the osmosis, what is osmosis? The movement of solvent. The movement of solvent across the membrane. Movement of solvent as per its concentration gradient, along its concentration, down its concentration gradient through a semi-permeable membrane. How do we define this osmosis? Movement of solvent. We are specifically talking about the solvent here, right? We are specifically talking about the solvent here, right? So basically solvent, right? Your water, it will move through a semi-permeable membrane along the concentration gradient, down the concentration gradient. Are you getting my point? So as the polar molecules cannot pass through the non-polar, charged molecule cannot pass through that non-polar lipid bilayer, they need some carrier protein to facilitate their transport across the membrane. So few ion can transport across the membrane against their concentration gradient, bache. That is active transport. Pumps are required. We have the example of sodium potassium pump. We see it in the neurons membrane, right? In the case of neurons, we talk about this now, sodium potassium ATPase pump. The trick here is Nokia kin 2. The trick here is Nokia kin 2. So sodium will move outside, potassium will move inside, A for ATP is used. So just to make it clear, kin 2 matlab potassium will move inside, right, and 2 potassium will move inside. So 3 potassium will move outside. That sodium potassium ATP is pumped. That is also the example here. Is that clear? Is that clear? So just look at this particular diagram. Unsaturated versus saturated hydrocarbon tails. So unsaturated hydrocarbon tails prevent packing and enhance the membrane fluidity, right? Because these kinks will form here. This bending of the plasma, the fatty acid chain will be there. And here, bache, when it is the saturated hydrocarbon, they are packed together. So it increases the membrane viscosity, but it will increase more fluid nature, right? And how this cholesterol is helping? It reduces the membrane fluidity at moderate temperatures by reducing phospholipid movement. But at low temperature, it stops solidification by disrupting the regular packing of the phospholipid. That's why cholesterol is important. Please take a note. Please take a note. It is something very important, guys. You are not going to get this content even after paying fees in lakhs. So please note down. Yes, please do it. Done? Yes? Is it clear or not? Done? Chaliye. So the next is the cell wall that we have to discuss. So if there is any doubt, do let me know. If there is any doubt, do let me know. Right, so you know that cell wall. We will see cell wall in the case of fungi, right? Even in the case of plants, we are going to see the cell wall, right? So when in the case of plants, the cell wall is having cellulose. It is having hemicellulose, right? It is having pectin as well, right? So guys, we have already completed anatomy of flowering plants. If you remember, right, we also know sometimes in the cell wall, some other deposition, just suberin, lignin is also there, right? But generally, this is what you are going to see. This is what you are going to see in the case of plant. And in the case of fungi, what will you see? You will see chitin, right? What are we going to see, guys? We are going to see chitin. So chitin is sometimes referred to as fungal cellulose, right? So bache, it is a homopolysaccharide. Even your cellulose is also a homopolysaccharide. So homo means same. It is made up of same type of subunits, right? Right. So chitin is made up of N-acetyl glucosamine. So many N-acetyl glucosamine, N-acetyl glucosamine, they are together. Many N-acetyl glucosamines together, right? They'll join, they will form chitin. So it is not just the chitin which is there in the fungal cell wall, some proteins are also there. So one more thing that you need to remember, these are the direct PYQs. This chitin is also present in, yes, it is also present in exoskeleton of arthropods. Done? Done. Okay. Chaliye. So, cell wall. So, firstly, let's discuss what NCERT is saying. Then I will explain it in my way. So, NCERT is saying, bache, that a non-living rigid structure, cell wall, forms an outer covering for the plasma membrane of fungi and plant. It not only gives the shape, matlab it is providing the shape, it is protecting the cell from mechanical damage and from the infection as well. Plus, it is also helping in cell-to-cell interaction. So, functions are important here indeed. Functions are important here indeed. Right, bache? Right, bache? So, undesirable macromolecules cannot, right, cross through it. Though cell wall is permeable. It is permeable, but undesirable macromolecules cannot cross. So for cell-to-cell interaction, for giving it the shape, for protecting the cell from mechanical engineering, I'm very sorry, not mechanical engineering, from mechanical damage, right? From mechanical damage, right? It is going to protect the cell. So one point that you need to highlight here is, algae have cell wall. Algae have cell wall that is made up of cellulose. Algae is also a plant. Algae is also plant. It is having cellulose, galactins and mannans. They are also the polysaccharides and minerals like calcium carbonate, right? But other plants are having cellulose, hemicellulose, pectin and proteins. Please mark it. Please mark it. Hello, wake up guys. Please mark it. Okay? Okay. Done? Sure? Are you sure? Done? Tell me please. Done? Sure? So cell wall of a young plant. Chalo, theek hai. So this part clear? Now, let me discuss that. See, you know that in the case of plants, right, we have the meristematic cells also. Yeah, we have the meristematic tissue. So meristematic tissue will have small size cell. It is going to contain small size cell, and that small size cells, when a cell is small in size, it is more active. When a cell is small in size, it is more active. It is immature. So here you people will see a very large nucleus, or you can say that conspicuous nucleus. That's what we say. Dense cytoplasm. The cell here are small sized, or you can say that immature, right? Conspicuous nucleus is there, dense cytoplasm is there. I'm talking about the meristematic tissue here, and you can say that no intercellular junctions are there. No intercellular junctions are there. No intercellular spaces are there. It is a sign that I'm tired now. No intercellular spaces are there, and obviously abundant plasmodesmatal connections are there. I'm talking about the meristematic tissue. When I talk about the meristematic tissue of a plant, right, I know these kind of things are there. These cells, na bache, these cells, they are having thin cell wall. These cells, they are having a very thin cell wall. Why am I talking about it? Because we have already covered this chapter, the anatomy chapter, right? The plant for physiology chapter, right? We have already covered it. That's why I'm telling you this. So they are having the thin cell wall, right? And that is the primary cell wall. If you will read it from NCERT, in the starting itself, it is given a cell wall is an outer layer. It is outside the cell membrane and it is non-living, right? It is non-living. But here we are saying primary cell wall. It is thin and it is living. It is living, right? And here, because these cells, they are immature, they are dividing. They are just dividing. They are having the primary cell wall. Now, when this plant cell, when it will become mature, right? Mature means its size will increase, of course, right? It will acquire a permanent function. Then in that case you will see that it will also contain, because now it is a mature cell, right? It is also going to contain what? It will also contain secondary cell wall. Just here, this is the primary cell wall, this outermost white in color. And what is this? Inside this primary cell wall, this part is the secondary cell wall here, right? And inside the secondary cell wall here, you people will see the cell membrane. Actually, this is the sequence in the case of a plant cell. Remember it. It is the sequence in the case of a plant cell. Just remember it. It is the sequence. So basically, when you talk about the cell wall here in the plant cell, when you are talking about the primary cell wall, because when a cell is immature, it is having primary cell wall. When a cell is small in size, when it is dividing, when it is active, it is having what? Primary cell wall. And this cell wall is living, right? Because of the cell wall, the cell is capable of growing, right? It will show that division. Okay, bache? So it is the living structure. So here when you talk about the growth now, it is going to be intussusception. Do you know that? It is intussusception. What is the meaning of that? It means internal growth is there. The kind of growth which living organisms show. It is a kind of growth that living organisms show. Internally they grow. So primary cell wall, it grows internally. And let me tell you students, in the case of plant cell, how do plant cells show growth? How do they elongate? Water will enter there. Water will enter there and the turgor pressure will be created, right? Even the cell wall will also kind of expand. That will help in elongation and ultimately the maturation there, right? So the cell wall is very important. It is providing these cell wall is very important. It is providing it the structural stability, that rigidity. It is also helping in the growth. Are you getting my point? Are you getting my point? So primary cell wall is living, and in the case of living things, the growth is through intussusception. The growth is, how the growth is possible? It is possible through intussusception. Internal growth is there. Internal growth is there, right? Just like the living organism. Now when you talk about the secondary cell wall, so secondary cell wall you guys are going to see in the case of mature cells, because once it is formed, the cell will no longer divide. That plant cell will no longer divide. Obviously composition also varies. So this layer is considered as dead, right? So here, how come the growth will occur? It will be accretion. What is this accretion? A for accretion, A for accumulation. A for accretion, A for accumulation, right? Matlab deposition. It means deposition will be there, right? Like in the case of secondary cell wall, I made the layers, right? Just say one layer will form, over it another layer will form, over it another layer will form, over it another layer will form. It is like that. Just say, let's say this is S1, this is second S3. So it is like this. It is like this, right? The older one, the new one layer. So accretion is there. It is formed by accumulation. Just say one layer, over it another layer will get deposited, over it another layer will get deposited. It is like this. Are you getting my point? It is like this. This is the secondary cell wall. Deposition will occur, right? So inner to it will be the plasma membrane. And if, let's say, if there is the tertiary wall, in some gymnosperms it is possible. So inner to the secondary cell wall, your tertiary wall will form. So what is the sequence in the plant cell? Plasma membrane towards inner side, then secondary cell wall, the layers of secondary cell wall, and then there will be primary cell wall, and finally middle lamella will be there. Middle lamella will be there. Now let me explain what this middle lamella is. So imagine one plant cell, another plant cell, another plant cell, another plant cell. ठीक है बच्चे? बच्चे diagram is not that appropriate, but trust me, it will make this topic easy and understandable. These are the plant cells. In the case of these plant cells, bache, remember this thing please. Remember this thing. Just this pinky colored is primary cell wall. Right? I'm trying to explain you. No doubt once the cell will become mature, you will see the secondary cell wall there, but I'm just trying to explain you. Then you have this secondary cell wall. The layers of secondary cell wall. This yellow colored is your plasma membrane, and this green colored here is what? It is middle lamella. It is middle lamella. Students, it is a cementing layer that we are going to see in the case of plant cells. What's that? It is a cementing layer that we are going to see in the case of plant cells. What is it, bache? It is the cementing layer that we are going to see in the case of what? In the case of plant cell. Right, it will connect one plant cell to another, one plant cell to another. So outermost is middle lamella, then primary cell wall, then secondary, then finally plasma membrane. Then finally plasma membrane. The arrangement is like this. Now there is a very famous question that middle lamella is made up of calcium and magnesium pectate. That is the calcium, that is the composition here of this middle lamella. That is the composition here of this middle lamella, that it is made up of what? It is a thin, sticky, amorphous layer. It is a cementing layer, bache. Right? Right? Yes? So it is calcium pectate, right? Calcium pectate is making it. And one more thing I would like to highlight, when you talk about this middle lamella, na, it is not present on the outer exposed surface. I'm writing it here. In the diagram, it is appearing like this, na, it is present on the outer exposed surface. No. Middle lamella, because this diagram can be deceptive then, middle lamella is not present towards outer or exposed surface of plant cell. It is not present on outer or exposed surface of plant cell. Are you getting my point? This middle lamella, it is not present on outer, so it is present in between these plant cells. It will help to join them. That's why it is the cementing layer. So it will form during the cell division only. Right? The next chapter is the cell cycle and cell division. So we will see that during cytokinesis, a cell plate will form. Right? That cell plate is actually forming this middle lamella. Right? This cell plate will grow. So actually in the pectin, the calcium will get deposited. It will become hardened. That's why we are saying it is having calcium pectate. It is made up of calcium pectate. And bache, in the ripening fruit, right? Again this pectate, it will become some, kind of soft. The softening will occur. Just ripening fruit, they are soft because this pectate. What will happen to this pectate there, bache? What will happen to this pectate there? Obviously, its softening will occur. Is that clear? Is that clear? Is that clear? It will solubilize. It will make the fruit soft. Okay. Done. Okay. Sure. So middle lamella is present there. So it is not on the outer exposed surface. It is there in the middle only. That's why the name is middle lamella. Now, you know, in the case of plants, right? Just in the case of animals, we have junctions now. We have that desmosomes. We have that gap junctions and all. In the case of plant, we are also having a kind of gap junction. That P for plant, P for plasmodesmata. Just we say that in the meristematic tissue, the cells are having abundant plasmodesmatal connections. That's what we say now. The cells are having abundant plasmodesmatal connection. So in the case of plant cells also, right there we will see a kind of gap junction there. We will see a kind of continuous junction there, right? We will see a kind of what? We will see a kind of continuous junction there, right, bache? That is the plasmodesmata. What's that? What's that? That is the plasmodesmata, that continuous cytoplasmic connection. That is what plasmodesmata is there. Plasmodesmata is there. P for plant, P for plasmodesmata. Is that clear? Is that clear? So these are the things that you people should know. Okay, so look at this structure here, right? The plasma membrane is there, primary cell wall is there, middle lamella is there, right? Middle lamella is there, right, bache? Right. Okay, so I have many diagrams. So see this, right? So firstly, plasma membrane, then secondary cell wall, layers of secondary cell wall, then you have primary cell wall, then you have middle lamella, right? So see here, A, primary cell wall here, then secondary will form and primary will become like this. Okay, so once the cell is getting mature, it will have this secondary cell wall. Is that clear? Is that clear? Right? So ultimately, what do we have here? See, remember, cellulose, very important. But this cellulose is a polysaccharide. What is it? It is a polysaccharide and it is unbranched, right? Unbranched, right? It is having that beta glucose. It is a polysaccharide. It's a polysaccharide. So these cellulose molecules, many cellulose molecules together, they are going to form microfibrils. So microfibrils are nothing. They are the bundles of cellulose. What are these microfibrils, bache? Cellulose, as I said, it is unbranched polysaccharide. Right, bache? It is made up of glucose. Right? Ultimately, it is made up of glucose. Glucose is going to make your cellulose. Right? Glucose is going to make your cellulose. So cellulose molecules, they are going to form bundles. That bundles are known as microfibrils. That bundles are known as microfibrils. Right? Right. Yes or no? Yes? Right? Right? So obviously these microfibrils, so microfibrils are the bundles of cellulose. So they are arranged. I'm not talking about their orientation. They are oriented at 90 degree, this degree. I'm not talking about that. Simple thing that you have to remember is that cellulose is an unbranched polysaccharide of glucose. Many glucose subunits together will form cellulose. And when that cellulose together come up as bundles, they are forming the microfibrils. These microfibrils are oriented in a proper direction. They are present in that matrix there. And these microfibrils, they are having very high tensile strength. Do you know that? Right? Just like steel, they are having very high tensile strength. Okay? And here in the matrix of the cell wall, you will see this cellulose is there. Even hemicellulose, which is branched. Even pectins are present there. Water is present there. Proteins are present there. Are you getting my point? Are you getting my point? Yes? Done? So matrix में you have polysaccharide, hemicellulose, pectin, you have water, you have proteins there. Right, बच्चे? Yes? Right? Okay, so now just now I told you that cell membrane can also protect, right? Cell wall can also protect. And look at the structure: secondary cell wall in layers, primary cell wall, and middle lamella. Just have a look. Okay, so hemicellulose and all these pectins and all, they are present. Okay, they are present. You can see here, right? Sometimes you know that lignin and all these depositions are also there. So cellulose microfibrils, they are very important. They have very high tensile strength. Clear? Now, bache, this pectin, it can even hold water. It is having the capability, it can even hold water. Hold water. So if it can hold water, obviously it can form gel-like consistency. Am I right? It can form gel-like consistency if it can hold water. Tell me, it can form gel-like consistency if it can hold water, right? So this pectin is capable of fighting with the infection. Remember that it can hold water, it can form gel which is present here in between the spaces. So whenever a plant is attacked by pathogen, so fragment of pectin, they will release a defensive response. Right, so pectin, when it is purified, right, this purified pectin is used to give gel-like consistency to jams, to jellies, right? This purified pectin is used to give gel-like consistency to the jams and to jellies, right? Jelly. So hemicellulose, sometimes lignin is also deposited. You know about the sclerenchyma and all, right? Bache, so bacterial cell wall mein peptidoglycans are there, murines, right? NAM, NAG, N-acetyl glucosamine, N-acetylmuramic acid. They will join, they will form this. Fungal cell wall mein you will see chitin. So it is a polysaccharide. It is a polymer of NAG, N-acetyl glucosamine. So it's a wonderful, right? It is a wonderful flowchart. But yes, you will understand it once we complete your, what do we say, biomolecules. Okay, so middle lamella thing is very important, bache. That middle lamella thing is very important, right? So that plasmodesmatal connection, right? Middle lamella composition, right? It is made up of calcium pectate. It holds, which holds or glues the different neighboring cells together. So if this middle lamella is not present on the exposed side of the plant cells, keep it in your mind, okay? Keep it in your mind. So what do you want me to start? Endomembrane system or cytoskeletal element? Tell me, what do you want me to start? Endomembrane system or cytoskeletal element? Yes. Chali, let's start it after a break. See, you are not even asking. I'm still giving you the break. So let's meet at 11:30. Okay, and now we will just go very fast. Just say we have to complete endomembrane system. There is nothing much there. Then we will move to the further organelles. Mitochondria we have already covered in respiration in plants. Chloroplast we have discussed that in the plastids. We have discussed that in detail even in the what do we say, in the plant physiology. Okay, so we'll finish endomembrane system. We will discuss after that. We will talk about that microbodies. Okay, we are going to talk about what? We will talk about the cytoskeletal elements. Then what's in the remaining time? We will just read the NCERT for mitochondria and chloroplast because we have already covered it. Okay, so after this break, I need 2 hours and then we will finish the chapter. Okay, we'll finish it in detail. So see you all at 11:30. Take care. Yes, now I hope I'm perfectly audible and visible. So students, next thing that we need to start is endomembrane system, right? So here we are going to talk about the organelles which are working together in a coordinated manner. That is what we have to discuss. What are we going to discuss here, students? We are going to talk about the organelles that are going to work in a coordinated manner. Like we have the girl complex there, where G stands for Golgi complex or Golgi apparatus. ER stands for your endoplasmic reticulum and L stands for your lysosome. Plus here in endomembrane system we also talk about the vacuole. So that is what we have to discuss here, right bache? That is what we have to discuss here, right bache? So when we talk about the endomembrane system, it is clearly mentioned here that each of the membranous organelle, they are distinct, right, in terms of their structure and function. Their structure and function, it varies, it is different. Are you getting my point? Ma'am, I'm 26-year-old, this is my third attempt. First of all, Ziaur, what have you done before? And I don't know about this age limit thing. As of now, there is nothing like that, right? Let's see what are they going to implement, right? So rather than just thinking about it, I think you should focus on your studies. I think you should focus on the revision, okay? Right? Just crack the NEET this year, forget everything, just focus on your preparation, right? No need to focus on other things. Okay, chali. So these membranous organelle, they work in a coordinated manner, right bache? Right, usually organelles, they are different in terms of their structure and function, but many of these are considered together. Why? Because their functions are coordinated. So as I said, here we are talking about the endoplasmic reticulum, Golgi complex, lysosome and the vacuoles bache. So mitochondria, chloroplast, peroxisomes, they are not coordinated with the above components. So they are not the part of endomembrane system. Though I don't know what's the need for this particular line, but yes, remember when you are talking about your this endomembrane system, so endoplasmic reticulum, Golgi apparatus, lysosome, right bache, and the vacuole is there. So they all are single membrane bound organelle, right? They all are what? They are single membrane bound organelle. So we are going to start with the endoplasmic reticulum first. Okay, what are we going to start? We are going to start with the endomembrane, endoplasmic reticulum first, and then we'll talk about the another thing. Okay? So any doubt here? Yes. Do you have any doubt here? Yes, students, please tell me. Do you have any doubt here? Sure? Chaliye. So when you talk about the endoplasmic reticulum, we cannot see the structure of endoplasmic reticulum under simple microscope. We need electron microscope for that, right? So this is also important, right? So electron microscopic studies of eukaryotic cell, they reveal the presence of a network. So when you use the word reticulum, when we use the word reticulum, reticulum means network, my dear students, right bache? Right bache? So it is having many tiny small tubular structures scattered in the cytoplasm, right? So it divides the intracellular space into two distinct compartments. So are we going to proceed in this way? No, we have to discuss few extra things here also, right bache? Right? So what are we saying? We are talking about the endoplasmic reticulum. We are talking about the endoplasmic reticulum. So it is present in almost all eukaryotic cells. First of all, it is present in almost, the word is almost all eukaryotic cell. So bache, I have used the word almost. I have used the word, right? Which word I have used here? I have used the word almost. So in almost all eukaryotic cells, you are going to see this, right? You are going to see this. 1 minute. Just 1 minute. So you are going to see this, right? So but we have, just say your mature, yes bache, mature RBCs, they lack endoplasmic reticulum, right? So ova, that is female gamete, mature RBC, they lack endoplasmic reticulum. Are you getting my point? They do lack endoplasmic reticulum. Are you getting my point? Yes. Are you getting my point? They do lack endoplasmic reticulum, right, bache? Right. And its amount also, it varies. Its amount also, it varies. So when we talk about its structure, so in the structure, we will talk about the cisternae, tubules, and vesicles, right? The same three forms we will also discuss while discussing the Golgi complex. So what do we have here? We have the cisternae, we have the vesicles, we have the tubules here, right, bache? So they are, these cisternae, what are they? They are long, right? They are long and unbranched, right? Unbranched units. They are long, unbranched, even you can say flattened units. Okay, they are long, unbranched, flat units, bache, and they are arranged in stacks. They are arranged in stacks like this. Something like this. Cisternae, right, bache? Right, bache? Yes. Okay, so when you are saying tubules, they are irregular structures. Plus, right, they are irregular structures, plus bache, they are often branched, right? They are often branched. So they may be or may not be associated with the cisternae. And when you are talking about the vesicle, they are kind of round, or you can say that oval structures they are, right? Oval structures they are, something like this. Are you getting my point? So these are the structures when you are talking about what? When we are talking about the endoplasmic reticulum. Is that clear? So cisternae is going to be flat. It is going to be unbranched. They are stacked parallel to one another. Is that clear? Is that clear? Yes or no? Is that clear? Yes or no? Yes or no? Yes or no? Tell me. Please tell me. Done? So now, bache, when you are talking about the cisternae, as I told you clearly that they are unbranched, they are not branched. So in their case, just remember one thing. Just say it is a kind of sac-like structure. It is a kind of sac-like structure, a pouch-like structure. Definitely it is covered with a membrane. So in the case of cisternae, you are going to see some ribophorins. I know it is not given in NCERT, but I believe you should know this, right? So they have the ribophorins, and because of that ribophorins, ribosomes are able to get attached to the cisternae. So basically these cisternae, because when ribosomes are attached to it, they will form RER. So what is this RER, bache? What is this RER? It is the rough endoplasmic reticulum. What is this RER? It is the rough endoplasmic reticulum. So these ribophorins are present there. These ribophorins are present there, right? Ribophorin 1 or ribophorins 2. They are present here, my dear students. And this is what? This is the glycoprotein. What are they? They are the glycoproteins. So these glycoproteins, because they are present here in the cisternae, with the help of that, right, this ribosome is able to get attached here. So if there are no ribophorins, ribosomes cannot attached here, cannot get attached, right? So in the case of, so this cisternae, they are forming your rough endoplasmic reticulum. They are forming your rough endoplasmic reticulum. And if you know the structure, just say when we talk about the nucleus, we say na outer membrane and inner membrane. We say na outer membrane or inner membrane, and we say this outer membrane is continuous with what? It is continuous with this rough endoplasmic reticulum, if you remember, right? So they are, so these cisternae here are bearing ribosome, right, bache? And it is possible because of the ribophorins. So because it is bearing the ribosome, so don't you think that protein, ribosomes play a role in the protein synthesis? They will form the protein, right? They are going to form the protein. So wherever, right, in the cells where there is active protein synthesis is going on, you will see that more number of rough endoplasmic reticulum is present there, right? More number of rough endoplasmic reticulum is present there. Is that clear? Is that clear? Yes or no? Yes or no? Yes or no? Yes, bache? Yes, bache? Sure. Done? Chale. So now when you talk about the endoplasmic reticulum, they are also having the matrix. And bache, that matrix, जैसे surrounding में it is the cytoplasm. So this matrix is different from the cytoplasm. Are you getting my point? This matrix is different from the cytoplasm. So when you talk about this endoplasmic reticulum, it is given in the NCERT na? So this is the lumen part here, or we can say that luminal side, yeah, lumen side here, and outside is, this is going to be the extra luminal side. It is going to be the extra luminal side. It is like that. Just let me show you this thing from the NCERT. See, it is given in your NCERT that ER, endoplasmic reticulum, divides the intracellular, in between the cell space, into two distinct compartments. Because it's a membrane bound structure, definitely it will do the compartmentalization of the cytoplasm. And how is it dividing? It is having one, this luminal side inside the endoplasmic reticulum, and this one is the extraluminal side. So when you are talking about the extraluminal side, you are talking about the cytoplasm here. And remember, the endoplasmic reticulum is also having the matrix, which is different from the cytoplasm. Is that clear? Is that clear? Okay. Okay, now in the case of plant cells, I told you about the plasmodesmatal connection. I told you about that continuous cytoplasmic connection. Yes, I told you about the continuous cytoplasmic connection there. We call them as plasmodesmata. We call it as plasmodesmata. So, bache, endoplasmic reticulum, right? This ER, through this plasmodesmata, it can transfer from one cell to another. And how? In the form of desmotibules. So if you ever read this word desmotibules, you are talking about the endoplasmic reticulum in the plant cell. Right, bache? Right, bache? Yes? Yes or no? Yes? Done? Done? So desmotibules, so in the plant cell, endoplasmic reticulum can pass from one cell to another, in the form of desmotibules, through plasmodesmatal connection. Through what? Through plasmodesmatal connection. So yes, do remember this thing. Okay, do remember this thing. Okay, okay, chale. So this cistern thing clear? Okay, so here they are saying that, see, we have two types of endoplasmic reticulum, rough and smooth. So if ribosomes are attached, it is going to be rough endoplasmic reticulum. In the absence of ribosome, the smooth surface is there and smooth endoplasmic reticulum. So obviously ribosome is associated with the protein synthesis. So the cells which are associated with the active protein synthesis, they have more RER. Right, bache? So they are extensive and continuous with the outer membrane of the nucleus. Already explained you this. Okay, bache? Already explained you this. This is what we are talking. Right? And do you know ER? The structure of ER is quite dynamic. Matlab, this endoplasmic reticulum can form fragments. They can reform. Some fragments can also join. Right? Its fragment can also join. Keep it in your mind. Okay? Its fragment can also join. Just keep it in your mind. Okay? Okay, okay, okay. Yes. Is that clear? Sure. Right. So can I say that from the HITS structure, can I say that cisternae forms the rough endoplasmic reticulum? Cisternae forms the rough endoplasmic reticulum, and tubules and vesicles, they are free from ribosomes. They don't have ribophorins. They are free from ribosomes. So can I say that they are forming smooth endoplasmic reticulum? Yes. Can I say that they are forming smooth endoplasmic reticulum? Yes or no? Yes, bache? Yes or no? Please tell me. Then we will talk about their function. Now these organelles, we can finish it like this. Right? We can finish it like this. So tell me, is it possible or not? Because your cisternae, it is having the ribophorins. Because it is having the ribophorins, the ribosomes are attached to it. But your tubules and vesicles, they do not have ribophorins. They are smooth. They are smooth. Right, bache? So they are free from ribosome. So they are forming the SCR. Can I say so? Can I say so? So when you are talking about your rough endoplasmic reticulum, right, you will see it in the cell where active protein synthesis is there. जैसे pancreatic cells में, right, we'll see, मतलब you will see their number more in the pancreatic cells. Are you getting my point? Where are you going to see this? In the pancreatic cell, and not just the pancreatic cell. You can take the example of some plasma cells. You can take the example of goblet cells, because it is going to form the proteins, and obviously it will secrete it as well. It will secrete it as well. Are you getting my point? Are you getting my point? Yes, bachche. Yes, bachche. Yes. Okay, so here let's say ribosomes, they are forming the proteins. So there is a kind of pore here by which that proteins will enter here in the lumen, and in the lumen also there are some enzymes which will help in the modification of that protein. We will see that how endoplasmic reticulum and Golgi complex, they are associated with each other, right? They are helping in that modification and all. We are going to discuss that. Are you getting my point? So this part clear. Now when it is smooth endoplasmic reticulum, where are you going to see it? Basically, as I said, your tubules, your vesicles, they are forming the smooth endoplasmic reticulum. So in the case of your adipose cells, you'll see their number. Muscle cells may, right, their number will be more. So remember, it is your smooth endoplasmic reticulum. It is a very important function of smooth endoplasmic reticulum. It is a storehouse of calcium, right? It is a storehouse of calcium ions. Remember, just in the muscles, we say sarcoplasmic reticulum. The SR is there, sarcoplasmic reticulum is there, SR is there, sarcoplasmic reticulum. So it is the storehouse of calcium. So that sarcoplasmic reticulum, which is the storehouse of calcium, actually it is what? It is your smooth endoplasmic reticulum. It is your smooth endoplasmic reticulum. And you know what? When it comes to the cholesterol synthesis, right, your smooth endoplasmic reticulum is involved. Yeah, when there is the steroidal hormone synthesis, because for steroidal hormones we need this cholesterol also na. So this smooth endoplasmic reticulum is required. So these are one of the most important functions here, right bache? Right. So formation, right? So remember, when you are talking about the SCR, this calcium storage thing is a very important point. I think I should highlight these things. When it comes to their functions. Matlab, overall, you should know the functions of each and every organelle, but I'm just being very specific that when you are talking about the SCR, never forget that it is the storehouse of calcium. It is the storehouse of calcium, and we have studied its role in the case of what? We have studied this thing in the case of what, bache? We have studied this thing in the case of muscles as well, right? Right, bache? Right, bache? So not just जैसे your adipose tissue, because for the fats, the steroidal hormone synthesis, it is playing role. Interstitial cells, adrenal cortex cells, they all are having your smooth endoplasmic reticulum. They all are having your smooth endoplasmic reticulum. Is that clear? Is that clear? Muscle cells, of course, right? Muscle cells, of course. Is that clear? Sure. Sure. So synthesis of lipids, right? Synthesis of lipids, right? Matlab, it is providing a surface for that. So you will include your cholesterols also, right? Steroidal hormones also. So for their synthesis, we need that thing, right? We need the smooth endoplasmic reticulum. Is that clear? Is that clear? Right? So sex hormones, hormones of adrenal, you know na, steroidal hormones, sex hormones, androgens, they all are steroidal in nature. Hormones of your adrenal gland, and to be more precise, adrenal cortex, right? They all will be synthesized with the help of SCR. Okay, bache? Okay, bache? Yes. Done? Okay, so it helps in the detoxification. It's a very important detoxification. It's a very important role here. Jaise I still remember when I was a student, I gave an entrance test. Right now I don't remember. It was for PhD actually, and in that entrance test that question was there. This question was there that in the liver cells, in the hepatic cells, in liver cells, which organelle plays role in the detoxification? So when you talk about the detoxification, so detoxification in liver cells is done by smooth endoplasmic reticulum. Right, it is done by smooth endoplasmic reticulum. So this in the liver cells, just say, let's say if there is a drug, alcohol, so they will be detoxified. So for such kind of toxins, na hydroxyl group will be added to that. They'll be made soluble and then they'll be excrete out from the body. So these are the things that you need to remember. Okay, bache? Okay. 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 Any doubt? Any doubt? Any doubt? So this is the function of your smooth endoplasmic reticulum. Yes? Is that clear? Any doubt? Sure, bache? ठीक है. So even it is also said that smooth endoplasmic reticulum helps in the formation of other organelles like your Golgi apparatus, vacuole and lysosomes. Right? मतलब it also play the role there. Okay? Okay, okay, okay. चलिए. So I'm writing it here. So this helps in the formation of Golgi complex, lysosomes. Okay, your smooth endoplasmic reticulum. Is that clear? Right? Even for the vacuoles also. Now, when it is the RER, when it is the rough endoplasmic reticulum, so obviously, can I say that it is providing a site for ribosome attachment? It is providing a site for ribosome attachment. And how is it possible? It is possible due to the ribophorins. Now, it also helps in the modification of proteins. This rough endoplasmic reticulum, it also helps in the modification of proteins. Jaise bache, please imagine this thing. Here you have this rough endoplasmic reticulum. Here you have the ribosome. Ribosomes will make protein, that protein will enter here. And here they will be modified. Matlab their phosphorylation can be done, or if they can be converted into glycoproteins, so their modifications will be done here. That is their role. Plus, in these endoplasmic reticulum, from these endoplasmic reticulum, this particular protein, right, some vesicles will come out from this rough endoplasmic reticulum, and these vesicles, they are having that protein. So it will then fuse with the Golgi apparatus. It is going to fuse with the Golgi apparatus. No doubt it will be done through, just let's say if it is having something modified, it will give it to SCR, then finally to the Golgi apparatus, and further its modification, your transport and packaging will be done. So that is also the function of RER. So packaging may it is also playing the role. So it is going to modify the protein, right bache? So that will be that from ER a vesicle will bud off, and then it will carry the protein to the Golgi apparatus, where it will be further processed, packed, and it will further pass to the secretory vesicles. Is that clear? Is that clear? So that is how it works. That is how it works. And from this rough endoplasmic reticulum, if required, the ribosomes will get removed from it, and this rough endoplasmic reticulum can form smooth endoplasmic reticulum. Okay? It can form smooth endoplasmic reticulum. So the functions here are very important. अब ये देखो, जैसे smooth endoplasmic reticulum, major site for synthesis of lipid. In animal cells, steroidal hormones are synthesized here. Very important thing it is, right? So you should know the other functions here also. So look at this diagram. this endomembrane system, just have a look. So with the outer membrane of the outer nuclear membrane, because your nucleus is what? Your nucleus is what? Your nucleus is a double membrane bound organelle. What is it? It is a double membrane bound organelle. Okay? So with its outer membrane, your rough endoplasmic reticulum is in continuation. Your rough endoplasmic reticulum is in continuation. Are you getting my point? Are you getting my point? And here you guys can see, this is the rough endoplasmic reticulum, and here you people can see the Golgi apparatus. This is the Golgi apparatus, and from this Golgi apparatus also you will see some vesicles will bud off. Just say it is the secretory vesicle. These are some transitional vesicles which are coming from this endoplasmic reticulum, and here the lysosomes will also bud off from this Golgi apparatus, right? So its structure is also like this. ये देखो, these are cisternae, sac-like structure, and even in endoplasmic reticulum also, बच्चे, the different components, they communicate with each other, right? Right, so see, transport vesicles from ER, they will fuse with this Golgi apparatus, and here secretory vesicles are formed having properly modified protein. If it is required, it will be released out from the cell by a plasma membrane. This is exocytosis. The lysosomes are also formed from it. So this is what we need to understand now, right? This is what we need to understand now. So the next organelle that we have to study is Golgi apparatus. Okay, so Camillo Golgi, 1898. Camillo Golgi, 1898, he first observed some densely stained reticular. Again, it's a kind of network, and it is near the nucleus. So they were later named Golgi bodies after his name, right? So it is having flat, disc-shaped, some sac, pouch-like structure or cisternae of 0.5 micrometer to 1 micrometer diameter. Is that clear? From 0.5 micrometer to 1 micrometer diameter. Is that clear? Is that clear? Yes. Is that clear? Okay. So when you talk about it, definitely eukaryotic cells will contain it. So mature RBC mein again it is absent, right? Even in sieve tube cells, it is absent, right? It is absent there. Are you getting my point? Are you getting my point? Yes. So sperm cells of bryophytes, because that is also a eukaryotic cell, in their case also it is absent. But overall, yes, it's a fact that majorly we will see it in the secretory cells, right? In the secretory cells we are going to see it. We are going to see this. Is that clear? Is that clear? So they are also stacked parallel to each other, and their number of cisternae, varied number of cisternae are present there. Okay, bache? So now we have to understand something. Let's say this is a nucleus. This is your But when you read about the cell, now I know some of you will be like, yeah, ma'am, it's a very easy chapter. Yeah, it is a very boring chapter. Yeah, it is like this. Actually, when at molecular level, when we study these things, we get to know that there are different types of signals which are given to these proteins, right? That will tell this protein that whether it has to go for the exocytosis or it has to be there in a cell. There are different things like that, how things are going to move from endoplasmic reticulum to Golgi apparatus. At molecular level, we see some signals are present there, right? So cell is also having its language, obviously in the form of these chemicals, in the form of these compounds. Okay. So when we discuss the structure of the Golgi body, here also of the Golgi apparatus or Golgi body, here also, bache, you will talk about cisternae, tubules, and the vesicles here, right? Here also we will discuss it in the form of these cisternae, tubules, and vesicle. Are you getting my point? So cisternae, I already told you, they are flat disc shaped, right? They are flat disc shaped, and when it is the tubules, they are branched, irregular tube-like structure, right? It is going to be same now. It is branched, so they are branched. Remember this: irregular tube-like structure, and it is associated with cisternae. And vesicles, they are what? They are oval या फिर rounded, है ना? These vesicles. And one more thing that you have to remember here is that मतलब we'll talk about two types of vesicles, transitional or secretory. Transitional or secretory. Is that clear? Transitional or secretory vesicles are there. Is that clear? Yes, बच्चे. Is that clear? Sure? Sure? ठीक है. So when you talk about this Golgi apparatus, मतलब the cytoplasm in which it is present, मतलब it is known as zone of exclusion. Do you know that? We use the word zone of exclusion here. Do you know that? मतलब this organelle is having the polarity. First thing, this organelle is having the polarity, right, बच्चे? This organelle, it is having the polarity, and what am I saying here? I am saying that it is present in the zone of exclusion. What is this zone of exclusion? So Golgi is surrounded by a zone of cytoplasm in which other cell organelles are absent, right, बच्चे? Golgi is present at the zone of cytoplasm where other organelles are absent. Are you getting my point? Where other organelles are absent. Yes. They are absent. So this is the zone of exclusion. This is the zone of exclusion. Now I am saying that this is the organelle having polarity. Matlab it is having two faces. The one which is facing the nucleus. The one which is facing the nucleus. It is your cis phase. Forming phase. Please note down, it is something very important. Which phase it is? It is the cis phase, right? It is the cis phase. What is it, bachche? It is the cis phase, forming phase, right? It is the F phase. It is the convex phase. The question will come from this part. Definitely question will come from this part. So convex phase, forming phase, cis phase, F phase it is. These are the different names of the site. Right, bache? So what is it? Cis phase, forming phase, F phase, convex phase, and you can also call it as proximal phase. Proximal phase. Right? And it is towards nucleus. Yeah, near the nucleus. It is near the nucleus. So let me write it properly. So cis phase, F phase, forming phase, proximal phase, and convex phase. Right? Proximal phase or convex phase. So it is near the nucleus. Okay? So then it will be the trans phase, M phase, maturing phase, distal phase, concave phase. So do not get confused in this concave and convex thing. Okay? Do not get confused in this concave and convex thing. Right, bache? Right, bache? Yes. So it is your, right, bache? It is the, right, these are the two sides, and obviously this one is towards the, this one is towards the plasma membrane. This one is near the nucleus or towards the nucleus, and this one is towards the plasma membrane. It's important. Right, this one is towards what? It is towards the plasma membrane. Is that clear? Is that clear? Right? Yes or no? Yes or no? Done? Any doubt? Any doubt? Sure बच्चे? Done? Yes. So now remember one thing. जैसे this Golgi complex, it is बच्चे, it is without, yes, it is without ribosomes. It is without ribosomes. So don't you think it looks like, because it looks like somewhere it looks like smooth endoplasmic reticulum. So this thing is showing that they arise from smooth endoplasmic reticulum. They arise from smooth endoplasmic reticulum, and it is near the nucleus. And you know that here, right, we are having the rough endoplasmic reticulum as well. So even if in the paper it is written that this phase is near the rough endoplasmic reticulum or towards the endoplasmic reticulum, it is correct. It is correct. Do not get confused. Okay. It is correct. Do not get confused. So now it is obvious. Just look at this particular diagram, right? Look at this particular diagram. So देखो बच्चे, right, from rough endoplasmic reticulum a protein will come. मतलब these two sides, they are quite different. They are quite different, right? One side is just like it has to receive something and it has to pass something. मतलब it is basically for the shipment and it is basically to receive something. Are you getting my point? Are you getting my point? जैसे here I would like to mention one thing, that when you talk about these two faces, I told you they are having the polarity, right? मतलब these two sides, they are totally different but interconnected. They are totally different but interconnected. Yes or no? They are totally different but they are interconnected. Yes. Yes, these sides, they are totally different but they are interconnected. Now one side is receiving something, other side will pass the process thing. जैसे from the rough endoplasmic reticulum, in the transport vesicle, transitional vesicle, the proteins, they'll come, they'll fuse with the cis side. Now they will enter in this Golgi complex where they will be modified. They'll be further modified. They'll be packed. They'll be sorted, right? जैसे this Golgi complex ना, do you know that it is also known as the traffic police of the cell? Traffic police of the cell. जैसे traffic police clear the things, no? So it is also what? It is the traffic police of the cell. Do you know that? It is even known as the export house of the cell. Export house of the cell. Do you know that? Yes, बच्चे. It is even sometimes referred to as internal reticular apparatus. Internal reticular apparatus. So it is having different names. So when we say traffic police of the cell, right? Just traffic police will be like, na, they will not allow the vehicles to get gathered. They are going to sort that traffic, that you have to go there, you have to go there. It is like that. So whatever proteins, yeah, whatever things it is going to receive, it will modify it if required. Then it will sort it out, right? It will add that signal ki yes, you have to release out from the cell. Your exocytosis is required. You have to move to this part of the cell. You are needed there. So sorting, packaging, everything will be done. So endoplasmic reticulum and Golgi complex collectively, they are helping there. So in the case of Golgi complex, we majorly say that it helps in packaging material, right? That the material that we have to deliver within the cell, or even if it is required to deliver outside the cell, right, bache? So it has to be. So it will be modified here, and from its forming side, from its mature side, right, in the secretory vesicle, it will come out, right? It will come out. So if it is having a signal to move to the intracellular components, it will go there. Otherwise, if it is having the signal that you have to release out, your exocytosis is required, then accordingly it will be done, right? Then accordingly it will be done, right, bache? So that's why this Golgi complex is in close association with endoplasmic reticulum. It has to do this job. Is that clear? Is that clear? Done? Okay. Yes, bache, is that clear? Fine. So this is the main function here. So it is also a kind of secretory center of the cell. Just mucus, tear, saliva, it will form there in the Golgi apparatus. Then it will be released out. Imagine. So it also play the role in the formation of lysosome. They will also come out from this. They will also bud off from this Golgi complex. So the enzymes of lysosome. This lysosome is synthesized by Golgi complex. Is that clear? Right. So it will form lysosome as well. The enzymes, the content there. And do you know that the material of cell wall, the material of plant cell wall, again Golgi apparatus will form it, right? Do you remember that middle lamella? Just during cell division, during plant cell division, during cytokinesis, cell plate form in between. That cell plate form in between, that is having that calcium pectate. That is also done by Golgi apparatus. So that mucus secretion, tear and all, that is also the job of this Golgi apparatus. And a very important question that they ask us, formation of acrosome. Do you know that? Matlab cell wall ka material, right? And this acrosome. So acrosome, if you remember, it is there in the sperm. This is sperm may, in the case of sperm, right, this at the tip, this cap-like structure, it is the acrosome. And this acrosome is having sperm license. The enzymes that will help during fertilization, the enzymes that will help in the infusing that egg membrane. So acrosome synthesis is also done by this Golgi apparatus. So it is forming that lysosome also, that acrosome also, cell wall material also, middle lamella also, and very important, it is a site of glycosylation and glycosidation. That's a PYQ as well, right? That glycolipid, glycoproteins will form here. So anybody in the class who can tell me what is the meaning of glycosylation and glycosidation? Anybody in the class who can tell me this? What is the meaning of glycosylation and glycosidation? Yes? Please tell me what is the meaning here? Please tell me, anybody in the class. What is glycosylation and what is glycosidation? Yes? But ultimately, if you have to add the sugar to the non-sugar molecule, if you are going to add the sugar to the non-sugar molecule, that is basically your glyco, right? What is this? What is this, bache? Glycosidation, very important step, right? Very important step. Tell me, do you have any idea of it or not? This glycosidation is a very frequently used term. Right, bache? Glycosylation rather, right? Yes, please tell me if you guys are having any doubt here. Do let me know. Do let me know. Right, done. So when it is the glycosylation, the sugar, right, the sugar molecule, basically it will be added. Ultimately you have to add the carbohydrate and the lipid and the protein. You will make glycolipids, you will make glycoprotein. Or glycosidation, it is said that when the sugar is added to another, right, non-sugar molecule, right, to a non-carbohydrate molecule. So that is how it is. So basically, in simple words, you have to remember that if we need to make glycolipids or glycoprotein, that is the job of Golgi apparatus, and it will help in sorting out the things. ठीक है, it will help in sorting out the things, right? So it is arranged concentrically, मतलब the center is same and it is arranged near the nucleus with convex or cis or the forming phase, right? Or the forming phase and concave phase or maturing phase is there. So both the phases are entirely different but they are interconnected. So it performs the function of packaging material to be delivered either to intracellular, matlab within the cell target, or secreted outside the cell. So if it is secreted outside the cell, can I use the word exocytosis? Can I use the word exocytosis? Obviously I can, obviously. So ER fuse with, this endoplasmic reticulum fuse with, right? Basically the transition vesicle from that endoplasmic reticulum fused with the Golgi apparatus, it moved towards the maturing phase. So that's why they are in close association with each other, right bache? So the number of proteins synthesized by ribosome on ER are also modified in the cisternae of Golgi apparatus before they are released. Okay, so it is a very important site for glycoprotein formation and glycolipids. Right, so this is the structure: cis phase, trans phase, secretory vesicle. So they are also making the lysosomes, right? They are also making the lysosomes. Is that clear? Is that clear? They are also making the lysosome. Is that clear? So if it is not clear and if you guys are having any doubt, do let me know. Do let me know. Please do let me know. Right? So it is the traffic police of the cell. It is the traffic police of the cell. Okay, traffic police of the cell, and it is also known as director of this Golgi apparatus, is also known as director of macromolecule traffic in cell. It is going to direct it. Bro, you have to go there. You have to go there. Sometimes it is also referred to as middleman of cell. Okay, so these are the different names here, right? These are the different names here. So next is bache lysosome, which is also known as suicidal bag, which is also known as suicidal bag, which is also known as recycling unit. Do you know that? It is also known as recycling unit. Do you know that? Lysosome. We are talking about what? We are talking about the lysosome, suicidal bag, recycling unit, या recycling space it is. ठीक है बच्चे, right? Its function is also very interesting. Are you aware of it? Are you aware of it? Right? Suicide bag, recycling centers, and sometimes बच्चे they are also referred to as cellular housekeeper. They are also referred to as, just imagine, they are different names, cellular housekeepers. They are also referred to as cellular housekeepers, and sometimes they are also the disposal units. Disposal unit. So who discovered this lysosome? It was discovered by D. Dubé. D. Dubé. That's how I remember. This is discovered by D. Dubé. Right, bache? Right? And yes, I think I mentioned it here. Oh, I didn't. Bache, plant Golgi apparatus is known as, in the case of plant, I think, I don't remember exactly, but I think, I'm not sure about it, I have to check it, that like a scientist also discovered some structure in the plant cell, he named it as dictyosomes, and later they realized that they are basically the Golgi apparatus only. So right now, I don't remember it exactly, but yes, do remember this name, dictyosomes. So in the case of plants, Golgi apparatus are known as dictyosomes, right? We also call it as, right? We also call it as dictyosome. What do we call it, guys? We also call it as dictyosome. Okay? So dictyosome is the Golgi apparatus. Desmotobule is the endoplasmic reticulum. Right? Desmotobule is the endoplasmic reticulum. Is that clear? Is that clear? Sure. Sure. So now lysosomes. Bache, lysosomes, matlab, they are not actually there in the, because the plants are having vacuoles that will help in the food digestion. So lysosomes, they are not mainly there in the plant cells, but in plant cells, lysosomes are spherosomes. They are having different function. I'll discuss that with you all. Okay? And one more thing that you need to remember here is, lysosome disposal unit. Plant cell mein they are spherosomes. Dictysome thing is also done. And here, yes, they are basically, they are with the exception of mammalian RBC. मतलब they are absent in RBCs बच्चे, with the exception of mammalian RBC. They are present in all other animal cells. Right, in all other animal cells. So what kind of structure it is? It is a membrane bound structure. Why do we call it as a suicidal bag? We will discuss it. So it is the membrane bound vesicular structure. It is in the form of vesicles. So it is formed from the Golgi apparatus. Right, so it is a vesicular structure formed by the process of packaging in the Golgi apparatus. So these lysosome vesicles, they are rich in almost all types of hydrolytic enzymes. Hydrolases are present here that can digest lipase, proteins, carbohydrates, right? And they need acidic pH to get activated. They need acidic pH to get activated. Are you getting my point? Are you getting my point? Right, so remember one thing, in the case of plants, when it comes to the digestion of food, they are having the, right, they are having the vacuole. So plant cells, they contain less lysosomes, very less lysosomes, मतलब very few lysosomes or they don't have, and they are spherosomes. Spherosome is something different. I will discuss it. मतलब it is associated here. I will discuss it. And in the fungi, right, fungal cells में तो fungal cells are abundant with lysosomes. जैसे plant cells are having very less, but here they are abundant in lysosomes, right? They are abundant. Fungal cells are abundant in lysosome. So they are having the hydrolases and that need acidic pH, right? So when it comes to that pH, right? When it comes to that pH, yes or no? When it comes to that pH, that is approximately 4 or 5 that is required. Okay, 4 or 5 pH is required for this, for the enzymes to get activated. And obviously if they will get activated, they can digest protein, lipids and nucleic acid. This is about the lysosome in general. Is that clear? Is that clear? Yes, is that clear? Tell me, is that clear? Sure? Okay, so now you have to understand one thing, that these lysosomes, na, this is the organelle that show polymorphism. This is the organelle that shows polymorphism, right bache? This is the organelle that shows polymorphism, right? What is it? It is the organelle that shows what? That shows the polymorphism. It is having the different forms. So I will explain it to you first, right? Just allow me one thing. Okay, chalo 1 second. जैसे when you talk about the lysosome, it is having that outer membrane, but this मतलब it is having hydrolases, right? So that hydrolases, they do not destroy the lysosome membrane. Why? Because of its composition as well. Firstly, these enzymes are in inactive state, and if for the activation of these enzymes, they need the acidic conditions. They need the acidic conditions. So in the lysosome membrane also, you have the H positive pumps, right? What do we have here? Here we have the pumps here, active H positive pump. So if I'm using the word pump, it's clear that obviously against the concentration gradient things will move, yeah, ATP will be used. So H positive will come inside, the conditions will become acidic and the enzymes get activated when required, and I'll tell you why is it required. Secondly, its composition. Just say there is one sialic acid is present in the membrane of lysosome, right? Sialic acid is present in the membrane of lysosome. What is present in the membrane of lysosome? Sialic acid is present in the membrane of lysosome, and that is going to protect its membrane from this degradation by the enzymes. So now I told you that this is the organelle that shows polymorphism, and I told you these are the suicidal bags also. So let's start it. So the very first thing is bache primary lysosome. Now see, This thing is not given in NCERT. This thing is not given in NCERT, but still question can come from this part. Jैसे first thing is the primary lysosome, which is also known as storage granules, right? Which is also known as storage granules. So what is the meaning of these primary lysosomes? They are simple, right? They are very simple, bache, right? These lysosomes, they are very simple. जैसे they are having that inactive enzymes present in it. Right, normally the lysosome structure, you can call it as the primary lysosome also. What can we call it? We can call it as the primary lysosome also. Is that clear? Is that clear? Yes or no? Is that clear? Right, so it is the primary lysosome. What's the word, bache? It is the primary lysosome. Done, bache? Done? Sure? Okay, okay, ठीक है. So right now let's talk about the polymorphism and then I will discuss it further. Okay, then I will discuss it further. Okay, चलिए. So now this is the first thing, the storage granules. Then comes the secondary lysosome. देखो, I told you that in the lysosome you are having the hydrolases, and that hydrolase, for their activation they need the enzyme pH 5. So it's not like that one lysosome will contain all the enzymes. There are 50 different types of acid hydrolases which are present there, right? And we have to pump the H positive within the lysosome so that we can activate it. So next thing is the secondary lysosome, which is the digestive vacuole also, right? Which is the digestive vacuole also, and which is the heterophagosome, and that is the heterophagosome. Now what is it actually? Let's say this is your primary lysosome. When it will get fused with the food vacuole, right? Like if let's say if you want to digest a food, this food has to enter here in the lysosome, then only the enzymes will get activated and they will, obviously at a certain pH they will get activated and they will digest it. So when your this primary lysosome and food vacuole, they get fused, so now it is having the food and this lysosome content will get mixed. This is your secondary lysosome, digestive vacuole or heterophagosome. Any doubt? Any doubt? Any doubt? Now obviously the food will get digested. Now the third type, the third structure will be the residual body here. The third structure is going to be the residual body here. The third structure is going to be the residual body here, right बच्चे? या फिर sometimes you can also call it as tertiary lysosome. So what will it contain? Residual माने remaining, right? Obviously undigested food that needs to get out of cell, right? That we need to throw out from the cell, right? It is the residual body. Okay, so obviously the food will be digested, right? And the undigested, indigestible matter will be there in the secondary lysosome. Now it is the residual body, right bache? Right. Yes, right. So now this residual body, it will fuse with the membrane, and now it has to do the exocytosis. It has to excrete, it has to pass out that indigestible material from its system. Yeah, you can call it as ephagy also. We can call it as ephagy also. Is that clear? Is that clear? Next is the autophagic lysosomes, right? Next is what? Next is the autophagic lysosomes. They are also known as self phagosomes. Now let's say if the cell organelles, if a lysosome, this lysosome, if it is having a cell organelle to destroy, let's say if a cell organelle is not functional, right, it is dead. If a cell organelle is not functional, if it is dead, this cell can digest its own organelle, right bache? So this is the autophagy. This is the autophagy, isn't it? Isn't it? Yes or no? Yes or no? Yes or no? Just say what is going to happen? This is your lysosome. This is your primary lysosome. And here you are having a damaged organelle. You are having what? You are having a damaged organelle. Are you getting my point? What do you have here? You are having a damaged organelle. So these two will fuse and this will form this autophagic vacuole, or you can say that it is a autophagic lysosome, right? So this lysosome can destroy these type of organelles which are not functioning properly. Are you getting my point? Are you getting my point? Yes, are you getting my point? So can we say that it is a disposal unit or recycling center because it is destroying the one which is not working properly? Right, so it is the recycling site, yes or no? It is the recycling site, yes or no? Autophagy clear? Done? Done? Done. So this is the autophagy, right? Now bache, there is one word. So matlab cell can, when required, जैसे lysosome, if there is a stored food in the cell, if there is a damaged organelle, it will eat it, it will destroy it. So this is the autophagy. There is another word, that is autolysis. Now lysis is the word means to destroy. जैसे let's say here in this case, what is going to happen? जैसे because of this function, it is the recycling unit. It is the disposal unit. It is the recycling unit. It is the disposal unit because of this function. And autolysis माने breakdown. जैसे let's say if this is the lysosome, its whole content is released in the cytoplasm, right? The enzymes will become active. They will digest that complete cell. So here it is destroying that complete cell. Now it is acting like a suicidal bag. This is what the autolysis is, right? Right. So this thing will occur during aging when we have to destroy the cell. अब जैसे tadpole, you know that tadpole is the larva of frog. What is tadpole? Larva of frog. When there is the metamorphosis, it will convert into an adult which is tailless. So for eating that tail, the lysosome is going to do that, right? So reabsorption of tail of tadpole is also comes under this autolysis, right? Reabsorption of tail of tadpole, it also comes under this autolysis. Now it is the suicidal bag because it is destroying that complete cell. Let's say it's not functioning properly. Yeah, let's say if there is any other problem, then it is going to be like this. Is that clear? So lysosome is the organelle that shows the polymorphism. Done? Okay. Done? Okay. Fine, chale. So look at this diagram, right? It's a very different one, but I really like it. Golgi carrier vesicles, lysosomes will fuse. When these lysosomes, let's say they are fused here, they are forming autolysosome. They will destroy the organelles there. Now this is the autophagosome. You know what autophagosome is, right, bache? So and one more thing that they are showing, Autolysosomes, that's it. Ki Golgi carrier vesicle, how are they forming the structure? Bus, right? Right. So I was just, when I saw this diagram, I was so happy. Wow, so many things are there. Chalo, I'll show it to my students. So that's why I added it here. Okay, that is why I added it here. So this is all about this thing. Chaliye. So now, guys, let's proceed. See, we are about to finish the chapter in next 1 hour, right? I want you people to stay alert, okay? Because see, mitochondria and chloroplast we have already covered in plant physiology. In detail, we have discussed their structure. Ribosome will hardly take 10 minutes. Then cytoskeletal element is there which need 30 to 40 minutes if you want to understand it in a better way. Right, bache? Right? Otherwise, though, I can just wrap it up. And nucleus, the chromosome and all, that we also discuss in the cell cycle and cell division. So there is no problem in that. Okay? Right? Right? So now let's continue. Let's discuss it. Next thing is the vacuoles, bache. So remember a very famous question. The membrane of vacuole is tonoplast. The membrane of vacuole is tonoplast. What is it? The membrane of vacuole is tonoplast. Okay. So vacuole is also a membrane bound space, bache. It is having water, minerals in it. That is forming the sap, excretory product, other materials which are not useful for the cell. So its membrane is tonoplast, and as I said, it's a PYQ. And in plant cell, the vacuoles can occupy up to 90% of the volume of the cell. Right? In plant cells, these vacuoles can occupy 90% of the volume of cell. That's why the nucleus is towards one side. So this tonoplast, this membrane of vacuole, a very important question. It helps in transport of number of ions and other materials against concentration gradient. So against concentration gradient means we will talk about the active transport. ATP will be used. So again here in the tonoplast also you will see the pumps are present. The pumps are present, right? That will help to move the things against the concentration gradient, right? So other than that, in amoeba is what? Amoeba is a freshwater protozoan. Yes or no? Amoeba is what? It's a freshwater protozoan. Yes. So it is having the contractile vacuole, and how that contractile vacuoles are helpful? They are helpful in osmoregulation. So students, what is this osmoregulation? To maintain the salt and water balance. Is that clear? Is that clear? To maintain the salt and water balance. Is that clear? Sure. To maintain the salt and water balance. And in many cells, as in protists, food vacuoles are formed by engulfing the food particle. That's it. That's what you have to remember here. Is that clear? So cellular digestion in the case of lysosome is autolysis. Then there is the vacuole, and this is what we need to know about the vacuole. That's it. That's all. So food vacuoles, contractile vacuoles, right? The membrane here is the tonoplast. Keep it in your mind. Right, there are some gas vacuoles also. There are some gas vacuoles also. Okay, okay. Done. Next is mitochondria, but I think I forgot to tell you about spherosomes. And I just forgot to tell you about spherosomes. So do you know anything about these spherosomes? They are also considered as plant lysosomes. They are also considered as plant lysosomes, spherosomes. Do you know about them? Do you know about them? Yes? So, bache, they are basically rich in lipids. And contain some proteins as well. So these spherosomes, they are present in endosperm cell. I hope you know what endosperm is. They are present abundantly in the endosperm cell. Is that clear? Is that clear? So they are abundant in endosperm cells of the oil seeds, right? They may arise from the endoplasmic reticulum. Chaliye, done. So as I said, we have already discussed about the mitochondria and chloroplast. So I will just give you a quick recap of that. So are you ready for that, bache? Are you ready for that? Tell me. We'll revise it very quickly. Yes, we will revise it very quickly. Mitochondria and endoplasmic reticulum. Okay, chaliye. So mitochondria, mitochondrion is singular, mitochondria is plural. So you know that it is the powerhouse of the cell. It is what? It is the powerhouse of the cell. So unless specifically stained, they are not easily visible under the microscope. Very important thing, bache. They are not easily visible under the microscope. But on the other hand, plastids, the chloroplast, it is easily visible under the microscope. Right, under the microscope, but it is not if it is not properly stained. So its number can vary. It depends upon the demand of the cell, how much energy it needs. Okay, shape and size, it also vary. Okay, bache. So typically it is sausage shaped. It can be cylindrical when it comes to their diameter. Some students, they ask me, do we need to remember this? Yes, because it is given in NCERT. So diameter is 0.2 to 1 micrometer. Length is 1 to 4.1 micrometer, and they are double membrane bound organelle. Okay, so bache, when we talk about the double membrane bound organelle, right, remember this thing. When we talk about the double membrane bound organelle, about what? About double membrane bound organelle, then we need to discuss that. Then we have mitochondria, powerhouse of the cell. We have this plastids, your chloroplast, and the nucleus. These three are double membrane bound organelle. Is that clear? These three are double membrane bound organelle. So they are having, right, you know it already, outer membrane, inner membrane, in between intermembrane space, in between intermembrane space. So students, we have covered everything, right? So you can watch the lecture of respiration in plant. This is the matrix here, having all the enzymes for Krebs cycles except, yes, do you remember this? Having all enzymes of Krebs cycle except succinate dehydrogenase. Okay, right. So you know that when we talk about the inner membrane here, it will show some enfoldings, and that enfoldings are nothing but the cristae. That is the diagram na mitochondria. That is the diagram of mitochondria. So inner enfolding is what? They are the cristae. They are increasing the surface area, right, bache? So this inner membrane is having what? It is having the electron transport system. And when you talk about the mitochondria, the plastid, they are having their circular DNA, and they have the 70S ribosome. So that is why these organelles, they are, even the plastids, even the this mitochondria, they are semi-autonomous, right? But they can divide by a binary fission. They can just divide by binary fission. And because it is having, right, it is having the circular DNA, it is having the circular DNA and 70S ribosome, obviously it is able to synthesize some proteins. It is not totally dependent on the nucleus. It is not totally dependent on the nucleus. Yes or no? It is not totally dependent on the nucleus. It can synthesize some proteins, right? In the genetics, we also talk about the mitochondrial inheritance, right? Right now, I'm not discussing it. So these are the things that you should know. And you know that it is the powerhouse of the cell because it forms the ATP. Krebs cycle will be there in its matrix. So again, I'm repeating, all the enzymes of Krebs cycle except succinate dehydrogenase is present in the matrix. It is present in the matrix. Clear, bache? Clear? So it is the semi-autonomous organelle. It is believed, right, that endosymbiotic theory, endosymbiotic origin is there for the mitochondria and the plastids. Just it is believed that some, a large cell engulfed the prokaryotic cell, right, and then that prokaryotic cell has started living in that large cell, right, bache? So with the time it evolved. It is like that, right? Just let's say one large bacterial cell, one small bacterial cell, it has engulfed this. The smaller cell has started living within that large cell, right, and they formed a kind of association, right, in which that small cell is also living within, it is doing its job, and it is having its circular DNA, it is having its 70S ribosome. So these two features are just like the prokaryotes now. That's why we correlate it in this way. Okay? So the structure should be clear to you, and when you talk about these enfoldings, that is your cristae, you will see some structures here known as oxysomes, also known as F0, F1 particle. It is basically helping in the transport of protons, ATP synthase. Okay? These are the names. So here always remember, this is the structure. It is having a base, that is F0. This head and stalk is F1. Okay, so this F0 is basically traversing, right? It is basically in the membrane itself, right? And this F1 is there in the matrix, my dear students. Okay, it is there in the matrix. Okay, so just let's complete it from the NCERT only. As I said, we have already completed it. So if you guys are having any doubt in the respiration in plant chapter, you can discuss about it in detail. You can watch the detailed lecture there. So it's a membrane bound structure, double membrane bound structure, outer and inner membrane is there. So it is dividing the lumen. Lumen means that empty space. It's a let's say it's a membrane bound structure. It is having the lumen. So it is having two aqueous compartments. Right, bache? The outer compartment and the inner compartment. So inner compartment is having your matrix. As I said, outer membrane forms the continuous limiting bounding of the organelle. So inner membrane forms the enfoldings known as cristae. Okay, towards the matrix. Okay, bache, so cristae increases the surface area. They have their own enzyme associated with mitochondrial function. So basically electron transport system, electron transport chain, that protein complexes are present there in the inner membrane there. Okay, so mitochondria is the site of aerobic respiration. You know that ultimately, right, the complete oxidation of the glucose is only possible because of this mitochondria. So pyruvate from, right, glycolysis will occur in the cytoplasm. Glucose will form 2-pyruvate. That pyruvate will move to the matrix there, and in the matrix, that pyruvate will get converted to acetyl coenzyme A. Then it will enter in the Krebs cycle. Again, we have completed it in Game of NEET 2.0 respiration in plant chapter. Right? That acetyl coenzyme A will further go, right? Oxaloacetate will accept it. It will form citrate. You know the trick. Come in and kick scooter fastly move on. Ultimately CO2 and water will form. So NADH2, FADH2, ultimately they will go to the electron transport chain where the terminal acceptor is the oxygen, the water will form and the ATP will form. So cellular respiration, that complete aerobic respiration will be there in mitochondria. Is that clear? Is that clear? Right? So do remember this mitochondria is having 70S ribosome and circular DNA just like prokaryotes. Now, plastids. You will see it in plant cells and in euglenoids. So euglenoids, you remember, it is from kingdom Protista. You are talking about the euglena here. Remember euglena? Spindle organ. No doubt it is not looking like a spindle. But yes, do you remember euglena? Spindle organism. Right, spindle organism having 2 flagella. Right, it's a connecting link. Yes. It is a connecting link, bache. What's that? It is a connecting link in between plants and animals. Right? Because when it is having the chloroplasts, right? So it can do the photosynthesis with the help of that. If no photosynthesis, it can directly with the help of its mouth it can eat the solid food. So it is a connecting link in between plant and animal, right? So euglenoid and plant cells, they have these plastids, right? So these plastids are having specific pigments which are much needed, because we know that plants, they are going to perform the photosynthesis. They are having different colors. It is because of these pigments. Okay, so there are 3 types of plastids. So always remember, the initial structure is same, that is proplastid. That proplastid will further form 3 different types of plastid. Your chloroplast is there, your chromoplast is there, leucoplast is there. So this leucoplast is present in the colorless. It doesn't have pigment. It is meant for storage of things, right? It is meant for storage of things. So it is having a myeloplast, a neuroplast, a lyoplast. So basically it is present in the colorless part of the plant. It doesn't have pigment. It is meant for storage. So if it is amyloplast, it will store carbs, your carbohydrates. If it is aleuroplast, it is going to store proteins. It is elioplast, it will store oil or fats. So chloroplast, green colored, it is giving green color to the plant. It is having chlorophyll, much important for the photosynthesis. Chroma mane color, it is having different pigments which are imparting different color to that plant. Right, so I'm not going to explain it in detail. Here also you will see that this is also a semi-autonomous organelle having circular DNA, and 70S ribosome is there. 70S ribosome is there. Is that clear? Right. So here you guys can see the chloroplasts contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy, which is important for the photosynthesis. So in chromoplast, when it is chroma mane color, it is having carotenoids, जैसे carotene. Carotene is giving the color to the beetroot, to the carrot, जैसे beet, beta carotene. So xanthophylls, which are responsible for yellow color, they are present. Right, so they are going to give yellow, orange and red color to the plant. And leucoplants, they are colorless, right? They are of different shapes. They store nutrients. जैसे amyloplast will store the carbohydrates, जैसे कि potato. Elioplast will store oils and fat, and aleuroplast will store the proteins. ठीक है? Aleuroplast will store the proteins. Is that clear? Is that clear? So majority of chloroplast of the green plants are found in the mesophyll cells, बच्चे. So again, for your quick revision, when you talk about the plants, you're talking about the leaf. When you take its transverse section, when you talk about its ground tissue or the cortex, for that we use the word mesophyll. We use the word mesophyll, right? Just say, let's say if it is a dicot leaf, that type of cells are going to be there. Yes. Right, जैसे palisade parenchyma, spongy parenchyma, mesophyll cells. So they are having the chloroplast, and that is responsible for what? That is responsible for the photosynthesis. Right, that is responsible for the photosynthesis. So when you talk about the chloroplast, बच्चे, again double membrane bound organelle. See, in between there is the intermembrane space. In between there is the intermembrane space, or we can also call it as periplastidal space. Periplastidal space. Okay, periplastidal space it is. This matrix here is known as stroma. In the case of chloroplast, you will see some membrane-bound sac-like structure which will stack over each other. Membrane-bound structures which will stack over each other. Let's say this. This is your thylakoid. Thylakoid. It is a membrane-bound sac-like structure. It is having the lumen here. It's a membrane-bound sac-like structure. It is having the lumen here. So when these stacks, when they pile over each other like this, like this, like this, like this, so this structure is granum. Okay, so plural here is grana. So number of grana in a chloroplast cell can vary, number of thylakoid can vary. Right? right? Again, in the chapter photosynthesis in plants, we have discussed that, right? We have discussed that. So we have the stroma here, and this, what's that? Thylakoid, thylakoid, thylakoid. So they will stack over each other. They are forming the granum. They are forming the granum. And these two grana, they are connected to each other. This is your intergranal thylakoid. It is also known as stroma lamella or stroma thylakoid. For thylakoid, we also use the word lamella. And it is also known as fret lamella. It is also known as fret lamella. So fret lamella, stroma lamella, intergranal thylakoid, it's all same. It's all same. ठीक है जी. So बच्चे, in the stroma, it is fully loaded with the enzymes for the dark reaction. So actually in the stroma, your CO2 will be reduced to the sugars, and that light reaction will be there in the thylakoid membrane. It is having that protein complexes. Okay जी. बाकी circular DNA 70S ribosome is again present here. So it is like this, and yes, you should definitely know about this. So there is one question, many teachers were saying that is a controversial question, but it was not. So I'm going to discuss that with you all. They say when you talk about the mitochondria, देखो, this outer membrane is more permeable than inner membrane. Outer membrane is more permeable than inner membrane here. What's the point, bache? Outer membrane is more permeable than the inner membrane. Remember this thing. So why? Because here you will see some pore-like structures. Some proteins are forming pore-like complexes. We call them as porins. If water passes through it, and even some, ions, say even ammonia or something, you can call them as aquaporins. Okay? If water will pass. And inner membrane, it is selectively permeable. Right? It is selectively permeable. So outer membrane is more permeable here in comparison to the inner membrane. In the case of chloroplast also, the story is same. The story is same. Ki outer membrane is more permeable. It is more permeable than inner membrane. Outer membrane is more permeable than inner membrane. Look, that is a straight away. That is a fact. That is a fact. Okay, in both the cases. But there is one more point. I think it is mentioned here only, or it was there in the. It was there in that physiology, right? So when you compare the chloroplast and mitochondria, right? Tell me what kind of changes are there. Tell me what kind of changes are there when you compare the outer membrane of chloroplast and mitochondria. Yes? Anybody? Anybody in the class, please tell me. Please tell me. देखो, overall तो it is a fact. मतलब you are not interested in answering the question. Overall तो it is a fact that outer membrane is more permeable than inner membrane in both the cases, because here also you will talk about the porins, right? You will talk about the porins, right? I'm very bad in that. Okay, you are talking about the porins, but still we compare, right? Still we compare these two, right? These two membranes also. So do you have any idea of it? Yes, do you have any idea of it? Anybody in the class? Do you have any idea of it? Please tell me if you know anything related to this. If you know anything related to this, please do let me know, guys. Do let me know if you know anything related to this about the membrane, because this is important and this is very important for us to know. This is very important for us to know. It is given in NCERT and even we have covered it, right? When we were discussing that plant physiology thing, we discussed that. So you have to. It is semi-permeable. Yeah, ultimately, देखो, inner membrane is more. It is selectively permeable. Outer membrane is kind of freely permeable when you compare it with the inner membrane. That is a fact in both the cases. But if I have to compare mitochondria's membrane and the chloroplast membrane, in that context, I'm asking if we have to compare. Yes, if we need to compare the mitochondria's membrane and the chloroplast membrane, that's what I'm asking. That is what I'm asking. Give it a try. Give it a try. Yes, please give it a try. Anybody? Arey yaar, it is one of the most important topics. Give it a try. No idea? Chaliye, even I am not going to discuss it then. If you people are not interested, then why should I? है ना? Why should I? चलो. Guys, it's simple. ये देखो, like mitochondria, the chloroplasts are also double membrane bound. Of these two, inner chloroplast membrane is relatively less permeable. मतलब, overall, it is a fact. Keep two organelles separate. Outer membrane of chloroplast and mitochondria, it is comparatively more permeable than inner membrane. Yeah, we can say that it is comparatively freely permeable than the inner membrane. Inner membrane is selective. Outer membrane is having the porins. That is why they are more permeable. But when you compare chloroplast and the mitochondria, when you compare chloroplast and the mitochondria, inner chloroplast ka membrane is relatively less permeable. Right, it is comparatively less permeable. When you compare the inner, right, they are saying of the two, the inner chloroplast ka membrane is relatively, matlab you are comparing it with the mitochondria. So comparatively it is less permeable. That is what I am saying. Okay, that is what I am saying. Okay, right. So this is very important. Last year, right, in NEET 2024, one question was there. So lens-shaped chloroplast, spherical, discoid, or even ribbon-like chloroplasts are there. Again, we have discussed it in the chapter, right, bache, photosynthesis. So you have to note down the examples there only. I'm not going to explain it again. We are running out of time. So their number varies from 1 per cell of Chlamydomonas to 20 to 40 per cell in the mesophyll cells, right? So the space limited by inner membrane of chloroplast is called the stroma. So number of organized membranous sac called the thylakoids are present in stroma. They are arranged in stacks. I told you about the grana, about the intergranal thylakoid. Okay, so stroma is there where the reactions will occur. The dark reaction, the carbon dioxide will be reduced there. So ribosome of chloroplast are also smaller than the cytoplasm. So cytoplasm mein you have 80S ribosome, but in the chloroplast and in mitochondria, it is 70S ribosome. Okay, so let's discuss the ribosome and then let's move to the cytoskeletal element, centrosome, cilia, flagella, and then the nucleus thing. So for me, the most important thing after the ribosome is the cytoskeletal element. Let's finish the cytoskeletal element properly, because nucleus part, the chromosome part, we can discuss in the next chapter. Jaise day after tomorrow, you will have the next marathon of cell cycle and cell division. Right? We can discuss it there. Okay. Obviously बच्चे chloroplast in the stroma, the dark reaction will definitely occur. We do not have choice. Mr. Crescent Polyclinic, definitely. ठीक है, चलिए. So now let's discuss the ribosome. Right, now let's discuss what? Let's discuss the ribosomes. So you already know about it, right? You already know about it. It's a universal organelle. It's a non-membrane bound organelle. It's a non-membrane bound organelle. It's a universal organelle. What kind of organelle it is? It is the universal organelle because it is present in the prokaryotic cell as well as in the eukaryotic cell. So ATS will be there in the cytoplasm of the eukaryotic cell, and 70S ribosome is the organelle ribosome. So if I have to be specific, we are talking about the mitochondria here, and we are, yes, talking about the mitochondria here and the chloroplast here. Are you getting my point? So Mr. George Palade, right? Their granular structure observed under electron microscope as dense particle by George Palade in 1953. Okay, so it is having ribosomal RNA and proteins and not surrounded by any membrane, right? Not surrounded by any membrane. Is that clear? Is that clear? So when you talk about the ATS ribosome or 70S ribosome, I told you smaller subunit and larger subunit, they will only join at the time of protein synthesis with the help of magnesium ion. So when it is the ATS ribosome, larger subunit and smaller subunit is definitely there in both the cases. Here also larger subunit and the smaller subunit is definitely there, right? It is definitely there in both the cases. So larger subunit mein here you have 60S, here you have 40S, here you have 50S and 30S. So as I said, no need to add it ki 50 plus 30 is 80. So this S is actually the Swedberg unit. This is actually the sedimentation coefficient. I told you na, cell fractionation is a technique in which basically the cell, right, we are going to rotate it in a high speed centrifuge, its components will break, and as per their density they will settle down. So on the basis of their density we are considering it. So sedimentation coefficient it is. So Swedberg unit, or it is the sedimentation coefficient. So your ribosome, it is made up of ribosomal RNA and some proteins are there, right? So when you talk about the ribosomal RNAs, when you talk about what? When you talk about the ribosomal RNA, so can you tell me which ribosomal RNAs are present here? Can you tell me which ribosomal RNAs are present here? Just say it is 60S. So here you have 28S ribosomal RNA, you have 5.8S ribosomal RNA, you have 5S ribosomal RNA. And for 40S, it is going to be 80S ribosomal RNA, and definitely proteins are present. You should know this, right? You should know this. Yes? Done? Here, 23S ribosomal RNA is present and 5S ribosomal RNA is present. And here, it is going to be 16S ribosomal RNA, and of course proteins are there. So when it is the ATS ribosome na, right, when it is the ATS ribosome na, so it is having 40% of ribosomal RNA and 60% of the proteins. Imagine, ribosomal RNA is less, proteins are more here. Okay? But on the another side, this in the case of this 70S, it is opposite. Okay, so these are the smallest cell organelles. They are the protein factory of the cell. Understood? Chaliye. So next thing, ये देखो, look at the structure, right? So do not forget about it. Okay, so next thing is the cytoskeletal element. So guys, I need your 1 hour and the chapter will be over. Yes, I need your 1 hour and the chapter will be over. In that 1 hour, we will finish microtubule, microfilaments, intermediate filaments. We will finish what is the centriole and the basal body. Then we will talk about the cilia and flagella structure. And I have seen students making the mistake there. So after completing it, I will just discuss the microbodies, peroxisomes and glyoxysomes. That's it. And then the nucleus part. If we will get time in that 1 hour, we'll complete it. Otherwise, we will discuss it later in the next class, because next class is cell cycle and cell division, and we will talk about the interphase nucleus there and nucleolus thing there. So we will cover it there, right? We will cover it there. So we have to discuss the cytoskeletal element. Just in our body, you know that we have this bony skeleton. We have this, over 206 bones are there, right? 80 bones are there in your axial skeleton, 126 bones are there in your appendicular skeleton. So it is providing us a structural stability. It is giving us a framework. The skeleton is required for the attachment of the muscles. It is giving our body a shape. Now when you are talking about a cell, the cell is not having any bones. The cell is not having any kind of bones in it, right? But it needs something which can provide it a rigidity, right? Which can give it a proper structure. So here we talk about microtubules, we talk about intermediate filaments, and we are going to talk about the microfilaments, which are also known as actin filaments. Okay, so I need your another 1 hour and the topic will be over. Okay, I need your another 1 hour and the topic will be over, and it is one of the most important topics, so we should not miss it. We should not miss it. Is that clear? Chaliye, so I am giving you a break for 10 minutes. So let's meet at 1:30 a.m. Okay, 10 minutes ka break. Fine. Hello, welcome back. Hello. So welcome back, guys. Welcome back. Am I audible? If it is a yes, do let me know in the chat section, guys, and please show some energy. See, I'm on time today, right? Isn't it amazing? Today I am on time. Okay, so guys, now we need to talk about the microtubules, the intermediate filament, and we need to discuss about the microfilaments. Okay, these are the cytoskeletal elements. They are going to support the cell. They are going to provide it the structural stability. जैसे microtubules, बच्चे, they are in return made up of tubulin protein, right? Obviously, it is related to the structural stability, rigidity, the framework only. So it is going to do what? It is made up of tubulin proteins. I'll give you one example. जैसे microtubules, obviously there is no need to enter in the depth here, but still, first of all, they are non-contractile. They will not undergo any kind of contraction, right? Just say your microfilament, your actin filament, along with myosin, you know that they are contractile. They can undergo contraction, relaxation, contraction, relaxation. But we do not have anything like that in the case of microtubules. They are non-contractile. So this microtubule is in return made up of tubulin protein. It is made up of what? It is made up of tubulin protein. जैसे you will see that alpha tubulin, beta tubulin, beta tubulin, they will polymerize here. जैसे here they are showing addition, right? They will polymerize. Right, बच्चे? They will polymerize, and when, so they are going to form protofilaments. That alpha beta tubulin proteins, they are going to polymerize and they are going to form protofilaments. Matlab that each filament is the protofilament. And when many protofilaments together, many protofilaments of this tubulin protein together are going to form the microtubule. Just in each microtubule, you can say approximately 13, right? Basically 13 protofilaments are there. It is like this. So what is it? It is the microtubule. It is the microtubule, right? So it is the microtubule. As I said, it is non-contractile in nature. It is non-contractile in nature. It is having 13 protofilaments, right? It is having 13 protofilaments. And bache, you have to focus on the diameter there. Matlab, its diameter is around, yes, right? It is, diameter is around 25 nanometer. So here you will see that these protofilaments, they are helically arranged, right? These protofilaments, they are helically arranged, right? This microtubule is hollow. 13 protofilaments are there. This is your microtubule. This is the microtubule, the cytoskeletal element. Very important it is. Very important it is, isn't it? Right, bache? So when you talk about the spindle apparatus, who's going to make it? Your microtubule. Who's going to make it? Your microtubule, isn't it? Isn't it? Yes or no? Yes or no? The centriole, it is, who's going to make it? Microtubule. Basal body, who's going to make it? Microtubule. Right, cilia, flagella in the case of eukaryotes, who's going to make it? Microtubule. So this is how it acts. This is how it works. Is that clear? Is that clear? Now, just have a look of this particular picture. It's a perfect one according to me, right? Here it is showing different types of cytoskeletal element. These are the cells here, right? Just under. Now, microtubules we have already discussed. Alpha, beta, tubulin, they will polymerize, helically arranged. 13 protofilaments together are forming this one microtubule. Diameter is approximately 25 nanometer. Now, when you look at this particular cell, here, just beneath the plasma membrane, this, what's that? What is it? These are the actin filaments. What is this? These are the actin filaments. They are also known as microfilaments. Dekho, if you focus on their names, microtubules, microfilaments. So microtubule and microfilament is different. Microfilament is your actin filaments. And then comes your intermediate filaments, IFS. So it depends upon their diameter. जैसे intermediate filaments में diameter is 8 to 10 nanometer. It is intermediate. जैसे microtubule 25, intermediate filament 8 to 10, microfilaments very less 7 nanometer. ठीक है? So intermediate filaments, right? See, their position is also marked. जैसे ये देखो, This. So just below this plasma membrane, this is the actin filament. They are bache contractile. It is the cytoskeletal element which is contractile in nature, which can undergo contraction and relaxation. Contraction and relaxation. So it is made up of your actin and myosin protein. Right? Actin and myosin protein. Right, bache? Right, bache? So this actin filament, it is responsible for cytoplasmic streaming. So what is that cytoplasmic streaming, my dear students? What is that cytoplasmic streaming? It is cyclosis. It is cyclosis that we see in the case of eukaryotes. So for that cytoplasmic streaming, your microfilaments are required, right? The pseudopodia formation, जैसे amoeba के case में that pseudopodia in WBCs, that pseudopodia, how is it going to form? This microfilament, a very important previous year question, right, बच्चे? Right, बच्चे? Muscle contraction में this microfilaments are playing role. In the cell endocytosis or exocytosis में your microfilaments are going to play the role. Is that clear? Is that clear? Right. Now when it is the intermediate filament, different proteins जैसे keratin, vimentin, some desmins, there are various proteins, etc., etc. So here in this case, what you have to remember, just say you have the nuclear lamina. Below the nuclear lamina also, you will see intermediate filaments are present. And if you know desmosomes, do you remember desmosomes? The cell junctions connecting two cells together. They are also, the disc-like protein is also associated with the intermediate filaments. Right, bache? So keratin, desmins, yes. Keratin, desmin, vimentin, right? So it is going to provide stability to the cell and cytoplasm. So it is present. It is present in the skin also, right? Right? It is present in the skin also. So that is how it is going to work. These are the cytoskeletal element. So look at the diameter here, approximately 25 nanometer. Intermediate filaments approximately 10 nanometer. Microfilaments approximately 6 या फिर 7 nanometer. So see these monomers, they will join, they will form this, right? Right. And here the keratin, and see coiled dimers are there. Their alpha and beta thing is there. So ये देखो, again the diagrams. Actin, microtubules, intermediate filaments. So they are making the cell skeletal elements. Okay? So this picture is not that clear, but again in the cell motility, cilia and flagella, for maintenance of cell shape, for chromosome movement in cell division, for organelle movement, microtubules are required. For tension bearing, so it is the compression resisting cytoskeletal element, right? It is the compression resisting cytoskeletal element. What's that? It is the compression resisting cytoskeletal element. It will resist that compression, and this one is tension bearing elements. Tension bearing elements. So change in cell shape, muscle contraction, cyclosis, cell motility, pseudopodia formation, it is going to play the role. And yes, cleavage furrow during cell division, especially during animal cell division, right? When you talk about the cytokinesis, you people will see that actin filaments are playing the role. They are making the contractile ring there. Are you getting my point? They are making the contractile ring there. So intermediate filament, right, for maintenance of shell shape, for anchorage of nucleus and certain other organelles, for the formation of nuclear lamina. This point is very important. This point is very important. So this is about the cytoskeletal element. ठीक है जी. Now, next thing. अरे यार, centriole. Let's discuss the centriole. What we need to discuss? Yes, Bache Vrinda, guys, are you up or are you people sleeping? Yes, are you up or are you guys sleeping? Tell me quickly. Are you up or are you guys sleeping? Hello, wake up. So centriole, as I said, it is a non-membrane bound organelle. What's that? It is a non-membrane bound organelle, right? Centriole. You are going to see it in the case of animal cell. Very few plant cells can also have this, but animal cells are going to have this. Right. Now, bache, when you talk about the centriole, I'm repeating this again. It is very, very, very important for the spindle formation. Right. Not the centriole, I'll tell you what's the story. When two centrioles, when they are perpendicular to each other, plus when they are covered with the pericentriolar material, that is a kind of amorphous substance, right? Plus two centrioles when they are covered with the pericentriolar material, what are they? They are known as, जैसे ये देखो, right? They are covered with the pericentriolar material. This structure is known as centrosome, right? This structure is known as cell center. This structure is known as cell sphere. So there is a difference in cell center, in centrosome, in centrosome and centriole. So centriole is normally single centriole, but when you have two centrioles perpendicular to each other, covered with pericentriolar material, it is a centrosome, cell center, cell sphere. And yes, we can also call it diplosome. We can also call it diplosome. Now when you talk about the centriole, bache, it is made up of microtubules. Microtubules are going to make it. It shows 9+O arrangement and its structure looks like a cartwheel. This is the PYQ that I'm telling you, right? It is the previous year question. These are the previous year questions that I'm discussing right now. Your centriole, right, made up of microtubules. It is having 9+O arrangement. 9+O arrangement. Its structure looks like a cartwheel. Its structure looks like a cartwheel. Important information, right? What's that? It is the important information that we cannot miss. It is the important information that we cannot miss, right बच्चे? Right बच्चे? चलिए, let's deal with that. देखो, जैसे let's say you are taking a section of the centriole, right? You are taking the section of a centriole. What are we doing? We are taking a section of the centriole. Guys, see, in the NCERT, firstly cilia and flagella is given, but we will talk about the centriole first. ठीक है जी? We will talk about the centriole first, and then we will discuss about the other structure. So you please make sure that you focus here. Okay? So, ठीक है, this is a kind of outline. A, B, and C. जैसे I told you, you will have 9+O arrangement. What is that 9+O arrangement? We are talking about the microtubules here. We are talking about the microtubules here. Just say these 9. This is the microtubule triplet. Triplet, you understand na? Triplet, you understand na? Microtubule triplet at periphery. So, but why do we say 0 here? Matlab no microtubule, doublet, triplet, singlet is there in the center. So, when I am saying triplet, A, then these hollow या C-shaped, B and C will be there. Matlab A is complete, having 13 protofilaments. Yes, A is, yes my dear students, A here is complete. Tell me, A here is complete, having how many? Yes, bache, having how many protofilaments? Having 13 protofilaments. Each and every one here in the class, please repeat it with me. Having 13 protofilament. Having 13 protofilament. Right, bache? Right, bache? Right, but B and C, they are C-shaped. Their walls are associated with A and C. They don't have 13. Now, 9, I'm saying. 1, my diagram is not going to be that good, but theek hai, chalo, let's try to adjust. Matlab it is at an angle. It is not something very straight here. It is at an angle. My diagram is not that appropriate. I'm telling you this thing directly, right? But still, I'll tell you what am I trying to explain here. So first of all, tell me this 9 plus O thing. Please remember that. Please remember that thing. This 9 plus O thing is very important in the case of centriole, even in the basal body. We'll talk about it. Okay? So this is the triplet. Now, you will see A and C linker. It is something like this. Can you see that? It is something like this. Can you see that? Can you see that? Matlab, we are talking about the peripheral. We are talking about the peripheral triplet. We are talking about the peripheral triplet. A, we have B, we have C. We are talking about the peripheral triplet. We have A, we have B, we have C. Now, what do we have here? We have the AC linker, right? It is connecting this A with the C, right? Now in the center, what are we going to see in the center? There is a central hub. There is a central hub. Just let's say this. And from the central hub, you will see some radial spokes. They will arise. They'll join with this, na? Some radial spokes, they are going to arise. You understand this. Everything is written in NCERT. Trust me, everything is written in this NCERT. You just try to understand the structure here, right? You please try to understand the structure here. Okay, ji? Okay, so we are talking about the centriole, right, which is found in almost all eukaryotic animal cell, in some protista, in fungi, and right, and in some eukaryotic plants where the flagellate structures are present, now you will see the centrioles there. Right, so angiosperms, higher gymnosperms, some algae fungi, they don't have, but others are having this. Okay, ji? Right. So triplet fibrils are present, which are made up of 3 subfibers. Is that clear? Observe the diagram. Observe the diagram. Focus on the A and C linker. Focus on the A and C linker. Right, bache? Right, bache? So central part of the region is also having the proteinaceous mass called hub. Right. So this central part. What's that? This central part is hub, and it is attached to the periphery with this, and what do we call it? They are the radial spokes. They are the radial spokes. What are they, bache? They are the radial spokes. So in the center also, there is a hub. It is also the protein, but it is not the microtubule. So don't you think that it is looking like a cartwheel? And looking like a wow. Looking like a wow. It is the cartwheel like structure, yes or no? It is the cartwheel like structure. Yes, it is the cartwheel like structure. So an electron microscope is going to show this 2 short hollow cylindrical, usually lying at right angle to each other, right? Usually they are lying at right angle to each other, right? 2 short hollow, matlab I am talking about the centrosomes there. ठीक है जी, right? So no microtubule in the center, that's why 0 pattern is there, right? Okay, so just imagine this is the centriole. So these triplets, they are tilted. They are tilted to an angle of 40 degree. They are tilted to an angle of 40 degree to the, to that cylindrical portion, right? To the radius of the cylinder, rather. To the radius of the cylinder, these microtubules are tilted. That's why I'm not making it like this, 1, 2, 3. It's a little tilted. I know my angles are not appropriate, but this is just, I just made this rough diagram so that you guys can understand this, right? So that you guys can understand this. So this part clear, that how is it going to form? How is it going to form? So around the centriole, you will see the pericentriolar material. Right, you will see the pericentriolar material, right, bache? So for that pericentriolar material, just that are also 9 in number. That are also 9 in number. It's not like totally covered. That is also 9 in number, the pericentriolar material. We call it as mesiule also. Just ye dekho, this is the diagram. A, B, C, and C and A will join. Right, so a 40-degree angle to the radius of the cylinder. So this is the central hub, again protein. These protein spikes are attached to the A here. These are the radial spokes. So firstly tell me, the structure is clear or not? Firstly tell me, the structure is clear or not? Ye dekho, this thing. Just look at this. Look at this. A is complete, B and C is C shape. So they are covered with 9. 9, right? Pericentriolar material, and we also call it as mesiule. We also call it as mesiule. ठीक है जी? Right. So now in the plant cells, मतलब the plant cells which are having flagellated structure in their life cycle, they still can have this centrioles, but gymnosperms में, higher angiosperms में, higher gymnosperms में, algae and some algae, they do not have this. Right. We will discuss that how in their case cell division is possible, and for that you know that you have to wait for the next lecture. ठीक है? So mesiules are there. These protofilaments clear. So no doubt A is complete, B and C they are going to be what? They are going to be C shaped. So the structure clear. So is it clear why do we call it as a cartwheel? Like so its appearance is like this. Now is it clear why it is 9 plus O? Because in the center there is a central rod that is known as hub, right? And 9 radial spokes are there. They are giving it an appearance of cartwheel like structure. Is that clear or not? Right? Is that clear or not? So remember, centrioles are self-duplicating units, and their duplication is also there in the S phase. Just like the DNA replication, the centriole duplication also occurs in the S phase. Is that clear? Is that clear? Is that clear? Sure? Yes? ठीक है, so these are the centrioles. Now बच्चे, there is something called basal body. There is something called basal body. What is that? There is something which is known as basal body. बच्चे, this basal body is also known as blepharoplast. ठीक है? Right, this basal body is also known as blepharoplast. ठीक है बच्चे? Blepharoplast. I'll give you the example. जैसे this is I'm taking the simplest example. This is your euglena, right? It is a euglenoid having 2 flagella. One is large, one is small. Now these flagella, they are arising from a structure. What is that structure, my dear student? That is the basal body. What is that structure, my dear students? That is the basal body. What's that? That is the basal body. This basal body, as I said, it is also known as blepharoplast. So it is the part from where your cilia or flagella will arise. जैसे this basal body, its structure, its form is just like, you know, centriole, right? It's just like centriole. मतलब its structure, its size, its form, its chemical composition, even its structure, its form, its size, even its chemical composition, is just like centriole. So if it is just like centriole, can I say that it is also having 9 plus O arrangement? It is also having 9 plus O arrangement. But remember one thing. In the eukaryotic cell, the cilia and flagella is having, right? It is those flagella, no, but cilia and flagella both are having 9 plus 2 arrangement. So just imagine, right? Basal body, it is its structure, size, form and everything is just like the centriole, 9 plus O arrangement. So even but at times this basal body can form centriole if required, right? They can convert into each other. It can form centriole when required. So it is having 9 plus O arrangement, and cilia and flagella is having 9 plus 2 arrangement. It is having 9 plus 2 arrangement. What is it? It is having 9 plus 2 arrangement. So matlab from a 9 plus O arrangement structure, a 9 plus 2 arrangement structure will form, and this is something you need to keep in your mind. And I will show you a diagram here. जैसे ये देखो, this is the diagram. So basal body या फिर centriole is going to, basal body or the centriole is going to form the cilia and flagella. So it is having the triplet, right? 9 peripheral triplets are there. or we can also use the word fibrils here. We can also use the word fibrils here, and these ABC as subfibrils. So they will form cilia or flagella, but that will contain 9+2 arrangement. That's what you need to keep in your mind, right? And why is it so? You will figure out the answer. There is an answer for it. You will figure out, right? So just say, let's say if this is your cilia or flagella, it is having that outer plasma membrane. It is having that inner rod-like structure. That rod-like structure is made up of microtubules, which is showing 9+2 arrangement. So now this is the next structure that we need to discuss. Is that clear? This is the next structure that we need to discuss. Is that clear? Is that clear? Is that clear? It is the next structure that we need to discuss. Is that clear? So again, 9, A, B. A is complete, B is hollow. Oh, 1, 2, 3, 4, 5. It is like this, A or B. Okay? So now this A, it is having arms. I'll tell you what's that. Just say last time we were talking about the A and C linker, and they are equally spaced. My diagram is not appropriate, but they are equally spaced. Okay? So basically the point here is that here you have 9 doublets at periphery. You have 9 doublets at periphery. What do you have? You have 9 doublets at periphery. And in the 2, what is this 2? They are singlets. They are singlets at center. They are singlets at center. Now what's the meaning of that? 2 doublets, and here you will have 2 singlets. जैसे ये देखो, this, and it is covered with a central sheath, and there is a central bridge. So in the basal body, it was 9+0 arrangement. In the center, microtubules were not there, but here you have 2 singlets, right? 2 singlets. Then obviously they are also attached with what? They are also attached to this peripheral microtubules with the help of what? With the help of these radial spokes, right? So again, here also you will talk about 9 radial spokes. Okay, right, bache? So this is the central sheath. So see, these are singlet, central singlet. It is covered with the central sheath. Yes, it is covered with the central sheath. So A is complete, B is semi. So see how they are joined. AB linker is there, right? A and B linker is there, right? Now when I'm drawing it, can you see these white colored arms? These white, but it is of dynein protein. Just say outer dynein arm. There is a protein dynein. So this one is the outer dynein arm. This one is the inner dynein arm. One is the outer dynein arm. This one is the inner dynein arm. So this dynein is what? This dynein is an active ATPase. It will hydrolyze the ATP, and that energy is used. That energy is used for the movement. जैसे sperm के case में flagella is there, 9+2 arrangement, so ATP is required. This dynein protein is going to hydrolyze that ATP. The energy released will be used for that further movement, for that flagellar movement. It is like that. It is like that. So outer dynein arm, inner dynein arm is there. And now this A and B, this A and B linker is made up of nexin protein. right? This is the protein which is joining it. So that is the 9+2 arrangement. Tell me if it is not clear. I'll explain it again. Tell me. So A tubule is having the dynein. It is just like myosin of mother cell. It can hydrolyze the ATP. Energy is released for the movement. Okay. So nexin protein is going to connect it. Okay. So central sheath, central bridge is there, right bache. So 9 radial spokes are there. So peripheral doublets are connected by the linkers, right? So that is all about the structure here. So if you guys are having any doubt, please feel free to discuss that. ये देखो, look at the structure. Two centriole perpendicular to each other, covered with pericentriolar material. 9+O microtubule arrangement. Your cilia and flagella. So जैसे let's say if it is a flagella, right? Though it is a cilia, चलो outer membrane and in between this cylindrical structure, this rod-like structure. जैसे in the case of flagella, I don't know whether you know it or not. This is the plasma membrane. This central rod-like structure. What is the word that we use for it? Axoneme. 9+2 arrangement, right? So they are the doublets. Here. Okay. So ये देखो, nexin protein is connecting it. Right. So A tubule is having outer and inner dynenarm. Radial spokes are there. So any doubt in the structure, I'm going very slow here so that you guys can understand. Right. So that you people can understand. Tell me. So ये देखो, basal body, look at the structure there, then transitional zones are there in between. Look at the changes here, and finally this is what you are going to get. Okay, so please observe this diagram. It is one of the best diagrams, right? It is one of the best diagrams. Do check it. Done, bache? Sure? Okay. Oh, sorry. So cytoskeletal element and cilia and flagella, centrosome all done. Now we will talk about the nucleus, chromosome and the microbodies. Sure. Then we'll complete the chapter. So cytoskeleton, elaborate network of filamentous proteinaceous structure having this. Again, it is discussed in detail. Now, bache, cilia and flagella they have given first, and look at the structure. Two central doublets. There is a central bridge, the central sheath. These are the radial spokes. So in detail, I have explained it. Right? Read it from the NCERT, and trust me, you are going to find it amazing. So cilia and flagella is having 9+2 arrangement. Basal body and centriole is having 9+O arrangement. Right? It is having 9+O arrangement. So cilia, singular is cilium. Flagella, plural is flagellum. They are hair-like outgrowths of cell membrane. You know that cilia is comparatively smaller, right? It's just like, it works like oars, right? Causing the movement of either the cell or the surrounding fluid, right? Either the cell or the surrounding fluid. Are you getting my point? So it is having a kind of pendular movement, right? Pendular movement, isn't it? Right? Done, bache? Okay. So flagella are comparatively longer, responsible for cell movement. So prokaryotic bacteria is also having the flagella, but that flagella is made up of flagellin protein. It's a simple kind of flagella, but here it is made up of microtubules. Okay, 9+2 arrangement. So electron microscopic study of the cilium or the flagella shows that they are covered with the plasma membrane, and that core, that cylindrical part in between having 9+2 arrangement is the exome. So it is having number of microtubules running parallel to the long axis. So it is having 9 doublets of radially arranged peripheral microtubules, pair of centrally located microtubules. So 9+2 arrangement is there. So I'm not going to discuss that again, right? Because I have already covered it. Okay, I have already covered it. So now let's move to the next thing. That is again a double membrane bound organelle, and that is your nucleus. That is your nucleus, right? That is your nucleus, the main, right? The main structure which is going to control the cell activities. Okay, what is it? It is the nucleus. What is it? Nucleus. So can we call it as a director of cell? Definitely we can call it. It is having the genetic material. So let's revise the things, right? So nucleus, who discovered it? Who discovered it? Obviously Robert Brown, right? And in the root cells, in the cells of the root of orchid plant. So do not forget about the discovery. Okay, so it is a double membrane bound organelle. It's a double membrane bound organelle. So when you look at its membrane now, students, outer and the inner membrane is there. So this outer and inner membrane at places they will fuse. They will fuse, and at that places there will be some nuclear pore complexes. Nuclear pore complexes. So outer membrane, inner membrane, at places it will fuse. It will form nuclear pore complexes, which is important for the movement of things outside and inside. Right, outside and inside. And now this is the nuclear lamina, and I told you who's going to help in the formation of this nuclear lamina. Who's supporting this nuclear lamina? Very simple, your intermediate filaments, right? Just now we covered it. It's very simple. It will be your intermediate filaments. It will be your intermediate filaments. Now in the nucleus, you know that outer membrane is in continuous with the rough endoplasmic reticulum, and even, my dear students, some content of this space, the space is also having a kind of fluid which will get exchanged there, which will move here, and here within the cell you will see, within the nucleus you will see chromatin is there, right? Chroma mane color. So we have to discuss it. So let's move to the NCERT directly. So here we have the structure, my dear students. So nucleus, that is a cell organelle described by Robert Brown in 1831. Later, the material of the nucleus was stained by a basic dye. It was named chromatin by Fleming. It is important. So you know that human DNA, bache, human DNA is approximately 2.2 meter long, right? Now we have to pack that structure in a very small nucleus, right? In a very small nucleus that is also microscopic. So in this particular nucleus, if we have to fit it in, then it is a DNA, right? DNA is a nucleic acid and it is negatively charged. Right now I'm not talking about the structural details. We will discuss that in biomolecules, but still I'm telling you it is negatively charged. Why? Because of the presence of phosphoric acid there. It is ic, phosphoric acid there, right? That acid is giving it the negative, this negative charge. Now it will coil over, because we are talking about the eukaryotes, not the prokaryotes. It will coil over the histone proteins. That histones are positively charged. So when your chromatin, when it coils over that histone proteins, it is forming the chromatin. When your DNA is coiling over the histone proteins, it is forming the chromatin. So chromatin is basically the DNA and protein. DNA and protein together, they are forming the chromatin, the thread-like structure. So when a nucleus or when a cell is not dividing, right, then its nucleus is in the resting stage. We use the word interphase for this, and that things you will understand when we will discuss the next chapter, that is cell cycle and cell division. That is cell cycle and cell division. So when you look at this nucleus now, you will see the nucleoplasm, you will see the membrane, you will see the nuclear pore complex and nucleolus. But this nucleolus is not something which is membranous, membranous. No, it's not like that. Let me explain. So who discovered the nucleolus? Who discovered the nucleolus? Fontana. Who discovered the nucleolus? Fontana. So nucleolus is considered as the ribosome factory. It is considered as the ribosome factory. Why? Because it is going to form ribosomal RNA and proteins. Matlab it is going to form the structures which are required for the synthesis of ribosome. So it is the ribosomal factory. So it is not something which is covered with the membrane. No. And the plural of nucleolus is nucleoli. Do not confuse it with nucleoid. Right? Do not confuse it with the nucleoid. That is something different. That is something different. So nucleolus, it's not covered with any membrane. It is a kind of structure which you will see there. Jaise ye dekho, it is given here ki interphase nucleus. What is the meaning of interphase nucleus? When your cell is not dividing, when it is in the resting stage, then you are not going to see the chromosomes like this. Yeah, like this. They are not compact. The DNA is simply coiled over the proteins, right? And it is showing some thread-like structures. We are calling it as chromatin. So it is the nucleus. It is the nucleus. It is the nucleus of what? It is the interphase nucleus. It is in the resting stage. The cell is in the resting stage. It is not dividing. Okay, ji? So it has highly extended and elaborate nucleoprotein fibers called chromatin. So nucleoprotein mane DNA and protein. DNA is the nucleic acid. So nucleoprotein complex. And one or more spherical bodies, singular is nucleolus, plural is nucleoli. So electron microscopy revealed that nuclear envelope is having two parallel membrane. It is having a space 10 to 15 nanometer. It is the perinuclear space. So it's a barrier in between the material present inside nucleus and outside. Very common thing that we have just covered. Okay. The outer membrane remain continuous with the rough endoplasmic reticulum, right, bache? And it is also bearing the ribosome. So at a number of places, nuclear envelope is interrupted by minute pore, that is nuclear pore complex, from which the RNA and protein molecules will take out. They will move. Okay. So usually there is only one nucleus per cell, but sometimes bache, that karyokinesis is not followed by the cytokinesis, and we can see multinucleated condition. So we call it as cynocytic condition. Multinucleated condition. Multinucleated condition. Is that clear? Is that clear? Right? And some mature cells, they do not even contain nucleus. Just say your mammalian RBC, sieve tube cells. They do not have nucleus. Important information. So nuclear matrix is the nucleoplasm having nucleolus and chromatin. So nucleolus, you know that they are continuous with the rest of nucleoplasm. They will synthesize ribosomal RNA, right? So larger and more numerous nuclei are present in the cell which carry out protein synthesis actively. That's it, that's all. And this is what you need to know about it. Okay. So bache, when your chromosome, jaise ye dekho, this is your chromatin. This is your chromatin, the thread-like structure. So when a cell will undergo division, these chromatin threads, their further coiling will occur. That we discuss in class 12, packaging of the DNA. And we will see that chromosome will become compact like this. That chromosome, it will become compact like this. Are you getting my point? It will become compact like this. And if you are thinking this is not a chromosome, this is a chromatid, no, it is also a chromosome, my dear students, and it is also a chromosome. So that if you know the difference, if you are a regular student of mine, then you know the difference. Otherwise, in the next class, after S phase, after cell cycle phase, I'll make it clear to you. So this is also a chromosome, right? So DNA and proteins, some histone proteins, non-histone proteins, they will just compact, they will just compress, and they will just become condensed, and they will form structure like this, right? So here this central part, this, it is the primary constriction. It is the primary constriction, and it is known as centromere. Centromere. And this centromere from both the sides, it is a primary constriction centromere to which these structures are attached. So from both the sides, it is covered with a disc-shaped protein, right? It is covered with a disc-shaped protein, and that disc-shaped protein is kinetochore. That disc-shaped protein is kinetochore. What's that, guys? That disc-shaped protein is what? It is the kinetochore. Are you getting my point? That disc-shaped protein is what? It is the kinetochore. And bache, this kinetochore, so during cell division, that spindle fiber, that mitotic fiber, that spindle fiber will attach to the kinetochore. It never attach directly to the centromere. It never attaches directly to the centromere. It will attach to the kinetochore. Are you getting my point? It will attach to the kinetochore. Are you getting my point? Are you getting my point? Right. So this is also a chromosome. It is the chromosome after S phase, my dear students. Now it is having the arms. So these arms, because it is attached to the same centromere, these are the sister chromatids. These arms are the sister chromatids. These arms are the sister chromatids. And bache, other than primary constriction, there is one more secondary constriction is there. And for that secondary constriction, what you need to keep in your mind? It is not having anything, it is non-staining. We cannot stain it. It is non-staining. We cannot stain it. Okay? So this is the kinetochore, the disc-shaped protein. So cell will show the chromosome, bache. So chromatin contains DNA and some basic protein called histone. I told you already. So in class 12th also we will discuss it. So these histones are positively charged proteins. They are positively charged proteins. They are fully loaded with the basic amino acid like your lysine and arginine. Again a previous year question. They are fully loaded with your basic proteins, that is lysine and arginine. Right, bache? There are some non-histone proteins and RNA also. So single human cell has approximately 2.2 or 2 meter long thread. So we have to pack it. Total 46 chromosomes are there, 23 pairs. So I believe this part is clear to you. So every chromosome is visible only in dividing cell. It is having the centromere and kinetochore is present. Right, bache? Right. So based on the centromere, the chromosome is divided. Right, in this category. Just see here. First of all, this is your secondary constriction. So it is non-staining. Remember, it will not get stained. It is non-staining. So, bache, if your centromere is in center, it is going to be metacentric chromosome. Okay? If it is a little away from the center, just say one arm is short, one is long, it is your submetacentric. It is your submetacentric. What is it, bache? It is your submetacentric chromosome. Any doubt? Any doubt here? It is your submetacentric chromosome. Yes, it is your submetacentric chromosome. Is that clear? Is that clear? Yes. So then it is acrocentric when your centromere is near the tip. It is not at tip, near the tip. And this one is at the tip. It is at the tip. So it will be telocentric. It will be telocentric chromosome. So centromere is in the center, metacentric, little away from the center, submetacentric. Matlab it is located near the center or midpoint of chromosome. So it is having short arm and long arm. So remember, your short arm, we denote it with P, and the long arm, it is denoted with Q. Short arm is denoted with P and the long arm is denoted with Q. Is that clear? Done? Done? Sure? Yes, students, please tell me, is it clear? So 1, 3, 16, 19, 20th chromosome. First, third, 16th, 19th and 20th chromosome of humans. I'm specifically talking about humans. They are metacentric. They are metacentric. Right, bache? Right, bache? Yes or no? Is that clear? So chromosome number 13, 15, 20. Chromosome number 13, 15, 21st is acrocentric. So telocentric chromosomes are absent in humans, right? So P and Q, if it is not mentioned in NCERT, that doesn't mean you are not going to note it down. It is important, right? So 13, 15, 21 and even 22, these chromosomes are acrocentric, right? And chromosome, okay, it is your acrocentric, right bache? So chromosome 2, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? So basically you can say from 2 to 12, right? Chromosome from 2 to 12, right? From 2 to 12, and other than that 17, 18 or X, they are submetacentric. So remember, telocentric chromosomes are absent in our case, right? They are not present in our case. Is that clear? Is that clear? So second, no, third is there na, not second to 12th, my bad. Second, 4, 1 second. Second, then 4th to 12th. That's how you have to write. Second, 4th to 12th, then 17th, 18th and X chromosome. And X chromosome, they are submetacentric. Right, bache? They are submetacentric. Is that clear? So 1, 3, 16, 19 and 20, they are metacentric. So we do not have the telocentric chromosome. Okay, we do not have the telocentric chromosome. So that's it, that's all. So shorter arm, longer arm, done. So about the chromosome, about the homologous chromosome, non-homologous chromosome in detail, even about the nucleus, even about the interphase nucleus, the condensed chromosome and all, we will discuss in cell cycle and cell division. So ye dekho, sometimes few chromosomes, they have non-staining. See, I am highlighting this part again and again. Non-staining, matlab it is not getting colored. This is an important question. So it is the secondary constriction at a constant location. Okay, it is the secondary constriction at a constant location. So it will give the appearance of a small fragment called the satellite. You should know about it. Now, bache, microbodies, they are again membrane bound structures. Okay, they are minute vesicles, very small structure. They have various enzymes. So here in microbodies, you talk about the peroxisomes, and animal peroxisomes and plant peroxisomes are there, and we talk about the glyoxysomes. These are the examples of microbodies. They are present in plant cell only. So they are present in both animal and plant cell. So it plays an important role in the beta oxidation of the fatty acid, right? Beta oxidation of the fatty acid, you should know about it. So first of all, write down, yes. Done? Okay. Yes, bache, is that clear? Okay. So now when you talk about the peroxisome, they contain both the enzymes, peroxidase as well as catalase. It is concerned with the hydrogen peroxide formation. You know na, H2O2, hydrogen peroxide, it is dangerous. So for its synthesis, peroxidase enzyme is there, but in the same organelle, there is an enzyme which will degrade it also, right? Catalase is also there. Catalase is also there. It is going to break it. It is going to break it into water and the oxygen. Are you getting my point? Are you getting my point? So catalase will degrade the H2O2, that is hydrogen peroxide, into hydrogen water and the oxygen. Is that clear? Is that clear? So in plants, if you remember, bache, in plants, peroxisomes, do you remember this? Peroxisomes are concerned with the C2 cycle. They are concerned with C2 cycle, that is your photorespiration, a wasteful process. Do you remember that? Chintu, Papa, Mummy, chloroplast, mitochondria, peroxisome. They are involved in photorespiration. They are involved in photorespiration. And this peroxisome, it also helps in the Yes, bache, it is also play role in the, it plays a role in the beta oxidation of fatty acids. Very important. Beta oxidation of fatty acids. So this is important. Okay, beta oxidation of fatty acid, it is important. Okay, bache. Okay. Done. So glyoxysome present in plant cell only. So it is concerned with glyoxylate cycle, right? It's a very special type of peroxisome, and these glyoxysome, matlab in this cycle they'll convert fats to carbohydrate. Matlab ultimately what's going to happen? Fats will be converted into carbohydrates, right? Fats will be converted into carbohydrate. That's it. That's all. So this is all about this particular chapter. ठीक है जी? So in fatty acids, you are going to see the glyoxysomes, which are highly oxidized peroxisomes. ठीक है जी? Done. So this is all about this chapter, right? Initially I was thinking that I'll miss some topics, but still we covered everything there, right? We have literally covered this chapter, and this content is more than sufficient. Now the things that you need to take care is you have to revise the functions of various organelles. You're not allowed to miss the functions of the organelles, right bache? So do practice the question, do solve the question. Let me finish the chapters, let me finish the syllabus. We will start the series in which we will only practice the questions, right? So make sure you revise the topic. Right after the class, I will share the PDF in the Telegram group, and here you know that you can go to our channel, you can see any of the video there. You have to go to the description box, just click on this link and be the part of our batch. So in my batch, I am going to start the class 12 syllabus soon. You will get the times to watch the recorded lectures of class 11. So without wasting your time, if you're still looking for a plan, right, be the part of this batch. And trust me, if you will join, or if you enroll in our batch, it's not like that you will get that particular batch only. No, you will have the access of all the batches on the Unacademy platform. Okay? So right after the class, I'll share the PDF of the notes in the Telegram group, right, in our official Telegram group. Okay? So do join it. That's it. That's all. Thank you so much. See you all on Thursday at 6 p.m. and then we will complete cell cycle and cell division. Okay, we'll complete cell cycle and cell division. So thank you. Take care. Do let me know in the comment section you like the session or not. Thank you so much.